mice that there can be methylation signals and small RNA molecules that transfer environmental information into the offspring. It's a very, very subtle effect. You need very large N numbers to really prove this, and it's probably a minority of the heritability that's going on in genetics. But it's fascinating.
Yeah.
It's one of these things that's brought back the debate between Darwin and Lamarck. This
right.
this whole Darwinian genetics,
nature versus nurture.
Exactly. It's back, and it's good that it is back. It's been a long time. Lamarck was silenced for many years, but the reality is a little bit of Lamarck was right, and that there are aspects of your environment that will get written onto your DNA, and a small portion of that may in fact find its way into the offspring, such that the environment can actually change the genetic outcome in future generations. But again, it's a very subtle effect, and not nearly as powerful as the mutations you might see that are actually written faithfully into the DNA.
Yeah, the thing that pops up in my mind to conceptualize this in a totally different way. My wife is a mental health therapist. It reminds me of the concept of intergenerational trauma. It's like, these subtle things that can, get passed on that relate to how, somebody grew up and their environment and everything. But it's interesting, and I know we're deviating, but I think this is something in general I wanted to ask you about, misconceptions around genetics and genomics and stuff. So the concept of epigenetics is talked about a lot. It's a very it's a big sort of buzzword in popular science, pop culture science, and stuff like that. And so I one of the questions I was going to ask, and maybe just ask you now, what are some of the misconceptions that you hear kind of popularly shared about genetics, genomics, and particularly epigenetics, whether it be related to plants or humans, that you'd kind of like to set straight?
So the scientific field is really excited about methylation for the complete opposite reasons. Cell that is responding to a signal to make something transcribe, and maybe even a feedback mechanism to turn it off. And so we like having those circuits all spelled out. So we're really excited about epigenetics for that reason. But a lot of that often gets pushed into pop culture as being, your actions from 3 generations ago are now impacting the direction of the human population, and there's very, very, very thin evidence for those things. I don't doubt that some of those studies are in fact real, but the Ireland famine is one that comes to mind. But it's such a weak p-value and such a weak effect that you'd be better off worrying about whether you're staring at your phone too long walking into a street. I mean, it's
really.
There's stronger selective pressures out there that are likely to affect your genetics than some of those epigenetic effects. So it's unfortunate, but it happens. The science often gets interpreted politically, and I think epigenetics is one that has a sort of guilt associated with it that's being leveraged politically.
Well, yeah, and for people that are not scientists or have a very vague understanding of the research that's gone into this, it can really freak people out unnecessarily. It can cause a lot of stress, like you mentioned, that really isn't warranted. And another example that comes to mind is concern over different types of waves that are passing through your body, and the effects of gravitational waves and this sort of stuff, that some people, when they start hearing about, like quantum gravity and quantum mechanics is a big one, where people start to hear these concepts and then they get really strung out worrying about these effects of things that, the effects may be real, but they're so small in comparison to other things that have much bigger pressures on you, like you're saying, that it's like it's not really worth exerting the mental energy to worry about that when you have much more real issues to deal with, that you probably don't have to worry about gravitational wave distortions being caused by comets and things moving through the galaxy.
Strung out is a great way to describe string theory.
So going back to the research study now. So, well, I guess one thing I want to summarize for listeners too, just to kind of talk about some of the techniques and the things you've described. So we talked about basic DNA sequencing, in that is looking at the possibilities essentially of the organism, what's there, the foundation to work from. Measuring the RNA allows you to understand what's actually at play within that DNA sequence. And then the methylation piece, being able to look at that, helps you understand how the environment is influencing these things that are at play. Is that a good, super simplified version of explaining that?
It is. And, methylation in many ways is I actually like the RNA data better because there's so much more information content there, and it's more accurately mapped. But the methylation is kind of another confirmation. When you see promoters have methylation patterns that promote the expression of the genes, you have more confidence in actually what's going on. So it's just another view onto the genome. But yeah, that's effectively the central dogma, right? As we have this genome, it is the hand that you're dealt, and if you don't have certain aces in that deck, you're never going to get them, no matter how much light or water you put in that plant. So if you don't have a THC synthase gene, you're not going to coach that plant into making THC some other way.
Yeah.
However, if you do have a THC synthase gene, the gene doesn't necessarily predict that you're going to make 7% versus 14% THC.
Right.
At least that gene doesn't. We're hunting down a couple other genes that are related to the precursors in the pathway that might, in fact, have a bigger impact on the magnitude of expression. But right now, the genetics between THC and CBD, they really just give you the ratio to expect. But they're not going to give you, if you push the magnitude of expression of a given ratio too far, it will break the USDA regulations. And the genetics today aren't going to predict that. I think in a couple of years, we'll know that. We'll know that there's a couple genes upstream that everyone's looking at that are involved, AA3 and AA1, and there's levitolic acid cyclase and parental transfer. All of those things are great candidates for mutations that may, in fact, be driving the magnitude of expression between a 7% and a 14%. But if you have a 14% CBD line, you're going to be risking that popping hot. It's going to probably approach that 0.3% under the given regs that are there. And so a lot of people are unfortunately trying to find 7% lines, grow twice as much material, only so the material, 99% of it ends up going into an extractor where the ratio no longer matters. It's absolute insanity by regulation, but it's what's there. But to the point of your analogy on cards, if you're missing both aces, CBD and THC, you're going to be making the precursors, the kings, if you will. And that's so the genetics can be very, very predictive, and they can be predictive at a very early age, and they can also scale. That's one thing that I don't think everyone yet appreciates. While they probably are never going to give you and never going to be a replacement for an HPLC value, you're probably never going to have a field portable HPLC that can run thousands of samples a second. That will be doable with genetics. It's just a matter of time. But it's much harder to imagine getting that to work with an HPLC system that's field portable. So they're complementary, and we always encourage people not to view them as one versus the other, because there are many more things that you can measure with genomics, and its price points are changing so radically. You should pay attention to it, but they're always going to be complemented by people who have good analytical testing equipment.
Yeah, no, I think that's an excellent way to summarize that. It is all complementary, and even within analytical chemistry, there's sometimes a strange competition between analysts that prefer kind of traditional chromatography and those that like to rely on mass spectrometry. And you've got some people that are very resistant to, they kind of want to stick to chromatography, and that's what they do, and others like really trust spectrometry, and then obviously they blend together. You use mass spectrometry as exactly, but there is a funny, there's within the science world there are these weird little camps and tribes, just like anywhere else, where
and they exist in sequencing too. We all bicker over what's the better platform to use.
Right. Are there any other lessons that that you gleaned from this recent research project that you want to talk about? One thing we mentioned, but I don't know if we really got into it, was the relationship between these cannabinoid acid synthase genes and the pathogen resistance. We touched on it early on, but I don't know if we really got into it.
Yeah. So the ongoing debate in the field is what is responsible for residual THC expression in plants that don't have a THC synthase gene.
Yeah.
Why are hemp plants popping hot? And the number one culprit for that is CBD synthase. Whenever people clone that into yeast and run it, they tend to get like 20 to 1, 23 to 1 ratios of CBD THC. So the gene in yeast will make promiscuous THC.
Mhm.
You can alter the pH of those conditions and even get those genes to make all the other cannabinoids.
Yeah.
In fact, there's a really good presentation I'd recommend people look at. It's a CanMed presentation from this October from Andrew Horowitz from
Mm.
Demetrix. Demetrix is a synthetic biology company out of Jay Kiesling's laboratory, and Jay Kiesling is well known for cloning a lot of the plant genes into bacteria and into yeast. So this freaks a lot of people out, but there's actually really good science that you learn when you isolate a gene into a model organism like
Mhm.
that, because you can perturb it in isolation of all the other cannabinoid genes that are in the cannabis genome. You take one of them out and put it into yeast, and then you can play with the pH and you can play with the temperature and all these other conditions, feed it different precursors and see what happens. And what they did is they fed those yeast hexanoic acid and they got THCP. So they
Yeah.
actually found THCP before we found it in the plants.
Yeah.
The Italians discovered it very recently, but if you actually go through the literature, you realize no, they were making this in yeast, and they learned that the synthase genes in cannabis that we currently have, if you feed it octanoic acid, will make THCP.
Mhm.
So we don't need to go be looking for a different THC gene. It's the same gene will fold THCV and THCP. We need to be looking upstream, maybe in the pathway that's governing what makes hexanoic acid and what makes octanoic acid.
Mhm.
So we're beginning to be able to use synthetic biology to teach us about the plant. We did a very similar thing in this study in that we took a couple of the genes that we saw were related to pathogen response, and we cloned them into E. coli. We took a thaumatin-like protein that was amplified in the Jamaican lion genome and also amplified in a couple samples that we got from Colorado Seeds and from Minibus, Mark Jordan's group. And those ones we put into E. coli. And once we had, that cannabis gene getting synthesized in E. coli, we can make gobs of it and then apply it back to microbes that we know infect cannabis, like Fusarium and
Mhm.
Aspergillus and Penicillium, and see if that peptide actually kills those microbes, at least in a petri dish. And we demonstrate some of this in the paper. It's not killing, we don't have evidence that the TLPs, the thaumatin-like proteins, are killing Aspergillus, but we do see that it's retarding the growth of Penicillium species and of Fusarium oxysporum. The next experiment we have running is grabbing the other gene we found that was correlated with this resistance, which is a chitinase. We have that getting cloned and expressed in E. coli as well. And most of the plant literature shows that if you combine these two, you get a better 1 + 1 = 3
Mhm.
effect. And you really can knock down the microbes once you have both of them present. So we're in the process now of taking those cannabis genes and just making gobs of that protein so we can test out the theory are these the proteins that are responsible for keeping cannabis powdery, are they some of the proteins I should say, not the only ones that are
Yeah.
making cannabis powdery mildew resistant, and can we consider those as like a foliar spray? It's one thing to have the genetics and know how to breed for resistance with this particular form of resistance. That's very helpful, but that
Yeah.
might take time. But there are some jurisdictions, particularly here in Massachusetts, where changing your genetics isn't easy. You have to kind of run with the genetics you put into the seed to cell tracking system. And if you want
Oh,
to
I
add new
see.
genetics, you got to go through a lot of
Yeah.
paperwork with the CCC, and it's a big hassle. So some people are kind of locked and loaded with the mothers they have, and they're looking for ways to fight off these pathogens with native peptides from the cannabis plant, not with some foreign molecule that's going to end up in a vape pen.
Yeah.
And so we're really excited about that side of the market because we like the idea of enzymatic pesticides because enzymes degrade.
Yeah, exactly.
And you can destroy them and you can get rid of them. So you can use them in a really ephemeral nature, and they're not going to end up like myclobutanol concentrating into a
Mhm.
vape cartridge. And we'd rather do this with peptides that we know are from the cannabis plant because the consumers are already consuming those. We
Exactly.
don't
Yeah.
want to grab something foreign that might trigger an allergic reaction. We want to grab something that has been time tested with human biology for thousands of years. And if it's come out of cannabis, it's probably been through that ringer before.
Yeah, that's super exciting. Using these technologies to think about different types of pest management, and something that came into my mind then too is, we've been talking about pathogens, but what about um, genes related in some form or another to beneficial relationships that the plant would have with
Yes.
microorganisms?
So the thing that kind of shocked us that came out of the paper was if you look at the RNA expression in the plant, the most heavily expressed genes in the genome are these chitinases. However, and they're also the most differentially expressed genes, meaning that the different RNA, the RNA expression is quite divergent between tissues,
Hmm.
but it's completely absent in the roots.
Hmm.
And I think the same was similar with the TLP. So why is it not expressing pathogen response to the roots? Well, we think that's due to a lot of the work people have just demonstrated before us, but these mycorrhizal interactions you might have in the root systems, if you
Right.
want to foster mycorrhizal interactions, you don't start off that handshake with a toxic chitinase,
Right. Yeah. Come on
right?
in and let me kill you while you're at it.
Yeah, so that's kind of fascinating to us. The roots are not making this stuff, and that's probably to have some type of commensal relationship with the fungi that are in the soil. Um, the other thing that was kind of interesting along those lines is that deletion that we see so commonly in the type 1 plants, which is a cannabichromene deletion. It's quite present in a lot of the hemp lines. And many of the hemp growers they're working in breeding programs to cross and capture that cannabichromene deletion into their hemp line so they can we think that's what's responsible for the THC levels that you see in type 4 plants. You'll see some type 4 plants out there that are like 100 to 1,
Hmm.
and they don't have THC or CBD synthase. So the most likely place that residual THC is coming from is probably the cannabichromene cluster. And so people are looking to just try and get rid of that,
Yeah.
particularly if it's already known to exist in the population and we can just do a couple crosses and get there. But in the process of doing that, we have to pay close attention to those other 4 genes that are on that deletion that are involved in pathogen response, because that may alter. You may breed yourself a really compliant hemp line that is loaded with botrytis and powdery mildew. And that's a little bit more frightening to us, actually, because the
Yeah.
USDA has nothing in the regs for microbial detection.
No, no.
You can have it loaded with aspergillus, and as long as it's below 0.3, it's cool. And that's a bit of a bummer for those who are looking at CBD flower for like a smoking
Yeah.
program. I mean, there's a lot of success in people swapping out CBD for tobacco, or swapping tobacco out for CBD. I think Switzerland's been doing a lot of that work, and
Yeah.
that's a much safer alternative, as Donald Tashkin's shown. But I don't know that I can say that if it's going to be hemp that can
Hmm.
be loaded with pathogens. So we need to do more work there. That's still just a theory that
Hmm.
these genes are missing when you select for cannabichromene deletions. Um, there's a lot more work that needs to go into proving that those are in fact more pathogen susceptible. We haven't done that work, which
Hmm.
just kind of pointed out the fact that it's a common deletion. People are probably going to breed for it, and it's going to come with these genes getting knocked out. But one of those 4 genes is a gene that's involved in viral response.
Mm.
And viruses, we're now starting to be able to detect those with the RNA sequencing data that's been
Yeah.
coming out of a lot of the laboratories. And the discovery phase of cannabis viruses is really just beginning. There's only a few that have been really quantitatively nailed, but I think we're going to see 10 more in the next 2 years, and that's going to change how we manage those as well.
Yeah, I feel like I keep seeing like every year since I don't know 1 or 2 years ago, I'm starting to see or people are messaging me saying, "Have you heard of this virus that's been identified?" And sometimes these viruses have very cryptic names because they don't have a name. It's
Yeah,
I can't remember the name of
one
one I
of them is
came
actually
across.
called cryptic cannabis virus, one
Yeah,
and
yeah,
two.
yeah. It is.
Yeah,
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 exactly. Yeah,
those are in NCBI, and I can't. I don't know. We made assays for those. We've never found them in cannabis, and
yeah.
it's probably just it's very rare. The challenge with the viruses is that they're usually RNA based. Sometimes
Right.
they're vibroids and they don't even have a protein shell in them, so they're very susceptible to breakdown, which means it's unlikely they're going to make it to your laboratory intact.
Yeah,
Um,
yeah,
across state lines, and very few people want to hang out for that experiment. That if they think they have a virus, they torch it. They don't.
yeah.
And let's put it over here so you can play with it. It's a bit to ask, saying, "Hey, can you save that one for me and ship it across state lines?" What usually means is you've got to teach people how to do RNA extractions in the field so that you can get something that's converted into
Hmm.
DNA or is on ice and can survive the shipment to a laboratory. And we're just not there yet as a community. We're still getting some of those tools out to the masses so that people can do on-site RNA preps. But the ones that are tightly linked to a close university can probably get those studies done, and we're starting to see that. I think Dark Art Nursery did some work looking up That was a viroid. That was a hops latent
Hmm.
viroid. But there was also some excellent work out of Israel looking at a lettuce sclerosis virus. Excuse me. That was they actually demonstrated white fly transmission in that paper.
Mm.
So the pest management is actually playing a role because some of these
Yeah.
pests are actually the vectors for the disease. But we've built quantitative PCR assays for all three of those for cannabis cryptic virus, hops latent viroid, or hops latent viroid, and
Hmm.
then lettuce sclerosis virus. Um, and we're just in the early stages of working with people to get them to test these out. So if you know people out there that are interested in quantitative PCR tests to monitor that, we have the tools. We just don't have the grows or the
Hmm.
gardens or the places to deploy them. We need to partner with people to see exactly how to put those things to use.
Yeah, I think it was the hops latent viroid that someone asked me about very recently, and I hadn't heard about that in the context of cannabis at all until they mentioned it. One thing that I was wondering is there any way to test a plant sort of generically to understand if it um, is likely to have a virus, regardless of identifying what that virus is? Because I know a lot of cultivators battle this. Sometimes viruses appear as like nutrient deficiencies that have very similar
Yes.
symptoms. And I
Yeah.
have cultivators that ask me a lot, how do I quickly know whether this is a nutrient problem or a viral problem?
So this is actually an industry-wide problem that's not unique to cannabis.
Hmm.
Viruses they don't have necessarily conserved sequences that we can rely on to universally capture them and sequence them. So
Yeah.
if you look at fungi and bacteria, there's tricks that you can play.
Yeah,
They
exactly.
all have to have ribosomal DNA operons, right? So you can design primers that will amplify all yeast and mold. You can design primers, or I shouldn't say all, most, because
Hmm.
there is even some divergence amongst primer sites you might pick to try and amplify all of these things. But likewise, with bacteria, you usually go after the 16S region, and that a single set of primers can probably get you everything on the bacterial side of things. We don't have the analogous primer sequences you might target for viruses that
Hmm.
would be conserved across all of them. They're having this problem with coronaviruses right now, that they have to engineer new
Yeah.
assays for these viruses so they can spread them around the world. And the virus mutates a little bit, so they have to
Hmm.
have multiple different primer sites that target the coronavirus. So what people do in the case where they can't use something as fast as qPCR is they resort to RNA sequencing, which just captures all of the RNA that's present, sequences that. Sometimes they'll try and deplete the really common ribosomal sequences that are in the RNA, so they get more efficient sequencing. But it's another expensive step. And then they blast that all through DNA sequencer, and then filter that information for sequences that look like they're from a virus. So there's several viral databases out there that you can leverage that will scan your RNA sequencing data for virus content. But that's expensive. That's
Hmm.
you're
Yeah.
talking about hundreds to thousands of dollars to do studies like that. So it's not something that you just do on a whim. Um, and so you find yourself in is the scenario of doing lots of tests for individual viruses of suspect. Now, I've not seen tobacco mosaic virus ever show up
Yeah.
genetically. I've seen people say they have it due to some pictures, but
Right.
I've not seen it actually show up in a test that proves it was that. It's probably something very related, but not exactly tobacco mosaic virus. It's probably a hops mosaic virus or something.
Yeah.
There's a few of those that have been listed. So it does make for a bit more cumbersome of a test. How they handle
Hmm.
this in human clinical studies is they tend to have multiplex tests that look for a panel of rhinoviruses. So
Hmm.
they maybe they first scan 20 or 30 of them with qPCR, and then depending on what hits they get, they might do
Narrow it down.
narrow it down to some other ones. So that's at least what they're doing with the coronavirus. They have a test that looks at a handful of common cold viruses, rhinoviruses, and noroc type viruses, and then if they see it's negative for all of those, they graduate it to maybe a SARS or a coronavirus test. So we'll
Yeah.
probably see something similar like that in cannabis, as there'll be a two-step screen.
Hmm.
And if you really need to know because it's plaguing your entire grow, you might graduate to doing DNA sequencing and RNA sequencing to thoroughly figure out if this is something novel that's not never been seen before, and you want to crush it. That's usually what you do.
Yeah. Yeah, fascinating. I know that's probably yeah, I know growers want that easy sort of detection fix, and I'm sure it's disappointing to hear that it's just not there. But I know many growers that have tried to battle what what they thought were nutrient deficiencies for far too long, trying to battle what actually ended up being a virus, and just wasted a lot of time and everything. And my response is usually like, if you're really worried about it, just can you just torch that plant and move on, rather than wasting so much energy and time. And some growers are now getting to that point because their fields are so big now, they're not worried about saving a single plant or a handful of
Right,
plants. So
right,
they're able
right.
to just cull the ones that seem to be causing problems and move on, which would be more typical in bigger forms of agriculture. Um, changing gears quite a bit. What are your thoughts on how genomics research is going to influence cannabis taxonomy in the future? Because that's a really muddy
Yeah.
area in general. Cannabis taxonomy is one of these things that nobody, I mean, even botanists and um, chemists and geneticists, it's hard to get on the same page on how to classify plants or even how to define what a species is. But where do you see
Yes.
that moving towards in the future?
So we're in this weird time frame where the entire phylogenetic tree of life is getting redefined.
Yeah, yeah.
And so I'm kind of waiting for that dust to settle before I weigh in because it's,
Yeah.
I mean, they're starting to do it genetically, which is if you're 95% identical to this other microbe, you're the same, genre or the same species. They're trying to make um, metrics like that to begin calling these, and it is needed because we have a lot of redundant nomenclature, even in the microbial side of things. If you try and get Aspergillus species out of ATCC or other cell banks,
Mhm.
you'll find that a lot of them haven't been sequenced before, and then you apply primers to them from the literature
Yep.
that are supposed to hit them, and they don't, and then you sequence its genome and realize it's not what they told you it
Yep.
was.
Yep.
So a lot of this stuff has all been done with eyeballs, people looking at the morphology and looking at these things on plates, and of course, organisms can express really bizarre phenotypes. I mean,
Mhm.
the classic case is like a caterpillar and a butterfly, right?
Mhm.
Same genome, complete. You wouldn't, if you looked at those things, you wouldn't call them the same
Right.
species. If you look at Aspergillus species, they can be just as genetically diverse, yet they look identical.
Mhm.
So the look of the species is no longer trustworthy for classifying them. Now, speciation in general usually involves whether they can continue to cross.
Mhm.
Right? We have cannabis plants that all cross with one another, so
Yeah.
they're arguably the same species. Now, if you want to subdivide them into subspecies indica, sativa, and all that, that's great. But we probably need some paleogenomics to really nail that. If that's in fact the story that the indicas came out of one geographic region and at
Mhm.
one point in time, and the sativas were more equatorial, then maybe that will bear itself out. But right now, they've been so mixed in inappropriately named during that mixing process that the sativa indica thing doesn't match up to any of the genetics right
Yeah.
now. So I usually don't get involved in a lot of debates on whether we're going to call these things chemovars, cultivars, or strains. I tend to mock them actually
Mhm.
because I don't find it a very productive conversation when the whole tree is getting rewritten. We can argue over
Yeah.
the taxonomy stuff later. I tend to just read the DNA because that's the answer you're really looking for. What you're looking for in this categorization is how do these things differ biochemically.
Mhm.
And so chemovar is a great name, but it doesn't right now. The chemovars we're measuring aren't really measuring the flavonoids, so
Mhm.
I
Yeah.
don't view that as a legitimate way to necessarily draw the taxonomy. Um, but ultimately, it's the heritable units that govern the taxonomy. That's taxonomy is DNA, and anyone who tries to tell you otherwise is going to be proven horribly wrong in time.
Yeah. No, I think that's really important to point out. I remember when in some of my early graduate work studying mycology, how frustrating this issue was of learning the taxonomy of different fungi, and then tomorrow it changes because, just like you said, there'd be some genetic work that's done and they realize, oh, these things that look identical even under microscopy and everything, they seem the same, the spores look the same, have same ornamentation, but they're different genetically, and now we've got to call them something else. So students, everything you learned, wipe your brains. Like,
Yeah, yeah,
now
yeah, yeah. Rename.
now we're changing changing the whole system. And even botany too. I was a botanist with the BLM for a while, Bureau of Land Management,
Oh,
scouting
excellent.
native plants around Oregon and everything. And I realized it had been a while since I've gone back and looked at the scientific names of a lot of the plants that I worked with. And so just a few months ago, I went and looked and saw that about half of them that I was studying that had already gone through like genera changes and that sort of things have already gone through changes again and been
Yeah.
reclassified and split and everything. And so it's just a headache of trying to keep up with all this stuff. And
there,
it's
there's an, to give you a sense of the wave of headache that's coming even further. The last study I heard, I think, was in the UK. They're going to sequence 1 million species. All right.
yeah.
So once that happens, everything that you've learned, you got to rewrite. So,
Yeah, yeah,
and it's, I mean, we see the Aspergillus species are getting renamed all the time.
yeah.
Braziliensis got carved out of Niger. So I have no doubt it's going to happen in this field. But I guess I've been advising people in cannabis, just stay tuned and be patient, because this is the wrong time, I think, to fight over those terms,
Hmm.
because we're just in the earliest stages of sequencing everything. And once we have a map of like 100,000 different cannabis
Hmm.
genomes, I think we'll have a picture. And we probably aren't going to try to force that picture into sativa,
Yeah,
indica antiquity. We're probably going to say, okay, the autoflowers are over here, and the
right.
genotypes that have these types of clusters are over here, and it'll be named in a coordinate system that's probably more related to large segmental duplications or deletions that are related to its diaspora around the planet.
Yeah, I agree. And I think the main thing, any listeners that are banging their heads against the wall about how to talk about cannabis varieties, the main thing is to think about like, what are you trying to communicate in the first place, and what do you care about? And that can sort of drive your vocabulary a little bit. If you're looking at cannabis varieties and you're trying to understand chemical differences, then talking about chemotypes and stuff could be valuable, as long as you understand that that's a tool for us to communicate things, not necessarily some hard coded taxonomical
Religion,
structure. Yeah,
yes,
exactly.
yeah, and I
Yeah.
think that's important in science in general, because it's very rare that science drives to a consensus. We want it to,
Yes. Yeah.
but
Yep.
I mean, I think they changed the definition of the kilogram last year, right?
They did.
Something
Yeah, they
is,
did.
yeah,
Yep.
so you're like, oh shit, that can change.
Right. Yeah.
So I thought that was like tied to some astronomical unit of the wavelength of light or something. I didn't
Yep.
realize
Yep.
it was ever going to change, but they do. And consensus, hey, there was a consensus against marijuana for 80 years. It was pretty successful.
Right. Yeah.
And clearly that's in the latest stages of changing today. So um, yeah, I think you're right. It's about communication, and sometimes the communication that's most popular is probably technically wrong. But if your goal is to
Yeah.
communicate, think the most amount of people that will understand what you're saying. Sometimes you just use the language that conveys that
Yeah.
most simply. I think that's probably the case of what's happening with strain. No one likes that name because it's a
Hmm.
microbial term. But if
Yeah.
you
Exactly.
know if you do a Google Trends on strain versus cultivar or chemovar, there's
Right.
no one talking about cultivar or chemovar. Everyone's continuing to talk about strain. So if you want to effectively communicate to large audiences, sometimes you have to use those terms. The reason people put things in Latin is because it's a dead language and the language doesn't change.
Yeah.
And so we don't yet have chemovar or cultivar necessarily in Latin. I think cultigen might be technically a combination of an acronym of Latin
Hmm.
terms, but it's I think it's all about making sure we have communication and not necessarily hard fixed religion. Because if there's one thing we're going to learn in the next few years, is that whatever we're saying in this podcast today will probably be partially mocked as being wrong
Yeah.
in a few years.
Yeah, I mean, there's two things that come to mind there. I mean, one is I've always been a fan of Richard Feynman and his work, particularly like as a science educator.
Yeah, yeah,
Um,
I love that guy.
yeah, he did a lot of great work for science education in general, much less the physics side of stuff he worked on. But something he talked about in one of his late books called "The Meaning of It All." And he talks about the problem with science education in our culture is that most lay people expect that what science does is it produces facts, and it produces, and those
Yeah.
facts, everyone
Yeah.
can form a consensus around them because they're facts. They are known, they're hard, objective truths. But the reality for people that are, actively doing research and, diving into all this stuff is you realize there aren't a lot of hard facts, if any, and consensus is harder than you would expect, depending on what you're trying to get consensus over. And what Richard Feynman said is that ultimately what science does is it gets at answers to questions. It may not necessarily provide you the hard answer, but it gravitates towards the answer, and it's always moving towards that answer, even if it doesn't quite get to that
Get
very
there. Yeah.
specific hard truth. But when you talk to people, like for instance, on a political stage, and you're talking about, let's say, climate change, people
Oh
want to
yeah.
know people want to know the facts. They don't want to know that you've got some general idea of where the science is moving towards, and they don't want to hear about probabilities and confidence intervals. They just want to know
Yeah.
what's the state of things and what do we need to do. And
Yeah.
so when
Yeah.
a politician is willing to just assert solid, bold, flat claims, people resonate with that because it's a lot easier to digest than the actual
Yeah.
muddiness of science.
Yeah. No, it's very true. It's very true. It gets weaponized oftentimes
It
into.
does.
I mean, my view on this is they only separated the church and the state so that they could replace the church with people in white robes versus black robes.
Yeah.
And now the scientists are propped up as some kind of priest that can read the code of life,
Yeah.
and everything that comes out of their mouth is ironclad truth, and we're going to drive politics off of that. And that's nothing to be further from the truth. Is what
Yeah.
really is happening in science is we're generating questions, not answers, most of the time.
Yeah, yeah, exactly. No, that's a great point. It is generally about trying to figure out what are the better questions to ask, and then that drives what research you're going to do and how you're going to approach a question. And um, Isaac Asimov, another writer, science educator, even though he wrote a lot of fiction, he had a paper that he wrote in response to someone criticizing a statement he made about, you know, basically kind of like what you said just earlier, that, in 10 years, everything we're saying now, a large part of it is going to be, mocked at and thought of as wrong. He made a statement like that, and someone said, well, if that's the case, then, does that mean that essentially a lot of things are wasted and that anything could be overturned at any time? And he was like, well, no, no, no. Like, what that means is just that our understanding gets sharper over time and it feeds off of what came before. It's not that what's going to come after is going to be totally, you know, irrelevant and in total opposition to the science that's happening now. It's just going to keep pressing forward. And I think the example he used was like the spherical nature of the earth, and how, eventually we learned that it's not a perfect sphere, and that it's, sort of like a lemon in a way. It has, this sort of oblong nature to it, and it even kind of like wiggles and moves. Um and before that it was assumed that, the earth was
You mean it's
flat.
not
And
flat?
you're right. Yeah. All these things. And so just pointing that, these aren't total overhauls of what came before, but they're refinements. And, it's like getting the clarity tuned up even more as things go on. I think that's important for people to understand, that, what we're saying is not that all of this is just getting thrown out, and that, a lot of scientists are essentially wasting their time, but that, we're all standing on the shoulders of everyone that's come before, and people
Yeah.
10 years from now are just going to have a very different set of eyes than we have now.
Yeah. I mean, that's to me, that's probably the most compelling argument I've heard in this climate debate is that um, the number of ideas that are human generated tend to be cumulative. Like
Mm,
there's idea bridging, and so the fact that we had twice the amount of population back when Einstein was around, or
mhm.
Edison, or Pasteur, right? Pick, if you did population control because you were concerned that we weren't going to be smart enough to figure out how to use things more resourcefully, you would have picked off, Einstein, or
Right.
maybe Pasteur, and then where would the world be if we only had electricity and not the germ theory of disease? Right. The fact
Yeah.
that we had the population we had back then we are so much more richer and more resourceful today than we would have been if we actually choked the population back then. So
Yeah.
um, I think it's in a really an important. It goes back to the concept of humans not necessarily being a cancer cell on the planet. We're
Mm.
actually part of this thing. We have to learn as much as we can about it so that we don't unwisely use the resources that we have. But ingenuity is very important, and I think the evidence is pretty clear. If you look at people's standard of living and if you look at people's income, they tend to become more environmental as that goes up over time.
Yeah.
A lot of the environmental damage is done due to poverty, and more
Yeah.
people are moving out of poverty than ever before. And so we actually have a really bright future that you won't hear about in the political sphere
Yeah.
because that's about selling fear
Yeah.
that we're all going to die tomorrow from coronavirus and tsunami or something. But I think I'm very optimistic about the fact that the population is going to be fine. That I think science is going to be it's exponentially building upon itself,
Yeah.
and we often underestimate that fact. I mean, just now we're in the first generation of people who have access to all information knowable
Yeah.
on their phone,
Yeah.
right? Like school is now irrelevant. No one wants to say that, but it
Right.
is, right? If you like to read and you're curious, you don't need school. You need your phone.
Yep.
And no parent wants to say that to their kid, but we didn't have that when we went to school. We had to go to
Yep.
centralized campuses where all the information was literally stacked on paper into
Yeah.
libraries so that in microfiche so we could comb through this stuff and be near the people who are smart and who already read it. But now we can be near those people on conversations like this,
Yeah. Exactly.
and we have all the information. That's going to radically change idea generation,
Yeah.
and it's going to radically change um, people's access to capital and the ability for people to grow and to invent and to
Hmm.
become more resourceful with the limited amount of resources we have. And I think it's really exciting that cannabis is like emerging from prohibition during all of this because
Yeah. Yeah.
we now have probably one of the most resourceful plants on the planet where people have been restricted from being able to really adequately study it. And now the gates are open and we can study one of the best photosynthetic organisms we have that can literally feed the world with this thing.
Yeah.
So there's a tremendous amount of potential, I think, for solving a lot of these climate concerns people have. I think just by doing what we do so well with hemp,
Yeah.
learn this thing, figure out its code, figure out how to tweak it, figure out how it interacts best with the environment, and that's going to be a combination of not its genome but also the microbes that are
Hmm.
in the soil.
Yep.
And the more we can learn about that, we're probably not going to come to a consensus about what it all means. But as Feynman's saying, we're going to be pushing ourselves in the arc of being more and more efficient with what limited stuff we have here.
Yeah. Yeah. Man. Yeah. That's such a great summary to bring this whole conversation back around. Um, I really appreciate how much time you've been willing to spend with me. We've been going for almost an hour and 45 minutes. I have a couple
Wow.
a couple of closing questions for you to cap us off. One is, um, what other research interests do you have beyond cannabis? I usually like to ask every scientist I interview that question.
So I'm really interested in other pathways that are in its neighboring plants lately. So I've
Hmm.
been looking a lot at the and also this concept of xenohormesis that your previous guest Kevin Spellman will probably have a lot to say about it. But um, so hops has a similar pathway that makes terpenes, and
Hmm.
it makes a different set of compounds that I would probably call the analogous medicinal compounds in hops or the xanthohumol, the
Okay. Yeah. Yeah.
humols.
Yeah.
Right? And those compounds are starting to show anti-cancer properties, much like the cannabinoids. We don't know really what receptors they hit and why,
Hmm.
but everything that's needed to make those compounds uh, has some homology to the cannabis genome. So it's
Yeah.
possible the cannabis genome could be making some of those compounds as well. Um, I don't know if it's going to need to be crossed into it or
Right.
how we genetically engineer it, but it's something that it makes you turn back and look at all these other plants that we've taken for granted and start to ask, well, what are the secondary metabolites in those plants? Because
Yes.
the only reason we've paid so much attention to cannabis is that one of them has a really easy bioassay to tell you when you're getting something. It's
Right.
THC.
Yep.
We don't have that in hops. We might have beta caryophyllene, which is a CB2 agonist, but you probably can't really feel that unless you're in pain and you take a lot of it. So all these other compounds that we can't really perceptually feel,
Yep.
we need to go back and look at because they've been the FDA system is selecting against these things.
Yeah.
And in many ways, the regulatory environment we have here in the United States favors largely capitalized companies that can then pay politicians to limit the regulatory space of the operators. And so the genetics tend to go through bottlenecks in those environments. And we end up having massive genetic shifts because of regulations.
Yeah.
And so I'm somewhat fascinated about the impact of regulation on genetic drift, because I think that
Hmm.
there's genocides going on all over the place as a result of this. This 0.3% thing is going to create a massive, just like prohibition,
Yeah.
create an
Yeah.
enrichment for type 1 genetics and probably a depletion of a lot of type 3s. This 0.3 thing is going to have the opposite effect. It's going to end up meaning we're going to have to select a very narrow set of cultivars to move forward that are compliant with the USDA, and it's going to wipe out all types of diversity we don't know yet, because those selections arguably are going to wipe out these other secondary metabolites no one's measuring.
Right.
No one's going to be measuring the canflavin pathways in the
Yeah.
process of doing these selections. I mean, we have sequencing that looks at that pathway, but very few people are necessarily going to jump on that train right away. So the process of even putting in microbial regulations in place, this means plants that like and enjoy a particular microbial background are going to get eliminated from the population.
Yeah.
Right? Let's say you have a plant that needs trichoderma. Trichoderma is not harmful, yet some of the regulations in some states don't have CFU language, right? Colony
Hmm.
forming units are
Right.
they're in
Yep.
they don't know what the hell they're counting. They're just saying,
Exactly.
bugs, bad, right? It's really really ignorant for to think of microbiomes that way. So when you put that in place, all of the plants that can get through regulation then go through a genetic shift, right?
Yeah.
You get this selection for plants that tolerate low microbe environments. Well, those may not necessarily be the most productive plants from a secondary metabolite standpoint. No one's looking at that. They're just looking at maybe THC and CBD, but all these other flavonoids might be abolished in the practice
Yeah.
of doing that. So I'm kind of curious at seeing where that's going. But the xanthohumol thing and the xenohermesis is just this concept that these plants are making these secondary metabolites to communicate to the things that propagate their genetics, right? So
Mhm.
if you were to actually look at the number of cannabis cells and the number of genomes that are dividing throughout space and time, there's probably a good argument that it's winning that race compared to humans. That we are
Mhm.
propagating more cannabis DNA right now than human DNA,
Yeah.
because of the nature of how big some of these plants can get, how big their genome is, the fact that they replicate every 12 weeks, and humans
Right.
don't replicate that quickly, means that some numbers I've drawn out suggest that the cannabis genome is actually done a remarkable job finding an ideal pollinator that
Hmm.
likes to propagate it all over the world. And how did it do that? It did that by being able to make secondary
Hmm.
metabolites that tap into our biochemistry to tell us that changes in the environment are coming, or what have you. So this work is all done by David Sinclair. He's the guy who does a lot of work on the fountain of youth and resveratrol and
Hmm.
many of these compounds that are involved in that pathway. But he's got a great paper out there called Xenohermesis, and it talks about how these plants, when the environment changes, they shift the secondary metabolites they make so that the things that
Right.
eat them, their mTOR pathway gets triggered, so that they start going into caloric restriction because the plant's biochemistry has changed to know that the environment is going to change and it's going to have an impact on the amount of material that it can feed the thing that's supposed to propagate genetics.
Wow.
So that whole interaction space, I think, is the key to
Fascinating.
cancer. And if we can understand how those plants are signaling the metabolism of the things that propagate their seeds, we will understand cancer in a much better way, and a much more thorough understanding of cancer. Because usually cancer is a scenario of your own genome having a metabolic defect going on in the subpopulation of the cells, and that
Right.
that metabolism is getting triggered, and it's usually because of an acidic or a different environment, a mutation that's happened. And the things that seem to be having the most potent effect on our cellular metabolism are the vegetables and the plants and the foods that we eat.
Right.
Um, and that's what we need to better understand to really understand cancer. So I kind of like that intersection of plant metabolites
Yep.
and cancer. It kind of touches home for us, and it's a field that I think our myopic focus at the FDA is constantly deteriorating, because they have this um, reductionist approach of looking at single compounds as opposed to symphonies of them.
Hmm.
And we're at a stage now where we have the tools to look at symphonies. They just don't fit into their trials very well,
Exactly.
and
Yeah.
we need to rethink whether their trials are the right way to do medicine. I happen to think they're not.
Yeah.
that you can get a hell of a lot more power out of a study with 100,000 to 1 million people connected via observational trials on digital equipment, like we do with peer-to-peer
Yeah.
networks, and 5,000 or 10,000 people in a trial that is a subsampling of statistics to the point where you end up with Vioxx-like compounds that escape that process.
Yeah, I mean, and not to mention the way that the clinical trial system is set up right now, it creates such a high barrier to entry to even try
It kills people.
to get through that
Yeah,
process, you know?
yeah, it literally kills people, and there's no one willing to look at the seen and the unseen here. It's like, okay, the fact that a drug takes $500 million and 10 years to get through. Has anyone done a survey on the number of people that died waiting for that drug,
Right.
knowing that it's available in Europe and it's available in Australia? Other countries have deemed it safe, but not here. And those people don't have the money to travel there to do it. So they sit here, they wait, and they die.
Yeah.
And there's people who study this, and it's a large number of people every year. The Independent Institute does a lot of work on this called, and they're FDAreview.org. It's worth reading because they go through all of the scenarios where drugs that were approved in other countries, so they pass,
Yeah.
other countries' criteria, and eventually passed here, but 3 or 4 years later, and they count the number of deaths that occur because of this. And no one's responsible for this.
No.
And you can't vote them in or out. And I think they regulate now like 25 cents of every dollar.
Yeah.
So this is something that is, I'm totally terrified that it's going to come into the cannabis space and somehow
Yeah.
make the same mess of this space as it did the opiates. That being said, there's a lot of good scientists that work there that
Yeah.
have all the right intentions, and they are trying to put genetics into all their programs. They're trying to build blockchains to track all the food. So they've got all the right vision. I just think they don't recognize that it's too big of a problem for one centralized organization to solve. And it's too prone to regulatory capture and corruption that they shouldn't set up that honeypot. They should somehow decentralize it and have competing agencies that can perhaps invent different ways of regulating, because it's certainly an important feature of a marketplace to have regulation, but not necessarily in the way that it's getting done today.
Yeah, and I mean, I also find it kind of funny that, a lot of times drug approvals get so hung up on needing to prove a certain level of safety, sometimes to kind of an absurd extent. I mean, right now we're seeing this with CBD right now that, with
So the liver tox stuff.
yeah, exactly. And, and not acknowledging the nuance around, some of that research and everything. But what's interesting is that the actual efficacy of a lot of pharmaceuticals that actually make it through the process are not that effective. Maybe for 20 or 30% of the target population, they may be effective.
Right. Yeah.
And they have terrible safety profiles. And so it's, I don't know. It's a weird dynamic that the safety profile of a lot of these natural products that people want to research and try to understand how they can be elevated out of the realm of sort of, this alternative medicine sphere and try to understand like what they're actually doing and how to actually, legitimize them as medicines. It's very challenging, even though the safety profiles of a lot of these things are pretty well understood by this point.
Yeah, and I think the other big hangup they've got is there is this gold standard, almost religious consensus, as you were mentioning before, on double-blind placebo-controlled trials being the only path to ensuring safety of a medicine. And I think that's being fundamentally challenged by, if you look at the work on what governs the placebo effect, it starts pointing back to the endocannabinoid system.
Yeah.
And so there's actually papers out there on the endocannabinoid system being involved in the placebo effect and analgesia, right? So how do you put cannabinoid through a double-blind placebo-controlled trial?
Exactly, yeah.
If the placebo effect we think is people's reactions, their ECS, their reduction in anxiety is caused probably by anandamide or something, that when they take something, they're taking control of their problem. They're taking action, and that action gives them confidence, and that boosts their anandamide levels maybe.
Right.
And so the very pathway that you're trying to interact with is what's involved in the placebo process. So I think that's just all going to create noise, and we should be looking at very large observational studies that we can enable now with these digital tools that we have. And that's probably going to give us a far faster and more, back up and just look at the vape crisis, right?
Right. Yeah.
So this is a COX-2 inhibitor, CBD. A lot of people are vaping CBD, sometimes THC, and they both have analgesic effects. And so I throw them into that category of Advil and ibuprofen replacements, right? Maybe even Tylenol. Tylenol is, in fact, an endocannabinoid mimetic. It
Hmm.
makes AM404 with a shitty liver metabolite. But so people are vaping these things probably because they like getting a little relief from some pain and some fun. Um, that took the field probably 10 months to resolve, right? Because everyone was sharing information. Rumor came out of this company's making crap. This one's got bad vitamin acetate. Ideas are flying. And with all of the networking we have, there's a crisis that was affecting maybe 1,000 to 2,000 people.
Right.
There was 9 million vape pens sold a month,
Right.
90 million, maybe 100 million over the course of the year. And we only had an incident rate of 1 in 45,000,
Right.
effectively. Now, that's one piece of arithmetic that's important for how frequently we sample for these contaminants in cannabis. We're not sampling nearly deep enough to find
Right.
a frequency that low. So even if we were testing for vitamin E acetate, I'd argue we still would have missed it because the frequency of this creating a problem is so low that you need to up the
Hmm.
sampling rate you do in the industry to find it. But that being said, can roll it back to Vioxx, right? Vioxx went through, probably killed 50,000 people, and it took the FDA 5 years to finally admit they were wrong, right?
Yeah.
And why is that? Well, because admitting they're wrong after they were
Hmm.
paid a lot of money by the pharmaceutical industry to put that drug through is harder to reverse, right?
Yeah.
It's more crow to eat than you want. And many people don't realize the FDA right now is governed by the PDUFA Act of 1992, which means about half to 2/3 of the revenue going into the FDA comes directly from the companies
Hmm.
that they regulate. Right? This is a
It's
way
such
of
a funny
defraying.
system.
Yes, it's a euphemism for bribery,
Yeah.
but
Right. Yeah.
it's effectively how can that agency, with a straight face, claim that they are out for the consumer when they're on the payroll of the
Hmm.
people they're regulating? This is going to lead to really bad decision making, even if it has the right intentions. The incentives aren't there. When they make a mistake, they're going to get a bigger budget.
Yeah.
So this to me is like we don't need this near cannabis. The cannabis hasn't killed anybody. So what is the FDA? What they've already done to cannabis, if you just look at Epidiolex, has made it 30 times more expensive,
Yeah.
made it an isolate, put in a strawberry flavoring
Yeah.
that is registered as a pesticide by the EPA, and then added in like sucralose, which like gives
Yeah.
people sneezers. I
Yeah.
mean,
Right.
they made it into a worse drug and GW has to kind of unfortunately put up with this.
Right.
And GW is filled with people who know
Yeah. Yeah. Yeah.
that know the extracts are more effective, but they can't get something that complicated through. I mean, they got Sativex through, but they haven't gotten it through
Not in the
our
US.
system.
Yeah. Yeah.
Yeah, so
And
they
if
had
it
to go
does,
for isolate CBD.
right. Yeah. I mean, Ethan Rousseau and I talked about this in our interview and a little bit off interview too. But, the fact that they're struggling so much with Sativex, and that if Sativex does make it through the U.S. system, it might end up being a very different pharmaceutical altogether than what you would find in the UK or in Spain or things in Canada too. Um, and it is really interesting, and I agree. I mean, I've been following GW for pretty much since I knew about them in, I don't know, long time ago in the early 2000s or something. And I've followed the work of several of the different scientists that have come and gone that have worked with GW, and I agree. I mean, it seems like, as far as I can tell, they understand the best way to actually apply these medicines. Some of the research they've done has actually highlighted, that
Oh
these
yeah, they've
that
got
these
some of the best
extracts,
data out there.
yeah,
Yeah, yeah.
but they're having to play the same regulatory games as anyone else, and um, you know, it's so common, especially in the cannabis industry, for people to bash anybody that is perceived as a dastardly pharmaceutical company. And GW,
Hey, they write the rules. They got to play by them. So I
right?
don't blame. Sometimes they write the rules when they're really big. I don't think GW's at that scale, but
No, they're not, and that's something I try to remind people of. Is one, they were doing this work in other countries initially, where they were able to do things that they can't really do here, and some of the products. I mean, Sativex in particularly is actually a very impressive product when you think about it. It's not. I think there's a misconception that it's isolated THC and CBD, but it's not.
well, it's a
It's
botanical
a
drug substance. Yeah,
standardized. Yeah, it's a
yeah, it's a
standardized
VDX,
extract. Yeah, and yeah, Ethan and I were
and as
talking
an
about
extract, they do actually a really good job measuring. They're measuring things that the labs are measuring. Like
right.
I think they're measuring sterols, a host of these other compounds
Yes.
that we're not seeing in the dispensaries. So they have, with hands down, the best manufacturing process for this that there is. I don't doubt that
Exactly.
one bit.
Yeah.
but they're getting asked to point that amazing scientific engine they have in the wrong direction.
Right.
You know,
They're like, but tell us what's the active?
yes, yeah, it's like they're all active.
Right. Yeah. 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 So it'd be interesting. I mean, I would love to see in the United States some process for getting herbal medicines approved as, legitimate standardized medicines. And, now we have like, um, what are they called? Like medical foods that you can get prescriptions for that often are usually just imported from other countries. But the example I always use is like standardized ginkgo biloba extract that you can get a prescription for, and it has standardized amounts of certain chemical constituents. But it didn't go through it wasn't produced in the U.S. and it it's an interesting process to even get it to be allowed to be used the way it is in the United States. And it's just one of those things that I think a lot of people don't realize, that there are alternative ways of determining what is a medicine and how to give people access. And that a lot of countries do have some pretty decent ways of handling herbal medicines. And this isn't a complicated problem to figure out. It's just a bureaucratic and political problem to figure out in this
It
country.
is, and unfortunately, I think it's that same process of politics and bureaucracy that are forcing these really odd distinctions between medicine and food. I
Yeah,
mean,
yeah,
food is
yeah,
medicine,
yeah.
and
It's all
this
chemistry.
concept of like recreational versus medicinal is to me is like mind boggling that people don't recognize that if you do the recreation, you won't need the medicine.
Right,
They're one and the same. It's just when you get into a chronic state of disequilibrium, you might call it medicine,
right.
but
Yeah,
but that distinction is purely political, and it comes. I don't know where it comes from. It comes from some hatred of people getting high, and
yeah.
yet they happily to have Bud Light commercials all over the Super Bowl. So
Right. Yeah. Yeah. I don't know. It's a strange state of affairs. Interesting time and place in history that we find ourselves. Hopefully, it evolves uh, in a more favorable direction in the future. Um, I don't I Now we've already gone over 2 hours, so I'm beyond ecstatic that you've been willing to talk to me for this long. I want to give you a final platform here. Let people know how to learn more about medicinal genomics. Also, one thing we didn't even talk about, but is Cannapedia, so where people can actually explore some of the genetic data and even some chemotype data that medicinal genomics and other groups have been bringing together. So I'll just kind of give you a platform here to let any of our listeners know anything that we haven't covered and where they can find out more about your work and everything else.
yeah, so medicinal genomics, we make in addition to doing sequencing for folks in the industry, we make a lot of tests to look for different genetics in the plant. So THC and CBD and XY testing, we make assays for testing those markers. We have markers that look for fusarium and powdery mildew, and basically all the microbes that might harm the plant,
Mm-hmm.
and also all the regulated microbes. So we sell a lot of quantitative PCR kits into many of the labs that measure Aspergillus and Salmonella and E. coli and those things. So that you can find in medicinal genomics. The sequencing work, there's a lot of it put public on a site called Cannapedia. A lot of people don't know that we run that site because of the way it looks, but that's a medicinal genomics site for hosting a lot of this sequencing data. It's serving some purposes for research and also for IP. There is a
Mm-hmm.
blockchain component of that will etch the timestamps that your data is generated into a ledger that we can't control. So if we ever become evil on you, you still have your data recorded somewhere else. And that's all managed at the Cannapedia site. And then thirdly, we do run a scientific conference every year called CanMed. So if you're interested in more of these topics, there'll be many people presenting there. Last year we had Raphael Mechoulam, and
Mm-hmm.
he's come most years. I think this year might be a little tight for him because he's getting of the age where it's hard to make that travel from Israel. But other folks of his caliber, we try and bring in and have scientific discussion around crop yield.
Mm-hmm.
How do you breed for better crop yield? How are we going to handle appellations is one thing that's coming up that we think is really important for the cannabis field, so we don't turn into a monocrop in the U.S.
Yeah.
And then we're also hitting analytical chemistry labs and genomics topics. So submit your abstracts, take a look at it. It's usually this year it's going to be in Pasadena, down at the Pasadena Convention Center. I think it's at the end of September.
Nice.
But it's usually around 800 to 1,000 people.
Oh, nice. That's awesome. I'll have to keep my eye on that. See if I can make my way down there. I've never been, but I'm also not. I'm not a huge. I don't know. I tell people I'm kind of a hermit. Doing this podcast and starting to like finally get out in the world has been a big challenge for me. I've got to like get out of my cave more.
Yeah,
um,
yeah.
but trying to make it to more conferences regularly. And CanMed's one that I've had my eye on a lot, just because of the caliber of people that usually show up there, and the sort of higher level of discussions. It's one I've wanted to make it to, so I'll have to try to get down there. Um, well, thank you so much, Kevin, for coming on and chatting about so many topics. I've really enjoyed it. Keep me updated on any big moves that you guys make, or any new publications like this preprint that you shared with
Absolutely.
me. Anything like that, and if anything big comes up and you want to come back on the podcast to talk about anything specific, just let me know. I'll be happy to have you again.
Absolutely, Jason. Thank you for the time, and keep up the great work. I love hearing these podcasts. I was running out of things to listen to. Now yours is on.
Well, I view that as a great compliment. Thank you so much. All
Yeah,
right,
yeah, great.
everybody. Thanks so much for tuning in and listening once again. If you want to learn more about Curious About Cannabis, you can go to cacpodcast.com, or you can find us on social media. We're on Instagram, Facebook, and Twitter. And we also have a YouTube channel now. And when we're able to get videos of interviews, we get those on there. And I've also started doing some like site reviews and book reviews and that sort of thing, or at least editing some of the video that'll come up pretty soon. So check all of that out. And as always, please tune in next time. I really appreciate it. Thanks so much. Stay curious and take it easy. Hey, everybody, this is Jason Wilson with the Curious About Cannabis podcast. Thanks so much for tuning in once again. Today, I'm joined by somebody I've been wanting to talk to for quite a while, Dr. Ethan Rousseau. Thank you, Ethan, for being willing to take the time to sit down with me today.
Thank you.
Yeah, so I almost struggle to figure out how to introduce you, because there's so many different angles I could take. You were involved at one point in getting Sativex going and approved, is that right?
Sure. Yeah. For 11 years, I worked for GW Pharmaceuticals as senior medical advisor and medical monitor, study physician in development of Sativex and Epidiolex.
Yeah. And then you also, on the side, are involved in a lot of different types of broader research and education.
Sure. I've been involved in cannabis and cannabinoid research for 23 years.
Yeah, quite a record. And some of the review papers that you've written have been critical tools in the education work that I do. Like Taming THC is now pretty much a classic. And there was a recent one you did too, the No Strain, No Gain review paper about the complex, what we call the entourage effects. Some people call them matrix effects, whatever.
Right.
But you've, in my opinion, you've done some really good work summarizing relatively complex ideas that are digestible to a broader audience.
Well, I hope so. One of the big issues right now is trying to sort out whether people should have single compounds, like single cannabinoids, versus whole plant extracts. And I'm clearly in the latter camp.
Yeah.
For the synergy, the boosting effect that these provide over and above the kinds of effects that we can get with single compounds.
Yeah, a lot of doctors and nurses that I've spoken to that are doing clinical work with cannabis products in the United States, a lot of them, it's been a pretty consistent message that they've said at this point they tend to only want to work with what they term broader spectrum extracts of cannabis, usually like ethanol sourced, that maintain a wide diversity of phytochemistry. And what they tell me, and what I'm interested to talk to you about, is what research data we have to kind of support it. But they're able to get
better outcomes at lower dosages with fewer side effects?
Sure. Well, we can illustrate this by going back a little bit historically. In 1985, synthetic THC as Marinol was approved by the FDA. The thought among certain people at the time was this would obviate the need for cannabis treatment for medical conditions. Instead, what we got was a drug that is very poorly tolerated and gained no traction in the marketplace. THC by itself is a very disorienting drug. People tend to be dysphoric, unhappy, rather than euphoric. Even people who are accustomed to cannabis find THC in pure form a very difficult drug to tolerate, and very few people remain on it for any length of time. And when people have had the opportunity to compare, almost invariably they prefer herbal forms of cannabis.
Yeah, that's been my experience too. I have a good friend of mine that, funny enough, to treat schizophrenia got approved for Marinol, which is sort of strange in one sense to me. But he reported similarly that he'd had some success with herbal cannabis controlling certain positive symptoms that he was dealing with, and the Marinol just didn't do anything that the herbal cannabis did other than making him feel uncomfortable, like physically unsettled is what he reported. And that's the only person that I personally know that has been on a Marinol prescription.
Sure. Beyond that, we have now a lot of evidence, both laboratory and clinical, that much better results accrue when we're dealing with complex mixtures from cannabis extracts as opposed to pure compounds. A recent illustration was an article from Spain, Blasco Benitez et al., in which they were looking at effects on cancer, both in cell culture and in animals, comparing pure THC, which worked to some extent in treating the cancers. But in comparison to a cannabis extract, there was much, much greater activity with very salient, statistically significant differences.
Right, yeah. I remember there was some paper I read that was looking at, was it TNF alpha levels in mice that was looking at, I don't remember the actual type of botanical extract that they used from cannabis, but they were trying to compare to isolate and look at dose response curves, essentially.
Yeah, another good example there was a paper by Pamplona in 2018 that compared pure CBD, as would be in Epidiolex, to the results that people got with CBD predominant cannabis extracts. What they found was equal efficacy, but with the cannabis extracts, the doses were about, on average, 22% of what was required with pure CBD. So instead of 1,500 milligrams a day, perhaps 300 milligrams would be enough of the corresponding extract. So this can represent a substantial savings in cost. Additionally, with the high doses of pure CBD, there is the possibility of drug-drug interaction, particularly sedation with some of the drugs that are used in epilepsy treatment.
Yeah, and I know there's some concern lately too, and there's controversy over some of it, but about the liver toxicity of high doses of CBD as well.
Well, I think that's a blind alley, to be truthful. Obviously, lower dose would be better, but the liver issues are mainly related to elevations of liver function tests. And this is in generally children who are on polypharmacy with a number of drugs, particularly valproic acid, which is well known for being a bad actor in terms of LFT elevation. So my strong suspicion is that's related to polypharmacy, multiple drugs, rather than a specific effect of cannabidiol.
Yeah, that's an extremely good point to make. Is that the risks, contraindications, and everything are very different circumstance to circumstance, depending on what a patient is taking. And,
Sure.
and I would assume not just the other medicines they're taking, but even possibly their diet, because I know there are different dietary elements that can influence.
Yeah, I would just add the point that I've seen a lot of people conflate, even in presentations at conferences, conflate the side effect profile of pure CBD at high dosage to CBD in general, and that's not a fair comparison. I would say that cannabidiol, despite its many complex mechanisms of action, is an extremely safe drug with very few adverse events.
Yeah, and what I mean, there's a tidal wave of research kind of coming out now that, I mean, especially now that the Farm Bill's passed, there are a lot of universities now ramping up to study CBD and hemp extracts, and in general, with legalization movements happening across the world, there are more researchers feeling more comfortable taking on this work. But in your opinion, where does the research, particularly around CBD and CBD extracts, need to go, I guess, to elucidate some of those nuances that can help particularly physicians understand what they're dealing with, and if they have patients that are coming to them telling them they're using different types of CBD products?
Yeah. Well, first and foremost is an issue of the preparation. Right now, it's sort of the Wild West out there. With online cannabidiol products. I can't vouch for the utility or safety of any preparation without a certificate of analysis. And I want to know exactly what's in it, not just the cannabidiol content, but other cannabinoids, the terpenoid content. I want to see evidence that it's safe, that there's no pesticide residue, microbiological contamination, or heavy metal contamination. And beyond that, I just won't comment. And I would urge consumers to be extremely diligent in trying to acquire that information. My bias is that that information should be available for every product, whether medical or recreational, at point of sale. So that's first and foremost. Beyond that, it really is a matter of what's in it. So we can have cannabidiol, but there's almost nothing that cannabidiol does that will not be enhanced by having at least a tiny amount of THC on board at all. It really synergizes. These normally occur together in the plant. It sounds corny, but that's the way nature intended it. And that's what usually works best medically. The effects of a given preparation are going to depend on those components. And we really can fine-tune things in terms of applicability to produce synergies towards treatment of specific disorders. What I mean is, for example, if we want to treat a mood disorder such as anxiety, we can look for possible synergy between cannabidiol, which has a proven effect at a certain dosage, plus an additional component such as linalool, which is a common component terpenoid that's also in lavender, for example, and is well known traditionally as something that produces a calming anti-anxiety effect. So that's just one illustration. What I'd like to see is that there's a greater appreciation of these nuances of how cannabis can be tuned to the individual need.
Yeah, and something I'm interested in too that segues into another thing I wanted to talk to you about, which is the endocannabinoid system. Is I'm interested to see research that goes into how to characterize the endocannabinoid system tone more? I think that's really fascinating.
Sure. Well, that's a tough one.
Yeah.
First, let's define some terms.
Yeah.
The endocannabinoid system is the master homeostatic regulator of physiology. What does that mean? Well, physiology is how things work in the body. Generally, you need a certain level of activity, neither too high nor too low. And that's precisely what the endocannabinoid system does, is try and keep things in happy balance. So if a system is overactive, it tends to bring it down. If it's underactive, to bring it up. This works through complex mechanisms. We have endogenous chemicals within called endocannabinoids, specifically 2-arachidonoglycerol, 2-AG for short, and anandamide. Anandamide in particular closely resembles THC. Both are partial agonists at the CB1 receptor, the main psychoactive receptor in the brain and elsewhere throughout the body. And what this does at the brain level is actually reduce the release of neurotransmitters. So if we have a glutamate neuron that's stimulatory, the reduction in the release of glutamate will lower that activity. Good example of why that's important is, we'll give two. Glutamate activity tends to be excessive in neuropathic pain, nerve-based pain. So if we have a person with chronic neuropathic pain, we can either raise their anandamide level or supply exogenous THC and bring that glutamate level down. And that's going to help with pain. Similarly, in brain injury, whether it be trauma, stroke, or things of that sort, we get an excessive release of glutamate. It can be so bad, there can be so much glutamate that it produces an excitotoxicity, that the cells actually stimulate themselves to death. We have situations in which people suffer a head injury. They might seem okay briefly and then rapidly deteriorate over 48 hours. And some of that effect is from this excitotoxicity from glutamate. If we're able to supply drugs like cannabidiol or THC early enough, some of that can be prevented and have a much better result clinically. So those would be examples. Going back to endocannabinoid tone. Endocannabinoid tone is an important concept, but we don't have good ways of measuring it.
Right.
The three important aspects are how much of the endocannabinoids are present. Second is, what's the activity of the receptors? If someone is using THC at high doses recreationally, the receptor number will actually go down. It's the brain and body's way of trying to keep things on an even keel. So the receptors become inactive. When they stop, they'll reactivate. So there is the number and status of the receptors. There is the amount of the endocannabinoids. And the third part is, what is the activity of the enzymes that make the endocannabinoids and break them down? So unfortunately, we don't have ways of measuring this directly, except with invasive techniques, such as doing a lumbar puncture or spinal tap to measure anandamide levels. But that's invasive. It's not something you can do on an experimental basis with most ethics committees, for example. Ideally, in the future, we would have scans, non-invasive methods of trying to assess endocannabinoid tone. But that's a dream for the future right now.
Right, yeah. And measuring the endocannabinoids, isn't it a little complicated too, because they're produced on demand? It's not like they're stored in vesicles like a lot of neurotransmitters are.
Yeah, that's exactly right. So these are in a constant state of flux. They're just not there for any period of time. Similarly, they're a very hard thing to study. Anandamide is rapidly broken down in the bloodstream. So when we are looking at serum levels, which may or may not reflect what's going on in the brain, the samples often have to be frozen in liquid nitrogen and then be transported to a specialty lab. This is not something where you can go in your local hospital and get your blood drawn and see what your status is.
Right.
We're talking some very sophisticated science here.
Yeah. And another thing that comes to mind when I think about the complexities of trying to measure the endocannabinoid system tone is that the endocannabinoid system is not just CB1 receptors and CB2 receptors. It's a really complicated system that overlaps with a lot of other physiological systems and other different types of receptors that would normally be associated with other systems in the body.
Right. Well, yeah, no, it's a broadening concept. I asked my friend and colleague Vincenzo Di Marzo whether he now would consider the serotonin 1A receptor part of the endocannabinoid system. And he didn't hesitate. He said yes. How this comes into play is cannabidiol was shown by my team at the University of Montana back in 2005. Cannabidiol is a serotonin 1A agonist. And we're finding that mechanism explains a lot of activity of cannabidiol, as well as its effect on nausea, prevention of brain damage from ammonia levels in hepatic failure, and a lot of other mechanisms. Another example would be cannabidiol is also an agonist, a stimulant on the TRPV1 receptor, the same place where capsaicin works. And again, most experts consider that part of the broad concept of the endocannabinoid system. So what's amazing to me at this point is for a system that's so fundamental to how our bodies work, this is not, in general, taught in medical school.
Yeah.
Somebody can go through medical school and learn nothing about cannabis, or except perhaps its alleged addiction potential. But unfortunately, it's highly likely they won't learn anything about the endocannabinoid system, despite the fact that CB1 is the most abundant G protein coupled receptor in the brain, exceeding the number of all the neurotransmitter receptors put together. So obviously it's really fundamental to how humans work.
Yeah, it's ignoring that system is like having severe tunnel vision when trying to think about physiology, considering how big of a role it is. And when I've talked to other doctors that are not, because I'm originally from Mississippi, so very conservative and much more of a deficit of understanding than you would find out here on the West Coast. And when I've talked to doctors about it, I've tried to use terms that might help them put that into perspective, talk about eicosanoids and G protein coupled receptors without getting into some of the vocabulary that we have, and they can start to wrap their mind around that and around how the body processes fatty acids and how important those are for signaling and everything. But it's true, and I haven't talked to anybody that's close to it recently about whether that's still the case, if medical schools are still ignoring whether intentionally or not, a lot of information about the ECS.
Yeah, I'm afraid that's still the case. There's quite a deficit there that I'd like to see rectified. It is myopic. It's intellectually dishonest, and I wonder how much of it is born from prejudices against cannabis.
Yeah. Yeah, and I know part of it coming from where I come from, there's just so much still taboo around the idea of cannabis, and a lot of feeling that you're putting your professional reputation on the line in order to talk about it in a serious way.
And that's one advantage of age. I no longer care about that.
Yeah. What are so I've got two things that came to mind when we were talking about that. One is I wanted to come back around to the serotonin receptor concept, because it's been shown that some serotonin receptors, and maybe it's the one you mentioned because sometimes I get them confused, but they're often heteromers with CB1 receptors. They often appear side by side at times and influence each other.
Yeah, that has been noted in a number of experiments, and to be honest, I don't always know what the ramifications of that are.
Yeah.
But it's always tough. For me, I'm 67, so my basic science training was like 45 years ago, and things change radically.
Yeah.
So I'm like a lot of people, and when I've got a new journal article I think has pertinence to what I'm investigating, I may need to read it 3 times
Yep.
to try and get it straight. So none of this stuff is simple. There's a problem trying to translate this into lay terms because the foundation is so broad in the endocannabinoid system, and the details can be so extensive. It really defies simple explanations. And to be fair, that makes it very tough for the average physician to be able to grab a hold of this when their schedule has them seeing a patient every 15 minutes. It's just not amenable to providing good education to the patient about anything, let alone something so complex as medical treatment with cannabis or the endocannabinoid system.
Yeah, no, exactly. They have everybody has such little amount of time and mental bandwidth to wrestle with much of anything, that to try to tackle all of this is definitely complicated. And something that I teach about is try to urge people to be cautious when reading research and trying to project the clinical value of that, because so much of cannabinoid research is cell culture models and rodent models and a little bit of human clinical data, and it's all interesting, and there's wisdom to be gleaned from all of these different studies, but you've got to be really careful.
Well, yeah, let's use an example, and I want to go back to the hepatic damage from CBD issue. There was an article that came out recently. It was a rodent study. The doses of cannabidiol that they were using purportedly producing liver damage would be impossible to attain in a human. And somehow this travesty passed peer review and was widely reported by the media. It has no correspondence to reality. I just wish that it had been sent to me, and I would have done my best to ensure that it didn't deserve publication. And this kind of thing, unfortunately, this kind of material is often produced with taxpayer money. And it's more than intellectually dishonest. It's downright dangerous and unethical.
Yeah, and it just perpetuates more misunderstanding among not just a lay audience, but physicians and other healthcare providers that are working with patients that are reporting use and trying to conceptualize what that means.
Well, I know that I got 10 personal queries about it that I had to address and dispel what are just spurious rumors and basically propaganda.
Yeah. What would you say would be some of the more real risks, limitations, contraindications of herbal cannabis use and extracts, depending however you want to go with that you think people should know about?
Well, we've got a public health crisis out there right now with additives in vape pens.
Yeah.
So we've got to mention that. It is important that people, again, apply critical thought to any development like this. That actually has nothing to do with cannabis.
Yeah.
It has everything to do with additives that are toxic that are put in these devices. And so it can happen with nicotine vape pens. It could happen with cannabis vape pens or whatever. But I'll put the finger on the government. You wouldn't have this situation if we had legal commerce federally in cannabis and had standards apply to any of these products because this is 100% preventable. If these things were available and regulated properly, there would be no need for kids to be going to a street corner to purchase black market vape pens with dangerous ingredients. So that's first and foremost. Very briefly, I'm not a proponent of smoking anything, particularly in a medical setting. Vaporization potentially is a safer approach, but when we're talking about medical uses of cannabis, we're almost always talking about chronic conditions. And in that situation, best approaches are for oral agents or tinctures, say in the mouth even, and there are reasons for that. When that type of preparation is used, it's got a much longer half-life. Basically, the need for more frequent dosing is limited. Often people can treat their condition with dosing 2 or 3 times a day. This offers other advantages in that there are fewer peaks and valleys of activity. You avoid what can happen with inhalation, where there's a rapid increase in blood and brain levels that is going to make a person prone to psychoactive side effects, specifically anxiety, paranoia, things of this sort that are much less likely with a slow titration, slow raising of dose with an oral agent. And therefore the risks of intoxication, reinforcement, withdrawal, and all these other effects can be minimized. Again, the ingredients are key. People need to know, and that can only come through regulation, because it is a situation, I can say without a lot of fear of contradiction, that companies are not going to produce lab results unless it's mandated. No, it's all about the bottom line, and if something isn't required, companies are not going to spend the money. But there's a happy balance there. It has to come on the side of safety.
Yeah, I totally agree. And that's an area of things I've directly associated with. Spent a lot of time working in analytical labs, helping them get accredited and onboard methods. And I've seen the response from producers that, complain about, the testing that they're required to do and the cost of it. And, I'm always thinking about, like, what's that total cost of, the few $100s to $1,000 you might be spending on testing versus the value of the whole batch of material you have and everything. But it is a struggle. And even producers that say they value product quality and consumer safety and everything, I see a lot of that go out the door really quickly when they're allowed to kind of skirt by. It's unfortunate, but it is what it is. Going back to the endocannabinoid system a bit, and we talked about a couple of things, but what are some misconceptions that you think that people commonly have about the endocannabinoid system? I'm sure you have a few in mind.
Well, the main problem, again, is ignorance, just lack of awareness. And this is across the board. It really, man on the street probably doesn't know anything about it. But again, his physician won't likely either. So big part of my mission over the 23 years I've been doing this work has been education. But I think the message I'd like to impart is this is an important aspect of how we work. What can we do, even apart from drugs, to improve our endocannabinoid tone? And there's actually a lot. The paramount lifestyle approaches that would affect the system would be, first, aerobic exercise. We've got good data, especially from animals and limited in humans, that show that you improve your endocannabinoid tone and overall well-being through aerobic activity. Humans weren't designed to be sedentary, as is prevalent in modern society. And the other one, which seems to be taking on more and more importance, is the microbiome, the bacterial content of the gut. A lot of the ills of modern society are nutritional in origin. We're seeing a huge increase in things such as the metabolic syndrome, type 2 diabetes associated with obesity. Again, this is not normal in human populations. In contrast, we're seeing a situation in which the American diet in particular is pro-inflammatory, as well as having an improper balance of nutrients, generally too much. But a lot of this can be counteracted by having a better bacterial balance in the gut, and that can be attained through eating more vegetables, more fiber, and particularly use of probiotics and prebiotics. And this can make a tremendous difference in health and regulating the endocannabinoid system.
Yeah, and I was going to ask, is there any specific research that you're aware of that is currently looking at those links between the gut microbiome and cannabinoid receptor expression or endocannabinoid production?
Yeah, it's a developing area. There was a very interesting study that was done in Canada, Clooney et al., that showed that THC produces a better balance in the microbiome. We don't see development of obesity in animals that are administered THC. Rather, they remain lean even in the face of a high-fat diet and a genetic predisposition to that kind of problem. So it is a real developing area. I see that as really key and key lifestyle approach. Some people can use this kind of dietary approach and hopefully not even need cannabis quite so much.
Yeah, and one thing that's exciting to me is newer research coming out that's showing how different flavonoids interact with the endocannabinoid system, because it then opens this discussion up much more broadly to talk about. I mean, and there are multiple reasons for that. Flavonoids are one, but to talk about diet and consumption of different types of plants and everything that are also going to be affecting cannabinoid receptor signaling and endocannabinoid production, that sort of thing. And something that excites me about cannabis in general as a natural products guy, because that's really my background, doesn't come from cannabis, but I've adopted the realm of cannabis given what's going on these days, is I like that it's kind of a springboard for people learning about other plants and, other concepts and rediscovering that, maybe we should be exercising and eating better. And maybe that will help medically more so than a lot of other approaches. One of the last things I'd like to get into, check our time here. We got a little bit more time. Going back around to your work with Sativex and looping around, I'm sure a lot of people are thinking, okay, cannabis has therapeutic value. There's research going on. It's becoming more and more accepted. There's also a major push by a lot of companies right now to get cannabinoid pharmaceuticals developed. So can you, and I know it can be kind of complicated, but as briefly as you feel comfortable, kind of describe to listeners what it actually takes to get a cannabis-based pharmaceutical or a cannabinoid pharmaceutical actually developed, trialed, and approved? Because I think that process is a little more complicated than folks give it credit for.
Yeah, it sure is. In most respects, the development of a cannabis-based medicine as a pharmaceutical is the same for anything else. But cannabis is a botanical, a plant-based medicine. There's still a common misconception out there that you can't make a pharmaceutical from a plant, but it has been done. And even though Sativex isn't available in this country, it is approved in 30 other countries, and it passed the chemistry, manufacturing, and control, and the safety signals. But basically, what's involved is you have to take your preparation and show that it's safe through toxicology. This means giving large amounts, unfortunately, to rats and dogs and seeing what happens. Then it's put through phase 1, in which normal people are exposed to the drug in varying amounts, and you see how long it lasts in the body, in the blood, and what the reactions of people are, monitoring the side effects, if any. Then it goes to phase 2. In this, moderate numbers of patients with a specific disease are given the drug. And again, you're looking for efficacy, safety, and then the other factor is consistency. Can you show that your drug, if it's a botanical, it's going to contain many ingredients? You have to show consistency of each component within very tight tolerances over the duration of the drug development program. The final step is phase 3, in which large numbers of people with the disease are given the medicine, again, to show safety, efficacy, and consistency. At a point where a drug is past all those steps, it's FDA approved, and then it takes about a year to designate a schedule. You know, if it's a drug that has psychoactive effects, it has to be put in a schedule that describes what its drug abuse liability is, whether it has addictive potential, how often a physician can prescribe it, et cetera, et cetera. And then label is developed and it goes on the market. So we're talking about, generally from start to finish, a 7 to 10 year proposition that will cost anywhere between $700 million and $1.2 billion. Easy, right?
Right. Yeah. Yeah. No problem.
But so that's one aspect of cannabis as medicine. However, as we're seeing now in this country, cannabidiol has been available in various preparations on another level where people can access it without prescription. So in the future, there are going to be other cannabis-based supplements. These should be also subject to safety and efficacy issues, as well as labeling. And we've got a final level or echelon of activity, which would be someone grows cannabis and may use it in a medical context. So there are these 3 different ways of getting cannabis medicine. I think people make a mistake when they think that one level can eliminate the others, much as the government thought that Marinol, synthetic THC, would reduce or eliminate the issue of medical cannabis. It never happened. It never will happen. Similarly, we've got some counterculture people that would just like to free the herb, so to speak, and forget about these other areas of commerce. God forbid a company would make money on something that is God-given. But again, those are extremes of philosophy, I think.
Yeah, I agree completely. And there are some, like you alluded to, already examples of how botanicals can be treated in a highly controlled manner. I mean, one example is looking internationally at how drugs are handled differently. But even in the United States, I mean, you can get, like a standardized ginkgo biloba extract.
Absolutely.
And get a prescription for it.
Right. And when I'm advising patients, I indicate just that. The best ginkgo biloba extracts are EGB 61. These are developed in Germany, where they have a thing called Commission E, which is analogous to Food and Drug Administration for herbal products. So these things can be as safe and effective, or even more so, than new chemical entities and prescription drugs.
Yeah, I guess we'll just see how the FDA decides to wrap their minds around all of this.
Well, I can guarantee it'll be a slow process because the level of understanding is not there, and it's hampered greatly by residual prejudices.
Yeah. I think there was an assumption when the Farm Bill passed that all of a sudden CBD was legal and we were going to be able to make dietary supplements.
Well, yeah, I mean, there's friction in the law. Congress said it's legal. However, the FDA and the DEA still consider all forms of cannabidiol, apart from Epidiolex, as Schedule I forbidden compounds.
Yeah, and the last thing that I've heard privately from folks that are trying to interact with the working groups with the FDA and CBD is that they're not very optimistic that the FDA is going to provide much allowance for CBD and dietary supplements and foods and that sort of thing.
No, we've seen this in Europe already. They have a novel foods regulation that basically is eliminated most CBD containing products from the shelves.
Yeah. So we'll see how it goes. Before we sign off here, because I want to make sure to get you going. We have a conference to get to and everything. Is there anything we haven't talked about that in the last 2 or 3 minutes here you want to make sure to share with anybody, or even just bringing up some of the work you're doing that people can learn more?
Sure. Well, a big focus of my attention right now is trying to educate, and I like to use the catch term, making cannabis safer and better. Mm-hmm. And that encompasses a lot of different approaches. But again, I think we need better genetics for cannabis. This is a very malleable plant.
Yeah.
We can make it do anything that we want. There is not the need, in my opinion, for genetic modification of cannabis or to make it from yeast. Mm-hmm. The full entourage of compounds in cannabis can be optimized by selective breeding without resorting to those other approaches. And the future is very bright in terms of what can be done. Again, I'd emphasize safety standards should be paramount. Consumers need to be asking hard questions, not just of their doctors, but of the purveyors of these products. And I think marshaling our forces together, we can greatly improve the situation as well as the public health.
Yeah, I totally agree. I know we still have a long road ahead as far as education goes and everything, but there's progress being made all the time, and I greatly appreciate the work you're doing. I hope our paths cross again, and perhaps, maybe sometime in the future we'll be able to do some educating together. Thanks so much for being willing to spend the time this morning to get together and talk with me. I've really enjoyed it, as I knew I would. And I look forward to catching up with you again sometime.
My pleasure.
All right. Thanks, everybody, for listening. If you want to learn more, you can go to cacpodcast.com, or you can connect with us on social media. Just search for Curious About Cannabis and you'll find us. Thanks so much and have a great day. Take it easy.
Hey, everybody, this is Jason Wilson with the Curious About Cannabis podcast. Thanks so much for tuning in once again. So today I'm joined with Murphy Murray, a fellow cannabis educator and also a cannabis extraction consultant. And today we're going to be talking all about, we'll see what we get into, but primarily cannabis extraction. Thanks so much, Murphy, for being willing to come on the podcast.
Yeah, absolutely. Thank you for having me.
Yeah, I'm really stoked. Our paths hadn't crossed before now, so this has kind of been long awaited for me, as far as I've followed some of your work and seen your classes like from a distance. So it's cool to finally connect and talk about common interests. For those that aren't familiar with some of your work, and because your background's kind of diverse, do you mind kind of sharing just a little bit about some of that background and what led you into getting into specifically like the cannabis chemistry and extraction and all of the work that you're kind of focusing on now?
Yeah, sure. I started in the cannabis industry when it was fledgling in Colorado. So we had the caregiver structure in 2009, and we started seeing retail dispensaries open up, but there wasn't licensing for it, and it still kind of mimicked what California has going on at the time in a lot of ways. And so, I got involved on the retail end. I lived in the Vail Valley, and we wanted to sell high-end cannabis products to those high-end medical patients. And in 2010, licensing started, and in Colorado, that meant vertical integration. So being from a marketing background and working on the retail side of things, I had no preparation for an extraction lab, and certainly no preparation for large-scale cultivation. And so it was all just something that was kind of thrown at us, and we had to get involved. I did my best to delegate where I could, but the lab was one of the hardest ones to delegate out, because there, a lot of people had grown for decades.
Yeah.
But there wasn't much for extraction historically. Like a lot of people were doing bubble hash, but that is not even, in the same department, especially at the time. We were just starting to see things like amber glass, which was like a shatter made from ethanol, you know, and we were starting to see the butane honey oil, and we were still really just calling it honey oil at the time and not even really referencing how it was being made, because everything was still a big secret for a really long time, which is part of the reason why education is so important to me, because the first 3 to 4 years of my cannabis career, especially in the extraction world, there was nowhere to get good information. Yeah. There was a couple of hard to navigate forums with a lot of code words and screen names. And it was really difficult to use that information in a practical way. And not having that chemistry background, was certainly a disadvantage. But at that time, we weren't doing chemistry. We were barely extracting, material. We were leaving a lot of cannabinoids behind. Yeah. We were not purifying anything, and we had no analytical testing to support it. So, the concentrates that we made, we had no potency data on. We had no pesticide data. We had no heavy metal testing. I couldn't tell you how many milligrams of THC or anything else. It was all anecdotal. It was all descriptive. And, I look back at that and just get anxious about all of the products that I made and sold that I wouldn't do today, because we just didn't have the tools. Yeah. So, fast forward a few more years, and the internet makes things a lot easier. 10 years is a long time in terms of internet development. And so more information became accessible, which is good and bad. A lot of bad information became accessible, but what is more relevant is that we have the social media aspect of it. And so now I could actually network with real humans. And as more states became legalized, people were less afraid to actually share what they were doing. And real consultant jobs started becoming a thing. And so we started to talk a lot more and develop methods. And that's where I kind of got into the extraction space, in a very much more serious way, because we started to actually have real standards to pursue. And I had started to meet the type of people who were doing things that I wanted to emulate. And from there, I got involved on the equipment end of things. In 2014, I started working for Extraction Tech Solutions, which was one of the only hydrocarbon equipment manufacturers at the time. And I started doing private consulting. From there, I just became fully immersed in the world of extraction. So from, just straight up cannabis extraction to also the burgeoning hemp industry. Yeah. And, we've just kind of gone all the way from black oil to white powder in the last few years.
Yeah, it'd be fascinating to kind of see that laid out on an image timeline.
My Facebook memories can be scary sometimes. I don't know.
What is it that particularly about extraction now that's really driving your passion to kind of continue that focus in that arm of things?
I think one of my favorite things about extraction has always been that it's very objective. Once we started bringing analytical testing into it and started actually doing chemistry in the labs, it was really rewarding because I could perform a process, I could get results, and I could repeat those results. And compare that to like cultivation, where every step you take today, you see the results of that weeks, months ahead of that point. So there's no instant gratification in cultivation, whereas extraction is instant gratification. Every 30 minutes, I get to see the results of what I've achieved. And so it feels very productive. Yeah. And it also gives me a lot of room for error, which is very exciting for me because I love the experimentation of it. Yeah. With cultivation, even just having a table that you try out new nutrients on can affect literally the rest of the garden. Whereas with extraction, I can have a new idea. I can try a new piece of equipment. I can tweak a process. I can make these changes, minute to minute, day to day, and get to evaluate the efficiency. And so the potential for growth is just phenomenal because it's exponential. Every single day I can try something new, and every new test result gives me 10 more questions to go chase down the rabbit hole. So it is constant change and very fast moving, which I find personally rewarding. I'm the type of person that rearranges my furniture often. I love change. Yep. And extraction is nothing but change.
Yeah, I could see that. That'd be really exciting to be caught up in that accelerated process of refinement of technologies and process and everything. You really get to see it unfold in a way that's unique. When you were learning about extraction, what were some of the resources that you found most valuable to kind of understand what you needed to do to take things to that next level? I know Skunk Farm is obviously one resource that we all probably know pretty well. That's a huge one. But what were some others that you found valuable?
I mean, those online forums were enormous, but through those online forums, I actually got to meet really competent people. Yeah. And I think both 10 years ago and even today, the mentorship from finding someone smarter and better than you is the most valuable. Like, I look back on my career to some specific individuals who brought me in and were willing to, share what at the time was like trade secrets, like the things that we just didn't discuss out loud, in part because it was illegal, but also in part, to keep our brands, unique. And those people were enormous for me. I had a couple of chemists who were involved early on that were really helpful in explaining some of like the basic safety issues that I would never have even thought to ask questions about, probably without their help. Not to mention just some, industry pioneers, people like Nick Tannum of Essential Extracts, who, was willing to teach me how to make bubble hash in his kitchen and, get me involved in this industry when it was still barely an industry. We were barely for profit at the time.
Right.
So.
Yeah. That's I think that's really, really good advice to anybody listening that's trying to figure out how to kind of break into the space. I've always been a fan of mentorship too. That's how I've gotten to where I am as well. It's like I can think of like really 3 key people that, I just happened to sort of serendipitously come across that felt comfortable and taught me things or gave me chances that all of a sudden, progress into things you can't imagine.
Yeah, absolutely. And we all learn that way. There's this common misconception in cannabis that like you've just got to be good at it. That right. Like some of us are just gonna like stumble into a garden, get gifted the right clone, and the rest of it's gravy, or like there's some kind of magic secret to it. But really, like, I've got one math teacher who made algebra click for me. Like I remember 2 English teachers that, made an impact on my love of literature or my language today. Most of the time, like, we need someone else to help us, cultivate our passion. And I think that's really valuable. For example, the person who started my extraction career, Nicholas Tannum, he works in a completely different section of the industry now. Like learning under him, kind of gave me the insight that I needed to know where I wanted to go and that it was different, these kinds of lessons are something that I wouldn't have learned if I didn't get that exposure early on. And, that's, as an educator, that's one thing that I see most in students is that people think that it's supposed to come easily.
Mm-hmm.
But most of the time, if you're going to learn something, you need a teacher, and that's perfectly okay, so seek out a mentor that you can have those open conversations with. It's so enormously helpful. Like the internet is still just talking to a wall a lot of times.
Yeah, it is.
Like Google is too much information. If I ask my friend what to Google, they're going to give me better search words.
Right. Yeah, no, it's a really good point. And a big part of the role of a mentor is sort of helping you craft perspective. And so, when you work with someone like that, that's knowledgeable, you walk away seeing things just totally different. And so you recognize different opportunities that you would have never seen before, or recognized where you fit, I guess, into a grander scheme of things. That can be really, really valuable. Yeah. What sort of extraction technologies did you work with along the way? You've mentioned a couple, but I know, isn't your focus now primarily on liquid hydrocarbons? Is that right?
Yep, absolutely. So my focus since about 2011 has been, exclusively butane and propane. Part of that comes from the trends in the industry. Most of the smokable products on the shelves are originating from butane and propane because it's one of the few methods of extraction that allows us to preserve the acid form of our cannabinoids, the non-decarboxylated products, which I think you're going to hear a lot more about in terms of, medical applications over the next year here. So I've been, predominantly focused on that. However, the development of the hemp industry called for larger scale than butane and propane is currently poised to accommodate. So I had to come back to ethanol extraction in a much more sophisticated way than the ethanol extraction that I started with, many years ago. So, while I got into it with, the most accessible stuff, just straight up bubble bags and water hash, I've been on the combustible solvent end of things for most of my cannabis career.
Yeah. And one of the, I guess, issues that people usually raise with hydrocarbons is the safety issue. So, and you mentioned earlier that that was a big part of your learning, is trying to figure out how to do a lot of this work safely. So what are some of those key safety issues that you have to be aware of working in that environment, and what do you do to mitigate those?
Sure. Well, I mean, the first thing that I've got to bring up is, that we have to respect the chemical solvents that we use.
Right. Yeah.
And I think that one thing most people getting into the industry don't take the time to value and appreciate is just reading a simple MSDS sheet. Reading the warnings literally on the labels of the products that we use. Because a lot of times a person shows us how to do something, and then we just try to repeat their process and we don't try to even give ourselves the whole picture. It's kind of like when you learn to drive a car, like someone teaches you how to, hit the gas and hit the brake, but you don't necessarily learn how the engine works.
Right.
If you did understand how the engine works, you'd be a better driver.
Yeah.
But a lot of times we leave that first part of the puzzle out, and we just teach everyone to be operators, which is not quite enough information. I'm one of the few people that I have met with the number of years of experience that I do in extraction that didn't start with open blasting these solvents. I always operated within a closed system. It never occurred to me to get involved in open blasting, because the first thing I understood about my solvent was that it was highly combustible. And so I needed to protect myself from it.
Right. And
so my career has always been, on that equipment side and that technology side, because it just came to me as a given. The people who first introduced me to solvent-based extraction showed me CO2 extractors and, closed ethanol systems. And so it just never occurred to me to go any other route. I started right out with that understanding of, like, every new solvent, you need to read what it says, because some solvents are poisonous and make you die, and some solvents are just flammable, and so then they're dangerous, and some things are inert and not a big deal. And understanding the difference between them is your life.
Right. Yeah, I know. That's the issue of needing to read the safety data sheets is something that's come up in multiple ways in my world, because on the extraction world and also in the product manufacturing world, just for trying to understand the safety of ingredients that are going into cannabis products. So there's been, I don't know, I've seen multiple examples on Instagram where someone will actually post a safety data sheet and claim that, like, look how safe this is because it's grass. And then you look at the intended uses and it's like topical application, like maybe hasn't even been studied, to be consumed or inhaled or something like that. So yeah, I think that's a really good recommendation to start, is actually read your safety data sheets for your materials you're handling. Absolutely.
Yeah, it's the most accessible information in the world. And so without it, you're absolutely at a disadvantage just from day one. One thing that I always recommend, even with like cleaning products or dry materials, is that you've got to pay attention, to like the cleanup procedures, to the waste procedures. One thing a lot of gardens are very guilty of is improper waste disposal, and nobody talks about it because no one read the information that said this is really harmful to just dispose of and put down the drain.
Yeah.
So,
so much that's dangerous to aquatic wildlife.
Right.
You lose the biodiversity of life in a river stream, then you're destroying the quality of your water overall.
No, absolutely. And in the same ways that, paying attention to what you put into your waste when you're growing can actually help you make better decisions with regards to what you put into your plants. We can have that same perspective on efficiencies in the lab if we pay attention to the waste disposal, because paying attention to that waste is actually what inspires the equipment manufacturers to invest in better solvent recovery.
Right.
It's all about, on one hand, keeping yourself safe, but on the other hand, being a good steward of the planet, and finding, a commercial or industrial application that doesn't just save you money, doesn't just keep people safe, but actually, reduces harm across the board.
Yeah. Yeah. And how would you describe, given that you've kind of been around all of these different extraction types, how would you briefly summarize some of the pros and cons to those different extraction methods? Like, what's sort of driving those decisions of why people are choosing one over the other, CO2 over hydrocarbons or over ethanol?
Sure. So the first question that everyone has to ask themselves is, what do you want to make?
Yeah.
Your end product is really important. And a really quick and easy way to simplify that is, does your final product need to have the original terpene and flavor profile from the plant? Yes or no? And, a yes to that answer means that you're looking at CO2 or liquid hydrocarbons. If it's a no, then you can look at CO2, liquid hydrocarbons, or potentially ethanol or heptane or, some of these other products. If you, the other question you have to ask yourself is, what type of cannabinoids are you looking for? Do you need the acid form? Do you need those, crystalline solids? Or are you looking for a decarboxylated product? Because certain extraction methods are also more likely to give you one or the other. And so those two questions will usually shorten your list right away. And so from there, ethanol is one of the best solvents suited for scale currently. And the reason for that is that so much ethanol extraction already exists in other industries. It's considered food safe.
Yeah.
And so there are, regulations and equipment in place that we can just straight up copy and, put into effect for cannabis. However, the boiling temperature for ethanol and, its interactions with water are really problematic, especially depending on the qualities of your inputs. And so you lose terpene content almost no matter what. And you are also at risk for some degradation and some pH issues, extraction of undesirable compounds like sugars. And so there's a lot of things to consider that are going to lead you to, first of all, a decarboxylated product, but also more than likely an isolated product. It's going to be harder for you to have a clean oil that doesn't call for a lot of post-processing and cleanup.
Yeah. That seems like a maybe that's like a misconception in some circles, that you would actually have like a wider diversity of some of these compounds in an ethanol extract or even a CO2 extract. But that's something I've run into in my own experience, working in some of these labs and seeing, like for CO2, a lot of times the material is decarboxylated before it ever, goes into the extractor. Right. And so obviously doing that process, you're losing terpenes, anything volatile, you're losing a lot of volatiles, especially those really lightweight ones. And then with ethanol, before I really got involved in some of this work, I didn't realize how much refinement went into ethanol extracts to clean them up.
Right.
And now I've learned a lot of that pathway leads to just making distillates or isolates.
Exactly.
So it's fascinating, because before I got experience in some of this, I had heard, I don't know, just people speaking very generally that, well, of course, your ethanol extracts are going to have more phytochemical diversity in them, or with CO2, that you can target these fractions. And it's like, yeah, that's true, but everything comes at a cost with each of these technologies.
Right. And so, the drawbacks are, definitely very chemical. But the benefit is that if you want to process 5,000 pounds or more per day of raw material, ethanol is your current best option. However, that 5,000 pounds that you extracted that day isn't ready for sale that day.
Right.
It's still in solution, most likely.
It's a big picture.
Exactly. So, there's still more days in that final product production end. Mm-hmm. If we look at CO2 is a very complicated product because CO2 is a flexible solvent. Mm-hmm. It relies completely on the operator and the equipment having a plan and sticking to it. And so CO2 is capable of being an excellent solvent for terpene extraction. It's also capable of extracting a lot of water and wax and nonsense, just depending on, how that equipment is used and how efficient that process is. A lot of people decarboxylate their material, like you mentioned, to avoid the water issues, which means sacrificing terpene content, which is a bummer because CO2 is capable of doing probably one of the best jobs with terpenes as far as solvent extraction would go. But, it comes generally with the cost of still requiring winterization in ethanol. Yep. So I tend to put CO2 and ethanol together because whether you make your crude with ethanol or you make it with CO2, you probably are going to be following the post-processing path. That's the same either way, which would be that winterization and filtration, and then probably most likely distillation.
Right. Yeah.
And the exception to not going to distillate is usually still just to be in that raw oil form. So maybe you might not distill, but it's still only available for pens or for, food. It's still not, it's still a limited product as far as what you can do with it.
Yeah.
And so hydrocarbon is a lot more flexible in that regard because while it is a very small input batch size, we're doing, 500 pounds or less per day in the average lab, which is not massive scale. We're not going to feed the world with that. However, I don't have any of that week-long post-processing that follows it up. Mm-hmm. So while I might only get to process 500 pounds that day, I could sell the oil that I made that day the next day. Yeah. Which is, a big difference. And it's something that, we often skip when we look at our production plans is, how many, like we think about how much can this equipment do, per pound, per hour, whatever. But we have to bring it all the way back and say, okay, well, all of this equipment in a straight line is going to take how long from raw material to packaged in for sale?
Exactly.
And hydrocarbon is absolutely the fastest for that. Butane and propane evaporate at very low temperatures, which means we get the acid form, which means I can go to Delta 9. I can distill it. I can make a tincture. I can make edibles, or I can make all of these other things that I can't do as easily or as quickly with CO2 or ethanol.
Yeah.
So that that post-processing flexibility is the primary advantage there. It is, however, combustible.
Yeah.
And so, with ethanol, we have the flammable issue. And with CO2, we have the high pressure issue. Mm-hmm. With the liquid hydrocarbons, we have both.
Yeah.
So, the risks are significant, and the risks are only mitigated via equipment and safe workspace. There's no safe way to just be careful,
Yeah.
And statistically speaking, for everyone who is going to listen to this and think, well, I've been doing it for years. Statistically speaking, the more days you've gotten away with it, mm-hmm. the closer you are getting to the day when you won't. It will come. And so, the safety risks are significant because it is combustible in nature.
Yeah.
However, it is a clean extraction method. Butane and propane don't dissolve mold. They mm-hmm. can't carry over, water as easily. So the final product, because it's very easy to remove the solvent, and the solvents are food safe, and our exposure to them is generally non-toxic. They're very safe products. Whereas with ethanol, high levels of residual ethanol can certainly be harmful, especially for oral ingestion. And, that high level of water content in a lot of CO2 products can lead to microbial growth.
Yeah,
and so we have, some pros and cons both on the processing side mm-hmm. as well as for the actual consumer. Because if I need a product that can store for a very long time with limited degradation, a hydrocarbon extracted product probably has a better shelf life than an ethanol extracted product or a CO2 extracted product based on literally just their moisture content.
Yeah. Yeah. No, that's a excellent summary. And I think something that consumers take for granted is that things like microorganisms can grow on extracts in certain right. environments. I've definitely seen it. I mean, working in the testing lab, I'll never forget the first time I saw it was essentially a moldy CO2 extract. Mm-hmm. I'd never come across that before, but now I realize it's less common now that people have kind of learned more, how
right.
to do their processes better. But yeah, that was something that was a shock to me, and I'm sure is a shock to some people listening that haven't encountered that before. And I'm glad you touched on the issue around the safety of the solvents, because that was going to be something I was going to ask you is some people, when they're first learning about extracts, they feel put off by the hydrocarbon extracts because they feel like there's a higher safety risk due to the solvent load in there. Right. So do you have any advice you could offer listeners that if they're worried about solvents in a product, what sort of levels should they sort of be looking for? What's typical ideally in a well-purged extract, you maybe wouldn't have anything detectable or very, very low parts per billion. But what are your thoughts on that if someone asked you, what's a safe exposure level?
So the first thing that everyone should do is the first thing I mentioned in this interview. Go read the MSDS for the solvent that you're wondering about, first of all, because the health risks that are associated with it are going to be on that sheet, and it gives a range. So that gives you the acute exposure range, the moderate, or the persistent exposure. And you want to see what your, best and worst case scenarios are. That acute exposure of, 200 parts per million butane in your product is largely non-harmful. Just breathe fresh air. You're fine.
Right.
And that, day-to-day exposure of over 5,000 parts per million for 8 hours at a time or longer does lead to dizziness and, some other issues. And so you want to see the best and the worst. You want to see how much of this is it going to take for, nausea versus pass out mm-hmm. versus stroke and seizure.
Right.
You want to know what the actual ranges are, and that information is not a secret and it's not up for debate. For those solvents to exist and be used, that information has been studied. Now, it hasn't necessarily been studied for how hot of a dab you're going to take at home and how many grams you're going to smoke per day. These applications aren't necessarily standard, but our human exposure from inhalation or ingestion has been studied for these solvents to actually be sold and be legally allowed for us to use in extraction. Licensed labs can't use solvents that haven't been tested in that way.
Right.
So, so that data exists, and I would encourage everyone to go read it right away, because it's easy for us to all have opinions about the parts per million limit. And a lot of those are based on state regulations, which were formed from the opinions of people.
Yeah.
Yeah.
Sometimes they're pulled
specific criteria.
Yeah. Sometimes they're pulled from really weird places. Yeah.
Right. And so those numbers are not based on human studies. Those numbers are not based on known and proven and, calculated potential harm. They're just numbers. They have, about as much data behind them as the safe speed limit for a road. Like maybe 35 is safe in your car, and maybe in my car 45 is safe, and maybe with my mom driving my car 45 isn't safe. Like maybe there's more variables than just that number. So it's hard to rely on that, because you're going to see a really wide range. And so the state of Massachusetts says 10 parts per billion is what's safe for butane. And, the state of Oregon says 5,000 parts per million is the safe amount. And, neither of them are in charge of how much is actually safe for you and your body. And so the numbers are a very difficult one to compare. Obviously, we want to remove as much of those, solvents as we possibly can. However, most of these solvents like butane, propane, CO2, and ethanol are food safe products. When we get into things like pentane and hexane and heptane, or some of the compounds that are being used for conversions like iodine or toluene, like we've got some more significant health risks. And so, and those actually are more common in like CBD products where there's no regulation. Scarier issue that we could get into later.
Yeah.
But, for the most part, you're going to have those solvents that are used listed on the label. And so you can do a quick MSDS check and read what the best and worst could possibly be. And then compare that to the parts per million. And, we can put that math together, and we can say, 5,000 parts per million for 8 hours a day of exposure to butane is what, the MSDS recommends as being dangerous enough to actually make you ill and make you dizzy. And so if you are smoking a product that has 5,000 parts per million in it, don't smoke it for 8 hours a day.
Right.
Because, now we can start to at least say, okay, well, that's something that, a dab at a time might add up to that acute exposure of, however many parts per million. Every time we flick our lighter, that's about, 5,000 parts per million briefly. And so we're being exposed to butane and propane. It's in your air. CO2 is in your air. Ethanol is in almost every flavored food product you eat.
Yeah.
So like anything that has artificial colors probably contains ethanol, so like a lot of these solvents exist. It's not realistic to fear them. And it is really important to judge them with math.
Yeah.
Like you've got to pay attention to how much is there. There's a big difference between 20,000 parts per million of butane and, 20.
Right.
And so you want to pay attention to those numbers as best you can. But I would also encourage anyone, especially anyone who's smoking concentrates, to listen to their body, because the solvents themselves are known, are studied, and are tested for. What we cannot provide to people right now is a comprehensive look at the potential conversions that are happening, the byproducts of some of this exposure.
Yeah.
Just heating up a solution that's extremely, high pH in ethanol that contains a lot of water could create some, converted byproducts that become really dangerous.
Yep.
Using iodine for conversions is, efficient to a certain extent, but it creates a bond with cannabinoids that isn't tested for. And now we have a cannabinoid compound that has been bonded with a little bit of iodine, and that can get to your brain, and that could probably end your life. We don't know, but that's not something that would be tested for.
Right.
And so the risks around these solvents, are the greater concern. And since we can't necessarily apply a lot of data to that right now, listen to your body. If it hurts to smoke, stop it.
Yeah. No,
if it's uncomfortable, if it makes you feel weird, don't keep doing it. There are so many options out there that you should, listen to, the cues that your own physical chemistry is giving you. For the most part, like our instincts are generally right. And, even terpenes can degrade to benzene. So, like our favorite parts of cannabis could kill us.
Exactly.
So you've got to, be as educated as you can about the solvents. You don't have to know everything about extraction, but in the same way that you would look up a hormone that's added to your milk, you could look up a solvent that's used in your extraction, and then just pay attention to what your body tells you.
Yeah. I think that's a really good point. You don't have to know everything, but you should at least have the skill of knowing how to find the information when the time comes. If you need to look that up. Yeah. Well, shifting gears a little bit away from the safety side and talking about the products, one thing I wanted to ask, because it's something that I hear a question about a lot related to extraction. What affects the various, I guess we'll say, consistencies of hydrocarbon-based extracts? Because it's well known that there's a wide variety of sort of types of hydrocarbon extracts. And so can you speak a little bit to some of the dynamics that influence those final products?
Yeah, for sure. So one of the reasons that hydrocarbon products have that, wide range in texture and consistency is because they have the potential to extract the acid form. And for a lot of cannabinoids, the acid form and the decarboxylated form of that cannabinoid have different physical states. So for example, THCA is, a solid colorless crystalline product at room temperature, whereas, Delta 9 THC is, a colorless liquid at room temperature. And so that's a big difference in consistency. So if my concentrate is mostly THC Delta 9, I'm going to get sticky liquid because Delta 9 is a sticky liquid. But if I've got mostly THCA, I am going to have a primarily dry solid. And so, that first differentiation in ratio of, THC versus THCA or, CBD versus, THC makes a big difference in, just the potential consistency that I've got in front of me, especially because I don't want to say decarboxylation can't be reversed, but generally we're not going back from that. So once we have crossed into that decarboxylated state, once I've got CBD, I've got a solid. Once I've got Delta 9, I've got a liquid. And so the hydrocarbon extraction process allows us to play with that texture a little bit more because we can start with a dry product and go liquid or not.
Yeah.
So that already makes a big difference. It's just the dominant compound in any given solution is going to dictate, the structure that it takes on. Then from there, we have, the other minor groups that are involved. Now, hydrocarbon extraction is also excellent for terpene extraction because it happens at low temperature, so we don't destroy them, and they're soluble, in butane and propane, so we're able to capture a lot of them. We also have the added benefit through hydrocarbon extraction of being able to extract wet or fresh frozen material because it doesn't take on water content in the same way that 5,000 psi worth of CO2 or any given amount of ethanol will. So we have, the potential to not only extract more terpenes because they're compatible solvents, but also because we can capture terpenes from the plant material before it has dried out, before those have changed, before those have evaporated. And so that terpene content is mostly liquid. Some terpenes are solid at room temperature, but mostly we're dealing with liquids. And so the amount of terpene content in any given hydrocarbon extracted product has a huge impact on that liquid versus solid situation because those terpenes can dissolve that solid cannabinoid.
Yeah.
And so why we might have 90%, solid THCA cannabinoid content, we might still have something that looks half liquidy because those terpenes can actually dissolve quite a bit of it and keep it in solution. So, that recipe gives us a lot of options. And that's just based on what it contains, regardless of what I might do to it. If I just pour it out of my extractor and here it is.
Right.
Then from there, because we have that acid form, because we have some of these solids, we have the potential for crystallization. Or, in the opposite route, we have the potential for, an emulsified or homogenized product. And so, shatter, wax, butter, those kinds of things are generally homogeneous products. Right. Shatter is everything mixed up and, solid and flat, whereas the butter and the wax is everything whipped up and actually aerated. But in either case, basically the same original content, which is way more THCA than that liquid terpene content, which allows it to be, dry rather than wet. And then from there, it's just, how we play with it. It's at that point, it's kind of, how do you take your eggs? You like them scrambled, you want them sunny side up. So for me, I try to pay attention to that chemical content first. Because it's a good indicator. If I really care about flavor, I probably don't want dry textured products because inherently I'm going to have less.
Yeah, that makes perfect sense. And how does the material coming in affect some of that process? One of the things that I was going to ask you is, what it's like working. You mentioned fresh frozen, so like the difference between like different types of like live resin and cured resin, and these different attributes that relate to the flower material as it's coming in. What are some of the big differences there?
There are so many important qualities for your extracted product. I think one of the most obvious ones that a lot of people fail to pay attention to is surface area. When we look at big, pretty buds that, we're putting in a jar, we're selling on a shelf, we look at them like flowers, and we want them to have a nice clean cut. And we want, we have these very aesthetic, perceptions about what is good and what is bad. And that is super irrelevant to me for extraction. All I care about is resin content, and big, dense buds have less of it by weight than little fluffy buds that aren't very dense, because the surface area is greater by weight on those products. And so, the type of material that you want to provide for extraction isn't these big, dense, rock solid nuggets. Those are annoying. Those take me time to break up. And it's literally more plant material versus resin. If I, grind up a big dense bud, homogenize it, and get an analytical test on that, I get totally different results than if I take the same amount of weight of small little fluffy buds, grind it up and homogenize it. They literally have more resin. And that's all that matters to me for extraction. However, where it grows on the plant certainly has an impact on the other chemical content that it's going to contain, especially terpene content that is produced generally to protect the plant from its local environment. And so it would only stand to reason that the buds on top are going to smell, and respond differently than the ones on the bottom, because they've got different jobs as far as that plant is concerned, and they've got different enemies. So, so there's definitely a really important role that just the actual harvested nug plays, not only in where it comes from on the plant, but the surface area, the density, and even the time of harvest. Every single day that you push that harvest, it produces different terpene content. It does, that terpene content doesn't store up the way that cannabinoid content does. It doesn't accumulate. And so if you harvest on a day when it isn't smelling amazing, that's all I've got to work with. And if you harvest on a day when it smells outstanding and you freeze instantly, I can get that. I can get that back. I can preserve it. So, the care that's taken during harvest is incredibly important for some of those volatile compounds. And I try to pay very close attention to that. And the people that we want to work with are people who are also going to care about that. It's difficult to get that kind of attention and care for your products if you're just, massively harvesting everything on a 45 schedule, regardless of what's happening in the room. And so sometimes that kind of massive factory approach to it is not ideal for some of the boutique flavors. However, if we're just trying to extract cannabinoid content, well, we can get it from whatever you've got.
Right.
There's still some other things to consider, like literally the density of the cuticle, how much wax content that trichome contains. If you've got really thick trichomes, I'm probably going to get lower yields from your material, not only because it probably just contains less resin, but also because the resin is going to be less accessible to me, because removing waxes
you're going to lose some of that
almost the same process as removing cannabinoids. And so it becomes more difficult to get, or therefore at least less cost efficient to get a really clean yield. And so, some of that chemical content is, really valuable. And that's something that the people who work in like the rosin space are starting to pay a lot of attention to, because high wax content is, one of the highest risks associated with their products. And it also contributes to their low yields. And so if they can identify that, wax content while they're growing it, they could impact it, they could change it, they could improve on it and reduce it, and actually grow plants that could produce 5-6%, yields on that rosin, which would change their business model completely.
Right. Entirely.
So, so those things, affect all of us, not just rosin. That's also something that's important for us with hydrocarbon extracted products. And so that quality is really important. The other thing I think that would be important to pay attention to is transparency. We only know that it's free of the pesticides we test for.
Yeah. Yep.
We only know that it's free of the mycotoxins that we test for at the time we tested it.
Yeah. Yep.
Like a lot of that data is not quite complete. And so we have to be able to trust who we work with. And we all have to be open about that trust, because sometimes we don't know that we're doing something wrong.
Yep.
Sometimes it is leaving their facility clean and getting moldy in my, fridge, like my freezer or fridge storage. So we all have to be able to be open with each other and work together to resolve those issues. It's really difficult to do a good job if you don't have someone who's willing to be transparent, bring you into the garden, and involve you in that harvest process, so that you can both make clean and sustainable products.
Yeah, no, totally. I remember when I was working in the testing labs, how sad it was when, a cultivator would end up with a testing result that they weren't expecting.
Right.
Or kind of like what you're mentioning, like in a wholesale situation where the farmer sold it, it was good. When it went to the wholesaler, it sat at the wholesaler's facility for who knows how long. They retested it, and it comes back, and then they're trying to hunt down what happened, who's at fault. And sometimes there's a lot of money involved in, some of these transactions. And sometimes it was devastating to see some of these farms that actually were putting in a lot of good energy trying to do things as best as possible, and then somewhere along the chain things go wrong and they don't understand why, and sometimes couldn't figure it out, what happened. Like somebody wouldn't fess up to some part of something, and sometimes it's really things you wouldn't necessarily expect. One time I remember a guy had failed for pesticides and then realized that someone on his garden used a hose that was used on another farm, that sprayed pesticides, and he just didn't keep track of, that hose. So yeah, it's a process that the industry's, learning all of these nuanced pieces of it, because you can understand the high level of like, okay, I'm not going to use pesticides, I'm not going to do this, etc. And then all these other factors come into play that you don't imagine.
Right. And it's easy to villainize, who's at fault, and say, like any failure is unacceptable, but but who's at fault is not as important as how to remediate
exactly
it. Yeah. Like what's most important is figuring out what happened and why, so that it can't happen again, not so that we can dissolve our relationship and say bad things about each other on the internet. Like the point is to move forward. We have all failed before.
Yep.
We have all done a bad job somewhere. You know, just because you've never failed a pesticide test doesn't mean you couldn't fail any pesticide test.
Right.
You've just never failed the tests you've taken, you know? And, you know, there's just so many ways that things can go wrong that, you know, we have to find people to work with that have the same, you know, ethics and morals and intentions as we do, so that we can work through them together. You know, like bad business deals are what sink otherwise, you know, strong, well-intended businesses. And a lot of that comes from this very, you know, like all in or all out kind of attitude that we tend to have, rather than working through it together to improve. You know, these relationships should be long-term.
Yeah.
And, you know, just because the industry is new doesn't mean we can't come at it with that kind of diplomacy and, you know, with a structure that allows us to, you know, prepare for things going wrong.
Right.
Because it does.
Yeah, we're human. Like, mistakes are going to happen, like, despite anyone's best effort. Things are going to go wrong. And you're right. It's how you respond to that and how you move forward that really is most critical. And in general, what most of us want to see is for the industry as a whole to learn and figure out its problem areas and then address those, teach, you know, as many people as we can to spread that knowledge, and then see the industry get, you know, better and better. It's to see quality improve and safety improve, and
absolutely
everybody, you know, mature along this industry together. So, but yeah, there's a lot of, like, I don't know, it's weird coming into the cannabis industry, seeing some of the like drama that goes on among the companies and the people, and so much, you know, egos fighting egos that it's weird. It's like being in middle school again sometimes.
Yeah. Well, the thing is that everyone makes it so personal.
Yeah.
But like, it took many persons, you know, to accomplish that. Even if it was one guy who used an old hose in this garden, you know, like that choice affected everyone. And so that means that everyone was responsible for making sure that there wasn't old hoses.
Right, exactly.
You know, like, yeah, he's the guy who used it, but there was probably 2 or 3 other humans who could have helped prevent it.
Right.
You know, and there was probably a few people down the line who could have done something about it. You know, like there's ways to work through it. And, you know, pointing the finger at who's at fault is just, it's like our, you know, our first petty instinct.
Yeah.
But it doesn't serve anyone, you know? And that goes, you know, for even small issues like, you know, getting low yields. I would rather go back to a farmer and say, you know, my yields are poor from this material. You know, like, let's work on a way to improve this, not say, I'm not going to pay you this much for this material and I'll never do business with you again, you know, which is, which is how most people would respond, is they would get, you know, they would get upset. They would blame someone. They would say, you're just bad at this, you know, and they expect perfection. It's so much easier to work together and, you know, take someone who gives you low yields into giving you even higher yields than you get from your high yield suppliers, you know, at the same price you started with. You know, like these long-term relationships keep us all in business. And I think they're the only things that'll protect us from large financial interests replacing us later, because they will have to do it all themselves. If we don't work together, we're not going to compete.
Yeah. No, I think that is absolutely the case. And, you know, it's cool to see organizations like, you know, from the Future 4200 Forum, the Good Life Gang, you know, to see these companies that are sharing SOPs and, you know, information and trying to create that robust network, you know, that'll hopefully be able to survive. That's been really cool to see. And even, you know, on the farm side too, we're seeing more and more kind of farms that are trying to find ways to work together and kind of co-op situations, figure out how to not necessarily have to fall into that trap of like large monoculture, you know, big ag scale cultivating, be able to keep doing things the way they want in a more sustainable way, but work together to provide enough material that, you know, these accounts that want to buy massive amounts at once, they'll still have their, you know, their weight there. But I wanted to check in with you for just a minute because we've been going for almost an hour. How are you on time?
Yeah. I'm good for right now.
Cool. Okay, excellent. Well, the next question I wanted to ask you is related to a hashtag that you make a lot about your classes that I'm just probably ignorant and don't know what it means. So I wanted to ask you while I was talking to you. Aquatech, what does that mean?
So Aquatech is kind of a marketing term. And basically it refers to the methodology that we developed, which is a chromatography application using, butane and propane right in the column. And so the idea was to perform that, chromatography separation in the original extraction solvent, although you can apply this to crude oil that's been extracted from almost anything else. It just poses different issues.
Sure.
All which are fun challenges to work through.
Right. Yeah, exactly. Puzzles.
Yep. But yeah, so the idea there is that using chromatography, we are able to have our liquid phase, our solution, moving through a solid phase of media. And that media is going to separate our solution by polarity, which allows us to extract our terpenes very quickly, very cleanly, get them out of the extractor and away from heat right away. And then we get to pull apart the rest of our solution. And so depending on the column that you build and the media that you use and the parameters that you put in place, we're modeling this after flash chromatography.
Yep, that's what I was
But
thinking.
it allows us to separate, kind of by category first, terpenes, cannabinoids, waxes, and, water solubles. So we're able to get extremely high purity crystallization results almost instantly because we've reduced our solution down to simpler parts.
Yeah.
Which is exciting from, an efficiency perspective, because it means even less post-processing from,
exactly.
Which, crystallization is already nothing, that's an easy post-process for most people. And, even in the pharmaceutical world, that's something that we've, got down. But this is just a fantastic shortcut. You're 99% right out of the extractor, almost every time.
Yeah.
But then it also does allow us to pull that terpene content out separately. So it's not interfering with crystallization and also not exposed to some of the same temperatures and conditions that we would potentially expose our cannabinoids to. So that allows me to remove the butane and propane at different temperatures, and under different conditions than it takes for me to remove butane and propane from, that crystalline dry product.
Sure,
so now I get to treat each of these compounds appropriately and bring them back together later. So anything that's Aquatech is a formulated product. It didn't come out of the extractor that way. It came out of the extractor in multiple fractions, and we pulled the best parts out of it, which are subjective. I decided what I liked the best for the most part. It's also based on analytical testing and, where the clean parts of our product are. But in that way, we're able to pull flavonoid content. We're able to pull terpene content. We're able to get high purity cannabinoid content. And then we can deliver it back to the consumer in consistent ratios.
Yep.
So, when your question earlier of explain these different textures, a lot of times those textures are based on what came out of the extractor. So if I didn't get a lot of terpenes, but I got a lot of cannabinoids, here you go. I've got a whole bunch of dry wax, or I've got really stable shatter. And if I do get a lot of terpenes and I still have a decent amount of cannabinoids, now I've got wet sugar. Now I've got greasy batter. Now I have shatter that won't stabilize, but I'm taking it from the entire other approach, which is get everything out of the plant, pull it into my different jars. Now I have ingredients and I make whatever I want. So I can make stable shatter from there. I can redissolve my THCA content back into butane and propane and pour it out as a slab, or I can whip it up together with some terpenes, or I can crystallize it into diamonds. I can decarb it. And, now I can have Delta 9 and THC, I can make whatever I want every single time. And I can still preserve the live resin profile that the plant gave me. But I can make whatever I want every single time, which is an enormous value, because sometimes you don't get the ratios of things and that you're looking for. It's really common to have more THCA content than pairs nicely with terpene content. And so it's great to not just have to dilute the flavor. I can just pull that THCA aside, put that into Delta 9 and, put it in carts. I can do whatever I want with it and, maintain that profile of the plant with a consistent cannabinoid content, which especially for medical patients is important because they, are only just now starting to figure out why sativa or indica is too broad of a, category. And so we start telling them to pay attention to these terpenes, but they do also still have to pay attention to the dose. The cannabinoid dose matters.
Yep.
And so if I can provide the same cannabinoid content every time, but different flavors, I can't say that it's good for anything. I'm not allowed to make any health claims.
Right.
But if I'm consistent, then you can figure out what works for you.
Exactly.
And you can take a bigger or a smaller dab. Or you could tell me that ratio is not cool, and I can literally change it.
Right.
I can make whatever I want.
Well, that's really exciting because I mean you're talking about making standardized product, which is so needed in the industry. And when I'm doing seminars or teaching folks, I always talk about journaling as far as when people talk about what'll work for this or that or the other. I'm like, the only way you're going to figure that out is by paying attention to how your body reacts to different things, and journaling's a great way. And one of the limitations that people are running into with journaling is the inconsistent product formulations. If they prefer extracts, they will usually hone in on maybe just the THC or CBD values, and maybe won't focus too much on other things, or they are paying attention to terpenes and anything else they can get data on, but batch to batch is just changing.
Right.
So that's really exciting because that it fixes a variable and can allow people to...
I'm just trying to cut them down.
Right.
We've only got like 300 variables per batch, and
right.
I just want just one fewer if I can make it work. And anecdotal data or journaling is still data. Like all we need to understand it is more data. It is a lot more data, but it's attainable. And so even if every person who smoked wrote down how it made them feel and we fed that to a computer, the algorithm would be accurate.
Yeah.
Like it would tell us what it does.
Right.
So wherever that data comes from, it's extremely valuable. And especially for the people who need it most, write it down. I mean, I can't be expected to remember what temperature I did something at during my extraction. I write it down.
Yep.
You cannot be expected to remember how fast your headache went away when you were high at the time. These are things that we really benefit from if people can write it down. And more data is available than ever before. We go a step above and put terpene content on every single package that we sell. So every single terpene is listed along with I shouldn't say every single terpene we can test for is listed, as well as the ratio. And then all of that's available on our website too. So even if you just write down, I tried this Kush Mints batch this one time, you can go back in time, look on our website and figure out, that Kush Mints had a lot of CBG.
Yeah.
In addition to having a lot of pinene, terpinolene, whatever, you can start to pull all of the pieces of data together. And the more data I get, the more data I'll put out. But I don't have the ability to predict what it's going to do for anyone. I can't say that it's good or bad. I'm relying on the plant itself being ideally safe and giving me, a profile of chemical content that is beneficial. And the only way I'll know is after people provide feedback on that. And so, I believe strongly in my method because it allows me to make changes, and I'm not just stuck with whatever has happened inside of my extraction system, that I can make it work for people, which is really exciting. I mean, I love sweet flavors and good dabs. I love that it's available recreationally, but, the medical products that we produce are, generally a disservice to the people who need them most. And so I hope that through this method, we're able to make, the right chemical profiles super cheap for people. I think it sucks that, it's easier for people to get high THC products at a low price, but not 2-to-1 CBD THC products at a low price, I would love to figure out what those ratios are and make those medical products available through our current system rather than forcing the pharmaceutical industry to take on that role.
Yeah. Yeah, I agree. And the overall just cost of cannabis products generally for medical patients, I'd like to see some states try to fix that problem. It's something that in a lot of the doctors I've interviewed, they bring that issue up, that when I ask them about dosaging, a lot of times, interview I did very recently with a pain physician in North Carolina that's working with patients using CBD. He's like, well, our patients, the ones that are, really in severe chronic pain, a lot of times they're either poor or they're elderly. They don't have a ton of money to spend on, uh,
Yeah.
all of
On
these.
trial and error, first
Right.
of all,
Exactly.
let alone just the regular cost of these products.
Yeah, exactly. And so
usually they can afford to try it once, and if they don't get the results they're looking for, they
Right.
give up on it.
Exactly. Yeah. That's something that's been on my mind, is how many people have given up quickly, especially. So now we can loop back around to talk about the CBD industry, because that's where right now I see this like really highlighted, with so many people flocking to buy CBD products, and they're all over the board as far as quality and potency and pricing, with some of them extremely expensive. And you've got these people trying to treat very severe medical conditions that are spending $150, $200 on a 1 ounce bottle of a CBD tincture that may have like 300 or 500 milligrams in the whole bottle. It's really fascinating. I did want to actually come back around. You mentioned some of your thoughts about quality issues related to the CBD industry and the extractions going to CBD. So do you mind talking about that? What is unique about what's going on in the CBD industry, and what has you a little concerned?
Yeah, so the hemp world is extremely unique, and it's very strange. Because in the traditional, medical marijuana recreational markets, it's all these are all state programs, right? So of course they vary, because the states vary. A lot of these states have home rule, which means that not only the state rules are in effect, but also every municipality can do whatever they want. And so you see in that licensed structure a huge range of requirements, from the safety and architecture of the building all the way out to the solvents that are allowed to be used, and what is required to be tested for and what isn't. So the regulations are all over the place within that framework. In the CBD world, it's kind of this like extreme polar opposite land where we have some actual federal regulations in place. We have truly GMP, USDA, certified facilities that are manufacturing pharmaceutically pure compounds, start to finish. And then you have literally anyone, anywhere in the world, making a bath bomb in their living room and selling it as medicine, to whoever will buy it. Like there's, so that, that spectrum is absolutely insane, because there's not enforcement on anyone except the people who are doing perfect things.
Yeah.
And the people who are doing the perfect pharmaceutically pure compounds, those are all going to formulations, and, a lot of times there's like white labeling, or they're just selling a bulk product. And so, it doesn't mean that small businesses aren't, having access to that level of purity necessarily, but there's just no chain of custody monitoring whatsoever. And so, it's a really scary place to be. I've gotten about a dozen messages today already, because we have our first potential confirmed CBD death.
I saw that.
I don't know if you read about that. So we have no data about that. I don't even want to make any, comments, because I haven't been able to read enough about what was being referenced in there. But it's not surprising at all that drug interactions would be our first, actual cannabinoid-induced death, because drug interactions are known, and when we break it down, it's all just chemicals. So this one company that made this one product is going to burn alive, probably over this issue. But
affects everyone.
But it affects everyone, and it doesn't mean that they're the only ones who are creating a product that could cause this, much like the vape crisis of 2019. Like even now, we still don't know
Yeah.
that vitamin E acetate caused the death.
Exactly.
We just know that it was in all of their lungs. And not even all of them, just most, you know, like we still don't have
there's
explanations.
That common factor has still not really been identified.
Right. So we get more data and more data and more data, but we still don't have a complete picture. You know, we're still missing huge pieces in our puzzle. We just have to keep working forward on it. And so the CBD world is in a unique position to innovate faster. Because they don't have the same regulations that we do. They also are in a position to infiltrate the public domain faster and influence the public's opinion of how cannabinoids are bought and sold, which is exciting in a lot of ways. People make fun of gas station CBD, but I think that if we're going to sell cigarettes at a gas station, if we're going to sell liquor at a gas station, if we're going to sell sugar and caffeine at a gas station, sure, we should be able to sell weed at a gas station. Why not? All cannabinoids should be able to be sold at a gas station if one of them can. So I appreciate, the way that some of that underground CBD pushes the boundaries, in those, very honestly political ways, because it does, give me hope about a future where I can still make a craft concentrate that people can buy. Because the other end of that spectrum, where they're making 99.999% pure compounds and formulating them back into, a gel cap or whatever, is boring. That's not what I, like it's very cool to build. And then I don't want that factory job. I want to keep innovating new things. So, that pharmaceutical world should exist, but I want to see both. And, the things that happen in between those two that are dangerous jeopardize all of it.
Exactly.
Is really unfortunate, because only those big ones have the potential to survive that FDA world. But if we don't have the market to support them, then they all go out of business. And we're watching that happen this year in the hemp industry. We are seeing some of the biggest players who had the most money and the biggest extractors and just millions of dollars in infrastructure right now that aren't gonna do, they probably won't even plant. And so that isn't encouraging for our industry. I don't want to see the money fall out.
It's been wild the past year seeing how many big names are folding or going under a change of ownership, trying to totally restructure to stay alive. It's been really, really bizarre. And that CBD death, I just saw that several hours ago, or sometime this morning. It's one of those things that those of us that have kind of studied cannabinoids and particularly CBD for a while, we've all known that the grapefruit effect, is a thing with CBD, and, CBD seems to exhibit, a higher potency than even grapefruit does in having that effect. And it's one of those things that, the market seems to be too far ahead of where, the medical establishment is. So, a lot of doctors don't know that. A lot of nurses don't know that. And I feel like if they did, they would probably inquire more about whether patients are using CBD, when they're on other drugs, that sort of thing. And it's one of these risks that could be mitigated with education and proper involvement between, the patient and doctor relationship and that sort of thing. But it's also one of these things, like you're alluding to, that, something wrong can happen and it's going to affect the entire industry in major ways. If, people start dying from drug interactions from CBD, and then you have new regulations that come in that affect how cannabinoids can exist on the marketplace, or, and then that's going to affect all of these other companies all over a risk that could be mitigated, just with proper education and oversight and recognition of something's own risks and limitations. I feel like the cannabis industry, and particularly with CBD right now, it's an interesting place because people really don't like talking about risk at all. Like, they don't want to acknowledge that cannabis has any negative, potential. And it's something that as an industry, we've really got to get over, because it's going to keep causing problems, and it makes the industry look bad if you can't acknowledge, the relatively few, risks that actually exist with cannabis compared to a lot of other natural products.
Right. Like safe is a spectrum, and poison is in the dose.
Exactly. Yeah.
And so we can't pretend that cannabis is the exception to, like the most common fundamental rule of what we put in our bodies.
Yeah.
Like it matters how much and it matters what.
Yep.
And I don't like, obviously I don't like to see anyone die from, anything preventable, let alone from cannabis, but I don't like to, to open ourselves up to, demonizing, the word cannabis represents 400 different potential active ingredients. And we can't say, oh, well, now CBD is bad, or, oh, well, now just CBD tinctures is bad, or just CBD over 200 milligrams a day. We can't generalize it, but we can specialize. We can say these drug interaction issues were known. And, all you have to do is, talk to your doctor or at least research, the drugs that you're using. If more people, like you said, just knew the grapefruit, like it's such a simple thing to communicate that says, if your drug says that you cannot consume grapefruit and citrus, you should avoid cannabinoids as well. Like proliferating that is something that would be extremely important information for all of the prescribing doctors who write medical recommendations, because I bet there's a large portion of them that aren't, keeping up on that data. Not to mention every single bud tender, because they're not medical professionals, but every single one of them is making recommendations every single day about dose. And we don't have any good data to support what is safe and what isn't. So I hope that this brings the right attention to those issues, because it's easier to abuse a product that's more readily accessible, right? But we really need to address what that abuse looks like before it just all gets taken away.
Yeah, it relates to another idea that came up in one of my really early interviews about, like, what does responsible cannabis use look like, and trying to have that discussion, and being able to have like an elevator pitch kind of response to that. Like, what does that look like, especially as legalization progresses and people are able to have more open, honest conversations with adolescents and young kids and stuff about cannabis. Like, how do we talk about the risks in a mature, honest way? How do we talk about responsible use in a mature, honest way? And and going back to this theme that keeps coming up of being able to just make the industry better, increase support public health and safety, and just overall as a society just be better, and handle these things in a more mindful way.
It's something I think about constantly, because I have a 2-year-old now. So
I have a 1-year-old.
It's something I think about a lot, because my parents never consumed any cannabis in their lives to this day. And so it was easy for them to say, abstain. Like even if they were wrong about saying it was unsafe, like they were in a position to just, not use. Whereas, I'm going to be in a completely different position when I talk to my son about it, because I'm going to have to justify my use, at a minimum, let alone, be able to justify my recommendations on his. And I have no idea what that's going to be. Because I don't even know, like, by the time he gets exposed to cannabis, we might not even be smoking flower anymore.
Right.
You know what I mean? Like we might just like download it into our like AirPod, like I have no idea what cannabis consumption is going to look like 10 years from now or 20 years from now, because I never would have predicted my life today, in 2009 when I, walked into a dispensary for the first time, like I was still impressed by, giant volcano vaporizer bags back then, like the world is different. And so, how I communicate that is something I think about a lot, because I educate adults. And that comes with the social contract of, I'm not responsible for you. Right. I'm going to tell you what I know. and you're going to interpret it and apply it to your own life, but I'm not responsible for your choices. But I am responsible for my son's choices, you know, and I am responsible for, you know, the way he makes those decisions. And so I hope that education is something that, you know, we can follow, you know, really hard, you know, like it was great to get rid of a lot of the dare, you know, scare tactic type of, you know, fear mongering, but we can't just leave it at that.
Yeah.
You know, we have to replace it with something valuable. If it turns out that, you know, all of these jewels are more dangerous than cigarettes, you know, then like telling kids not to smoke cigarettes and not being prepared to respond to the vaporizer situation, you know, was an epic failure. Yeah. And so, you know, we're in that same position with cannabis. You know, like edibles are crazy today. And I do not think kids should be going around eating dispensary edibles. Yeah. You know, they'll, you know, I think that they don't have a good understanding of what that dose looks like. Yeah. Whereas I at least did have, you know, the social knowledge of knowing the difference between drinking a whole bottle of beer versus a whole bottle of tequila, you know, at 14 or at 16 or whatever. But with cannabis, we don't communicate that very well. And we might put the milligrams on the package, but that doesn't mean anything to anyone.
If you have no context.
Ibuprofen is 200 milligrams per dose. So yeah, 200 milligrams of THC, right? Why not? Like we don't have any perspective that we provide to consumers, let alone the unintended potential consumers. So I really hope that the cannabis industry will uphold you know, its original vows, you know, to take care of medical patients through that education. The more we learn about cannabis, the better those recommendations can be. And, you know, we can explain these risks and hopefully prevent them, you know, in the future.
Does Colorado have potency limits on edibles?
So there's limits per package. It's 100 milligrams per package, and the recommended dose is 10 milligrams per piece. So whatever your edible product is, it has to be easily identified as what 10 milligrams would represent. So that either means like 10 pieces in a
segmented
package,
or whatever.
You have to be able to break it up, or notches on the drink that tell you how far to drink it down. You know, something that makes it easy, I guess, to figure out how much you've consumed at once.
Yeah. And that's interesting too, because even 10 milligrams to like someone that is new to cannabis edibles or cannabis in general. I know people that have consumed 5 milligrams and started to have overwhelming experiences. So that context that you were talking about is so important. And edibles in general, it's so hard to get, I don't know, predictable effects from them, depending on what you're eating and how they're made, what the dose is, and like
your metabolism,
exactly what you've eaten during that day and how that's going to affect it. And you know, it's something like a drink. I had some friends that were working in testing labs that were working on trying to understand issues specifically related to drinks. But one of the things I saw recently is that the liner inside of aluminum cans and things actually are lipophilic, and cannabinoids will gather into them over time. So the potency of the drink will change over time and will lower, which is fascinating. And then I know there's another issue with drinks that, you know, depending on the pH, they're seeing some interesting conversions in some of the like THC infused sodas. So it just adds another layer of complexity to the edible market. That I don't know. I really like the idea of these formulated extracts, standardized. It also makes me wonder if, you know, having a product like that you could standardize and that you could define chemically, that you could say this has, you know, this much of sort of the terpene class, and of that class there are these variations, and you know, this % concentration of the cannabinoid class and these variations, that, you know, then researchers might be able to take something like that in the future and actually be able to start to do some more interesting research with products that are more akin to what people are actually using in the, you know, in the actual market, and understand how people react. And because that's, several clinicians that I've spoken with have mentioned that like that's one of the big drawbacks of them wanting to push forward on some interesting clinical trial work is there's like, where's the standardized product?
Yeah. How do you get it? Absolutely. Yeah. And that's one thing that's always been really difficult for me, is that like, I want to make good products. I want to sell safe medicine. You know, I want to feel good about everything that I'm making, but we're just relying on the plant to give us those chemicals.
Yeah.
We're just relying on
it's a carrier.
Cannabis being great and that being good enough. And that isn't good enough for literally any other type of extraction. That isn't good enough for any other type of product. Like you always have to, consider all of those factors. And so, when we're trying to sell something that has dozens of active ingredients in a final product, at concentrated doses, compared to what they were originally delivered in, if you were to just consume raw flower, I feel like there's a lot of responsibility there that I don't even have the data to back up. Like I can't say that more pinene is better or worse or safe. I can say I like it, but that doesn't help anyone else. And that doesn't, that doesn't give me, a standard to work from necessarily, which is what we need if we want to, improve that data. And so, one thing I'm really excited about is being able to now track through different harvests and even just age of material, how that profile changes. Because if I'm extracting it the same way and I'm starting to see variations from the same producer, now we can actually take that data back even further and say, what did you do on these plants that you didn't do on those plants? Like now we can, I can do a better job extracting in the lab. That's great. But I could influence the decisions that help them be a better grower. Like I'm expected to, test my product at every step in my process to make sure it's getting pure. But do the growers go and test their bud every single week to see what it's growing, to see if that terpene profile is developing, to see if it's all limonene or if it's going to get better? Like we don't hold them to the same analytical standards, but we could.
Yeah. Absolutely.
And that would make it easier for me. So that'd be great. But it would make it better for the consumer because they could be more consistent if they knew the impact that, their behaviors had chemically on what I'm able to extract later.
Yeah. I mean, ultimately it creates like a win-win-win situation because the farmer would just get better. It would help you with your process to be able to make standardized materials more easily, more efficiently. And the consumer would be, sort of have this itch scratch that's been there for so long of just like, I just want to be able to get the same thing, every time I go to the dispensary.
Most people don't like change. Yeah. Most people want it to be the same. And that's the only thing I can't give you.
Yeah.
I can't give you, unless you want just THCA or just CBD, I can't make it the same.
Yeah.
So that feedback is invaluable, both from the consumer and to the garden.
Yeah. Yeah, totally. Well, to start to wrap things up here, there's like two sort of broad questions that I'll ask you. One is, what are some of the most prevalent misconceptions about either cannabis or cannabis extraction or extracts themselves that you encounter most frequently?
I think you brought up the solvent scare already, which is probably one of the most common ones, is that people look at solvent extracted concentrates as being dangerous. And I would agree that most of them are dangerous to do. The operation definitely requires skill, equipment, and safety protocols to keep everyone safe. But generally they're safe to consume. However, I think that the exact opposite is also true. One of the greatest misconceptions is that cannabis is inherently safe. And I would argue that cannabis is only as safe as the people who are growing and manufacturing it. Cannabis is not a wild product anymore. It's not Mother Nature. And it doesn't love us. It is a plant that's being grown mostly for profit by people, and people you got to choose who you trust. And so, cannabis is only as safe as the people who've produced it. And the absolute same is true with concentrates. They are only as safe as the manufacturer who processed them and packaged them and delivered them to you. And from there, they're only as safe as the person who's storing and consuming them.
Yep.
And so, the misconception that cannabis is ultimately safe is, I think, potentially a dangerous one. We really want to pay attention to what we're doing with this plant, because we keep doing new and exciting stuff, and we keep discovering new things. And it's not uncommon in science in general for us to be wrong.
Right.
And there are parts of cannabis that can be very dangerous, and there are parts of cannabis concentrates that can be very dangerous. And I think that those, it's not something that we should be afraid of, but it's something that we should absolutely be educated about.
Yeah.
And no one's going to look for that education if we all operate under the impression that it is blanket safe.
Yeah.
Fire is safe if you don't touch it.
Right.
Like
exactly.
we gotta, we have to, consider those, benefits and risks at all times.
Yeah. Well, it's definitely been refreshing talking to you and hearing the balanced way, that you approach all of this, being willing to acknowledge the downsides and risks, as well as the awesome potential and upsides, and just being very real about, acknowledging all of that. And I really enjoy hearing your perspective of, that ultimately it's better for all of us if we can talk through these things, talk through our conflicts, try to work forward, and ultimately, all be the better for it. So it's, thanks for being willing to come on and share that perspective, and all the great information that you've shared in the past. We've gone for almost an hour and a half. So I really appreciate it.
Of course. No, thank you for having me. I always enjoy, talking about cannabis and concentrates, and especially to in a way that'll educate some other people, and hopefully
I think this will be
start some conversations they might not have otherwise had.
Oh yeah, that's exactly my hope with the podcast in general, is like, hopefully, the things that the conversations we have, hopefully it stirs other conversations, and just helps drive things in a good direction. I want to make sure to give you, the last couple minutes here. Pretty much, you have the platform. Let people know how to find information about you or your classes or anything else that you want to share or promote. Feel free.
Yeah, absolutely. I teach public classes twice a month at my lab and at other labs across the country and soon to be world. And so depending on where you're at, there's a lot of different opportunities to get in touch with me. But you can always find out what's happening through my Instagram. You can also keep up on my brand, Sano Gardens, the products that we're making. We, of course, specialize in live resin and have vape cartridges and high purity extracts that are all available in Colorado, and that's sanogardens.com or Sano Gardens on Instagram. We also have a full service consulting team behind Sano Gardens, and that's through Nicola Consulting. And so we can help you with designing your lab, improving efficiencies with your equipment, as well as partnering on product development. Our goal is to see more high quality products on the market, whether that means that we help you produce them on your own or we work together to produce them as a team. We believe in elevating the space. So whatever we can do to help other people become better. And you also mentioned, Future 4200, the forum, and the Good Life Gang. I cannot recommend it enough. The Good Life Gang is a great place to find a new mentor. Mm-hmm. And that's a membership program that gives you access, obviously, to discounts as well as to all of our meetups across the country. If you're not involved in the cannabis industry and want to get involved, it's a great way to meet the right people first and find the mentors and the people who can help you land in the good, safe places, and the find people who share the values that you do. So goodlifegang.tech, check it out.
Awesome. Yeah, well, that's great. Well, yeah, once again, thanks so much for giving me so much of your time and energy. I really, really appreciate it, and I've enjoyed our conversation.
Yeah, thank you so much.
Yeah, totally. So if you want to learn more about Curious About Cannabis, you can check us out at cacpodcast.com. We're also on social media, primarily on Facebook, Instagram, and Twitter, as well as YouTube. So check us out there. Thanks so much for tuning in and listening. Take it easy. Bye-bye. Hey, everybody, this is Jason Wilson with the Curious About Cannabis podcast. Thanks for joining us again. Today I am joined with Dr. Kevin Spellman, one of my good friends, mentors, colleagues. Very excited to have you here today, Kevin.
It's a pleasure to be here, Jason. Thanks for having me.
Yeah, totally. Just to start us off a little bit, just to help our listeners understand who you are and the type of work you do. Usually I tell people you're primarily a phytochemist. You understand a lot about plant chemistry, and as well as how compounds and plants interact with the body. Would you say that's a fair assessment?
I'd say that's a fair assessment. People tend to stick me in the chemist box a lot. I think of myself as a biologist because I'm fascinated by the way that plants affect our physiology, and I'm fascinated by that pharmacology. So by training, I'm a molecular biologist. But yeah, I definitely have spent a lot of time looking at molecules and looking at them in the lab and playing with them in the lab and looking at analytical chemistry results. And
yeah, same here. That's very similar to me. I get pinned in the chemistry box these days because of all the analytical chemistry work I do. But at my heart, I'm a biologist and really more so an ecologist than anything else. But I think ecology translates really well to pharmacology in very interesting ways.
It's so true. And until there is this thin veil between them right now that keeps our lobbying efforts on more ecological and environmental standards from really being heard. Once the medical community joins the ecological community and the environmental community, we're going to have a much stronger voice. And that needs to happen soon, because as we poison the planet, we're, of course, poisoning ourselves.
Right. And not to mention, just on a totally sort of anthropocentric perspective, losing potential novel compounds we haven't discovered yet. I think there's a lot of times a false perception that we already know a lot about nature and these compounds that are in plants and fungi and even in people, and that there's not a lot left to discover. And I try to explain to people that couldn't be further from the truth.
I completely agree with you. We are, I think as scientists, sometimes we're quite arrogant in what we think we know and forget about the dark knowledge that we can't see yet and that we don't know. A really good example that you brought up, losing compounds because we're losing plant species. We lose about a plant species a day.
Yeah.
And we've only researched about 6% of all the plant species on the planet. So what did we lose today, right? Was it a cure for Hodgkin's lymphoma or, HIV or, So we've got a lot of work to do. And I want to say the number of estimated plant species is something around 500,000. That's a fuzzy number for me, but 6% of those is all we've done.
Yeah. There's a long, long way to go.
A long
for
ways
sure.
to go.
Yeah, totally. To jump into the cannabis side of things, do you mind describing, I know you've done some research in the past around the endocannabinoid system, and a lot of work, which I want to spend a lot of time talking to you about today, about plant synergies and compounds in other plants other than cannabis that interact with the endocannabinoid system. I know you've done a lot of work with echinacea.
Yep.
And so do you mind just talking a little bit about some of that research that you've done and kind of see where that conversation goes?
Sure. Well, let's start with the endocannabinoid system. I did have an opportunity to look at some signaling going on there, CB1 and CB2. That's not to say that the endocannabinoid system is only CB1 and CB2. That's, I think, a misunderstanding that is frequently, what's the right word here? That's a misunderstanding that is frequently used to talk about an endocannabinoid system. But the endocannabinoid system is really fascinating. I look at it as a lipid signaling network that if we were to diagrammatically look at it, I would say that it's at the bottom of a pyramid, and everything else, neurological system, cardiovascular system, immunological system, all those are stacked on top of it. And therefore, that foundation endocannabinoid system affects everything that's sitting on top of it. So it has this huge role in signaling throughout the body. And I think that's really, really key. And so it's so much more than just the enzymes that are synthesizing and breaking down cannabinoids, and the receptors that are directly considered cannabinoid receptors, such as CB1, CB2. There's, now there's a putative CB3. It looks like there's probably a CB4 and a CB5 at this point, but it's too early to actually call them that. But there are a lot of compounds that, there are a lot of receptors that actually bind our endocannabinoids, as well as the exogenous cannabinoids in our body.
Yeah, and are you referring to like a lot of those G protein coupled receptors?
Yeah, GPCRs, exactly.
Yeah.
And then you've got the ion channels that are coupled to receptors. So the whole TRP receptor system is really fascinating, and makes a huge difference in terms of pain and sensation, as well as just gut health and neurological health. And so it's a very pleiotropic system that affects so many different subsystems or tissues throughout the body.
Yeah, it's funny, the more research that goes into the endocannabinoid system and looking at these G protein coupled receptors that endocannabinoids interact with and everything, it's just the definition's getting wider and wider and wider. And while a lot of cannabis education is still on that, okay, the endocannabinoid system is CB1, CB2, these enzymes, these ligands, which for anyone listening that doesn't understand what the word ligand means, that's just any compound that interacts with another receptor. We call those ligands, kind of simplified definition of that. But it is important for people to understand the endocannabinoid system is very broad, and the TRPV1, the TRPV1, and all the other TRPV ion channels, those are starting to get investigated. TRPV1 is now pretty much considered an intimate part of the endocannabinoid system in a lot of newer papers that are coming out, but it is much wider. It's tricky because when you're talking to a lay audience that isn't familiar with a lot of medical science and all of that, you kind of have to keep it simple. And how would you describe molecular signaling to someone that is sort of new to trying to understand what the endocannabinoid system is, what a receptor is, what a ligand is, all these sort of things? How would you describe that system? Because normally it gets described as like a lock and key model, which we now know is not appropriate.
Yeah, thanks for saying that. I'm so tired of hearing about the lock and key model, and we're hearing it from a lot of marketing now in the cannabis industry. But the lock and key model, that old model was, there was, the one key fits one lock, and that could be, nothing could be further from the truth in terms of that. We're finding that receptors tend to be promiscuous, if you will. Especially, I spent a lot of time looking at these receptors, orphan-nuclear receptors. Inside the nucleus, the PPAR receptors, PPAR gamma is so incredibly promiscuous that two different molecules, two different ligands can fit in there at the same time.
Oh, wow, I didn't know that.
Yeah,
wow.
It's pretty fascinating. So, and that's, by the way, is also considered part of the endocannabinoid system now, is the PPARs, these orphan nuclear receptors. And they're called orphans because the original ligands were the endogenous ligands were never really established. There's definitely some deep insights into what they are or could be. But when you hear that orphan, it means that we don't really know what the endogenous compound that relates to that receptor is. So, yeah, I think we have, I agree with you completely. It just keeps getting broader and broader, and it's gone well beyond CB1, CB2, FA, MAGL, those enzymes. And it's an exciting time to think about it. In terms of signaling, coming back there, how does signaling happen? Carefully, number one. It is very orchestrated in a very profound way that I don't think most people realize. And so that first piece there, that it's orchestrated, I want to refer to this idea of it's a network. And this really comes from a lot of ecology understanding, where we look at ecosystems and we understand that they're very complicated networks. You can't just introduce rabbits into, right, or whatever it was.
Or pull something out.
Yeah, you can't pull something out or put a species in. In the same sort of way, you can't block a enzyme or a receptor system without having profound implications on the rest of the network. So genes, receptors, enzymes, all these targets, as we call them in pharmacology, respond to messenger molecules. Is a term that I really love because it makes it really clear what these molecules do. Each molecule carries a message, and so these molecules usually bind more than one site, and that's something that's misunderstood as well. And and by the way, then their metabolites, they're broken down by enzymes, and then those metabolites of those molecules then can bind other sites. So we have this incredible network that I like to think of it as network pharmacology. There's this model now that I think is really starting to embrace medicinal plants in a way that couldn't be embraced when we were looking at one target, one ligand model. So signaling is incredibly complex. It's orchestrated. It's about harmony. It's about all these signals happening simultaneously and sequentially, and therefore having a response that makes sense to the body. And without that complex network signaling that includes upregulating and downregulating genes, includes upregulating and downregulating receptors, includes upregulating and downregulating enzyme systems. Without all of that, we would be puddles of slime on the floor.
Right. Yeah. I mean, literally, it's you, if that intimate, I like the orchestration model, this sort of orchestra or symphony of activity that's going on. If things don't come together, you can't even think straight or perceive properly. There's a number of different things besides just like bodily functions not working. Well, an example I've talked about before, but I think illustrates the sensitivity of messing with these systems is, did you hear about the study that was done in France a few years ago, where they tried to do it was an FAH blocker.
Inhibitor,
It ended up sending several people to the hospital. One guy brain dead, went majorly wrong. And there's been some follow-up studies to try to figure out why it went wrong, because the rodent models made it seem like it should have been no problem, which I don't know if that's something you want to comment on at all. Because there's a lot of cannabis research currently. A lot is either cell culture studies that are looking at how cannabinoids are interacting with certain tissues or cell types, or different proteins like receptors, or they're rodent models, and results are getting scaled. And particularly this issue has come up just in the past couple of days. Forbes released an article about CBD, and the headline said something like, researchers find CBD causes liver damage. Very broad title. And I was like, oh, that's interesting. And I wondered, are they talking about the cytochrome P450 stuff? What are they referencing? And so I went and looked up the study, and, it was a rodent study, and they ended up taking their lowest dose of CBD that they gave the rodents was allometrically scaled based on the highest doses that went through the Epidiolex trials. So looking at 15 to 20 milligrams per kilogram body weight, scaling that down to rodents, that was their starting dose. And then they did doses that were 3 times and 10 times that. And what they found is in the doses that were 3 times and 10 times, they saw some pretty significant liver damage that started to occur in the rodents, particularly in the 10X model. They were feeding these rodents 2 grams of CBD per kilogram of body weight.
Holy cow.
Yeah, over 2 grams.
And then a journalist blows it up into, oh my God, my hair's on fire.
Right, yeah. And it's something that myself and several other people on social media that try to do science education, and particularly around cannabis, tried to actually post the article and say, no, actually read the research study, because the authors even talk about, something that you and I have talked about before, super physiological doses.
Yeah.
And that, while these effects occur, in most real life scenarios, you're unlikely to encounter them because most people aren't consuming that much. But is that something you can comment on at all? Just some of the nuances that people should be aware of when they're looking at studies that are using rodents versus humans, how that scaling occurs? And
yeah, so let me tell you a story that shocked me. I was running a R&D group that was a company. It was $1 billion company, so they had money and they had resources. In fact, my research budget was $10 million. And there had been a nutritionist there for many years that apparently was doing the dosing on any of the this company did multivitamins, so he would figure out the dosing. And so when I asked him, he mentioned a study to me, and I said, what is the HED, human equivalent dose? And he gave me a blank look. Now, he had been there for over 10 years determining this company's dosing. And I said, human equivalent dose? Do you know what that is? He said, no, I've never heard of that. So he had been determining all of his dosing based on animal models. And of course, the human equivalent dosing models are based on either surface area or metabolic rate. And hopefully they're based on both, and that'll give you a more accurate read of what the right dose might be for a human in an animal model. But he had never heard of this, and so I sent him all these papers on human equivalent dose that I had on my hard drive, and he never mentioned it again because I think he was incredibly embarrassed that he'd been determining doses based on animal studies that were not relevant to humans. So there's the first piece, is like people tend to, if it was 2 grams a kilogram, they tend to just multiply that times 65 or 7. Actually, the average human now is over 65. Yeah. At least in the U.S., kilograms. So they tend to do that, and that's wrong. Metabolically, I think a mouse its metabolic rate is 7 times faster than a human. I think rats are 11 times, and then you've got surface area to deal with as well. So those doses tend to change pretty significantly. But coming back to the animal model, yeah, I think we have to be really careful when we're looking at doses. Basically, anything over about 50 milligrams per kilogram is probably not realistic for human use. It's on that cusp, and that's even super high. So you get up to something like 2 grams, I mean, you're toxic. And that would happen with, look, you can drink too much water and drown, right? Right. So anything can be toxic, and that kind of brings us to this idea of hormesis and understanding what hormesis is. And this is that idea of a biphasic response, that a little does one thing, but a lot does another thing. And generally with this hormetic response, a little bit of a substance that potentially could be dangerous actually upregulates things like NRF2 or NRF2, it's called, which then launches this whole antioxidant system, protective buffer system. But then if you have a lot of it, it's toxic. And this is seen throughout all kingdoms, whether it's plants, whether it's animals, whether it's reptiles, whatever it is. And it's there were in the 40s, actually in the 40s is when it started losing favor, but throughout the early 20th century, there were journals based on this hormesis model. It wasn't always called hormesis, but there were a number of names for it. So we need to understand that, so the American way that if one is good is not two better, anything worth doing is worth overdoing, right? Right. That's not true, and we need to be really careful about that in medicine, in food, in supplements, that a little bit can go a long ways, and when you take more, it doesn't mean you're getting more of the same effect. It may mean that you've gone to the other side of the curve, and you're actually getting potentially toxic effects.
Yeah, and this is something that in medicinal plant science has been well known, that there are a lot of different plants that may be helpful, but if you're consuming a lot of them every day, it's known that you start to get liver problems, all sorts of different things. Even I was talking to someone recently just about reishi mushroom tinctures, and just explaining like, hey, just understand that this isn't something you just consume a lot of all the time and you're going to be super healthy. Like, you've got to understand the dosaging, understand what you're working with, periodically take some breaks, let your body kind of reset, and then pick it up again, and give your liver a break from having to process, some of these kind of foreign compounds.
I completely agree. The idea of drug holidays should be applied to dietary supplements as well.
Yeah.
The one thing I would say, though, in terms of dosing and dietary supplements, especially on tinctures, look, you got a 1 ounce bottle,
right?
And it's often now over $15. I've seen them at $25. Oh, but in the cannabis industry, they're 4 times that.
Right. Yeah. $150 for an ounce.
Yeah. Right. But the doses they have on those bottles usually are subclinical.
Yeah.
And so, for example, with echinacea, 30 drops is not going to do it, right? We're talking teaspoons, if on the first signs of a cold. But I completely agree with you. The idea we need to think of dietary supplements as food items, which is really in many ways what they are, and therefore we wouldn't eat nothing but blueberries for 5 days, right, straight. We would have judicious amounts in our diet, and that's how we need to think of dietary supplements. So, one of the things that I think isoflavones is a really good example where the isoflavones from beans, and usually people just think soy is the only place, but isoflavones are in basically almost all legumes.
Yeah.
That these compounds actually bind the beta estrogen receptor, and it's at a much lower affinity, but they still have a slight estrogen nudge towards the body. So, in our diets, maybe 50 milligrams a day was what you could get if you were eating a lot, if you were a Japanese or an Asian person eating a lot of soy-based foods. But I see supplements with 150 times that, 150 milligrams, rather than just 50 milligrams. So I worry about that sort of thing.
Yeah. Yeah. And it's weird, like now culturally microdosing is sort of in, but in the 90s and early 2000s, megadosing was really in, and especially around like vitamin C.
Yeah.
That was huge, taking mega doses of supplements to try to get certain responses that now a lot of research around that megadosing is not upholding some of the initial thoughts of what people thought that might do. Well, talking about these other plants, let's talk about plant synergy and herbal synergy and the endocannabinoid system and how that all comes together. I think probably a lot of people listening to this have probably heard of the entourage effect. That's become really popular in the cannabis space. And a lot of people act like that's a new thing.
Thanks for saying that.
But I think, as you can attest to, it is definitely not a new thing, and something we've been trying to understand for a long time. And cannabis is kind of bringing that to the forefront. So can you just kind of describe how you conceptualize these herbal synergies, entourage effects, whatever you want to call them, and then I'd like to get into talking about things like the alkylamides in echinacea and other compounds, some of the terpenes that are found in basically every plant,
right,
that are interacting with the endocannabinoid system, and I don't know how people should think about this, because it's getting so much hype.
Well, I think you used the word symphony. That's really what I think the best depiction of this idea of the entourage effect, or what we have called for probably longer synergy in medicinal plants, is a symphonic effect. So think about a Mozart symphony that's so incredibly beautiful, and you have a lead violin, and then you have all these other instruments, right? And all those other instruments basically support that lead violin. Now, what's happened in pharmacology is, under the illusion, and I will call it an illusion, that we can control the dose, we've isolated the lead violin and just used that in our pharmacology. And the rationalization for this is actually, if you really go back and look at the science, the rationalization for doing this is actually not very strong scientifically. In the 30s, in the early 20th century, 20s, you had the eclectic physicians doing studies on whole plant extracts, and then in some cases they actually used an isolated constituent. And this is the Lloyd brothers, the eclectic physicians were supported by some brilliant pharmacists called the Lloyd brothers. And the Lloyd brothers were really good at extracting. In fact, some of their instrumentation, their chemical instruments are still used today. They invented them to do extractions, and we still use them today in chemistry labs. So they had this opportunity to look at whole plant extracts versus isolated constituents. And they found in many cases, not always, but they found in many cases that the whole plant extracts were had a better outcome. Now, and I think that's really what we have to look at, because in an isolated model, for example, a cell culture in an animal model, you actually might get a better outcome with an isolated constituent.
Right.
Because you're pounding, essentially it's pharmacological dosing versus physiological dosing, right? So in physiological dosing being that idea of using just enough to get the effect you're looking for.
Minimum effective dose.
Exactly. And what we end up seeing with this in healthcare is that everybody's too busy to do that. The patient's too busy to pay attention, because they're going to have to pay attention to where they get the effect. The doctor's too busy to actually put an inquiry into where was that dose that worked for you. And everybody wants instant relief. So it's fallen by the wayside. But were we to pursue that further, I think we'd have a much more effective model. The other thing that happens here when we look at isolates versus whole plant extracts is the adverse event profile goes up. And this is a huge issue. And for example, in my own body, if I take ginkgo 24/6, the standardized stuff, I actually end up with a huge diuretic effect. I'm running to the bathroom every 20 minutes. If I just use ginkgo leaf that's thrown in a capsule, I don't have that effect. I noticed the blood flow effect in terms of cognitive function with both of them, but I'd rather use the whole leaf ground up. So we have that lead violin that's working. And what we found in reductionist chemistry now is that, there's more than one lead violin. Turns out that the oboe takes a lead in this section as well, right? And it turns out that the cello also ends up having a lead in the. So we have found multiple active constituents. In the case of echinacea, it's been hypothesized that at least four now compounds are considered the actives. When they came across the CB2 binding of alkylamides, what happened is everybody thought, we've finally done it. We have found the active in echinacea. Well, not so fast, because we've got the caffeic acid derivatives, which, by the way, are directly antiviral, but incredible antioxidants and potentially block histamine release. By the way, if you're looking at anaphylactic reactions and you're stuck in a pinch. We've also got the polysaccharides, the rabinogalactans in echinacea that are also super active and directly stimulate immune function. So that becomes very interesting, because here in one plant you've got something that's a bit of a immuno dampening effect, the alkylamides, and you've also got something that's a direct immune stimulator, that's the rabinogalactans, and then you've got something that tends to modulate immune function, that's a caffeic acid derivatives. And by the way, it's antiviral. So we can't simplify plants to one constituent, and that's the mistake that's being made over and over and over. It started in the 70s with ginseng. People were looking at ginseng and saying, what's the active constituent? And they came upon the ginsenosides, or the saponins of the plant. And so what happened from this ridiculous research in the 70s is there was a time when people would look at plants, and if they didn't have alkaloids or they didn't have saponins, they were thrown out. No, they're not active. It's all fantasy. No, these tribes using this are not getting any effect. We know best.
Right. We've studied it.
Exactly. So I could talk about this all day. I've written a couple of chapters on it in books, and published a couple of papers on synergy. But the idea that there's only one active compound is really naive when it comes to medicinal plants. And we need to look at it as there's this symphonic effect when every time we swallow a bolus of either a food plant or a medicinal plant, we're getting literally thousands of compounds, and I literally do mean thousands. The idea that there's only 400 compounds in cannabis, I think, is super naive as well, because if you look at corn and you look at wheat, respectively, they have found 3,000 and 5,000 different compounds in there, and these are probably the most studied plants, right? Analytically. And so I can tell you with certain clarity that there's at least 1,000 compounds in probably every single plant. As analytical chemistry gets more and more
widens.
sensitive, it's just going to widen further. So to think that there's only one active compound is delusional. And I think we need to step back and look at this network pharmacology idea where we have multiple actives basically nudging a network, not blocking any one site 100%, or not binding with great inhibition or great agonist activity.
Partial agonism, partial antagonism.
Exactly. Partial inhibition. And that combined effect of multiple nodes in a network being nudged has potentially a better outcome and a safer adverse event profile as well.
Well, there was one study just looking at CBD. I believe it was with CBD, looking at isolated CBD, and then I think they call it a whole plant extract, which the way that some of these terms are used in the industry drives me crazy. Because, in herbalism, the term whole plant extract does have a definition. You're using aerial parts, you're using roots, all these different pieces. If it's an extract just from aerial parts, you say that, but anyway, beyond that, there was a study that showed that with isolated CBD, there was a very limited therapeutic response profile, and it's pretty much a pretty standard bell curve. And that you, at certain dosages, you hit a peak, and then you start getting losses in therapeutic efficacy. But when they used these more, for lack of a better term, broader spectrum extracts, or whatever extract they used, that curve turned into a linear line, that they started to see better therapeutic outcomes with fewer side effects and better outcomes at lower dosages. But the main thing is that ceiling was seemingly eliminated when they introduced an extract versus an isolated compound. And I can't remember exactly how they measured all of that. I need to go back and pull it, but that's just...
It was a TNF alpha study.
Was it? Okay.
Yeah.
And so that just in a very narrow way demonstrates the huge difference between isolated compounds and extracts that have a broader spectrum of phytochemistry. And it's tricky. I want to know your thoughts on, so the traditional model of doing clinical trials and all of this is, standardized ingredients that are the same every time, very tight controls. What's the way forward given that medical science, and I do think this is becoming a more and more popular notion now, the idea of network pharmacology, understanding that these limitations of isolated compounds, the more and more people I talk to, people seem to be accepting that. But the pushback that I receive is, but how do you study it to get that included in medicine as our systems are set up right now? Because it's very hard to get funding to do clinical trials with herbs because there are much greater limitations into what product you're gonna come out with and market. And so what's the way forward to do research to identify the therapeutic potentials and the limitations of medicinal plants, given this model that we're stuck with?
Yeah, it's a really good question, and I'm going to give it a really simple answer, but the devil is in the details. And the answer is outcomes versus mechanisms. What we're looking for a lot of times in clinical trials is, of course, we're looking at outcomes, but we really want to understand mechanisms in a way that may not be realistic. And I was actually just at a traumatic brain injury conference, and I suggested the idea that there's dark pharmacology. They were talking about dark data that was negative, that never got published. I turned it on its head and said, let's talk about dark pharmacology. Dark pharmacology is pharmacology we can't see yet because we've been looking under the street light, right? Looking for the drunk that's looking for his keys, right? That's what we've been doing in science. We look for what we know, and not necessarily it's expensive to go off into the weeds. looking for something. So we start thinking we know everything. And I've seen this happen with a lot of medicinal plant studies where, oh, well, St. John's wort doesn't work for depression because it doesn't do this and it doesn't do that, the mechanisms that we know about. Well, actually, if you look at the outcomes, it actually does work. And so I think one of the answers is really when we can't necessarily standardize the amount of active compound A or active compound B because they're medicinal plants, we have to look at the outcomes. And I think that's really the key. And there's been a lot of, they're starting to happen. Let's not say that there's been a lot of them, but there's more and more of these studies on integrative medicine, complementary alternative medicine, whatever you want to call it these days. I just call it medicine myself. But there's been these studies basically looking at seeing alternative practitioners who do multiple things, right? It's not just one drug or one herb. They're suggesting this, and I want you to think about that, and I want you to read this book, and I want you to take these 3 herbal compounds, and one's a tea and one's a tincture and one's a capsule, and they're all doing different things. And then, that confounds the way that we've done research. And I think the key is outcomes and not necessarily mechanisms. The mechanisms are sexy, and believe me, as a molecular biologist, I'm seduced by molecular modes of activity, right? I want to understand, but it also gives us a false arrogance.
Yes.
That we think we know, and in fact, there's so much dark pharmacology right now that we don't know, and we only know what we know, and we do not know what we do not know.
Right. Yeah. There's a massive amount of humility that needs to be had by anyone working professionally in medical science and really science broadly. I mean, science has repeatedly been plagued by arrogance, a false sense of that understanding of like, oh, I know enough now that now I can run with this and extrapolate to, the ends of the universe. And
in the early 1900s, they wanted to close the patent office, right?
Right, yeah. It's like, no.
The steam engine has been discovered.
Right, we're good now. And no, I think that's a really good point. And to add to that, when I get into discussions about this with other people, I talk about assigning value to the safety profiles of a lot of these medicinal plants like cannabis, and recognizing that, we should feel more confident doing that type of research focused on outcomes given the safety profile of these plants and these compounds. Because if nothing else, we have a pretty good sense of how toxic a lot of these things are, at least when it comes to LD50s, lethal dosages, and that sort of thing.
And let me interrupt you there, right there, because I think you just brought up a really key point that I can hear the scientists, not so much, and the medical doctors probably very much saying, safety? What are you talking about? Let's talk about safety for a second here, because I'm going to try not to go off on a rant here.
Go for it if you want to.
But you end up with somewhere between 80,000 to 100,000 people die every year from properly prescribed pharmaceutical medications. And if we have 5 deaths in a year from dietary supplements, it's blown up all over the news.
Oh, yeah.
And if you look at the numbers, more people are using dietary supplements than are using pharmacological medications or pharmaceutical medications. Right. So we need to step back and go, this is not a safety issue. It's an economics issue.
Yeah.
We've got the pharma crying foul because we don't have to do the amount of research from substances that are naturally occurring, that most of them have been in our food, in our diet anyway. So, they're saying, well, they don't have to do this and that. Well, that's true. And maybe that is something to look at. But to say that they're not safe, I don't think so.
Yeah, no, I totally agree. I think that's a bullshit reason to limit research and encouraging people to
fear their dietary supplements.
Yeah. Well, and to touch on something you said earlier, talking about dietary supplements, thinking about them as foods. Foods are medicines, and it's important for people to recognize that and understand, like, the things you eat, they are chemicals. They're influencing your body. They're triggering all sorts of signaling in the body. Your foods are medicines as well. Pharmaceuticals aren't the only medicines, or standardized supplements and pharmaceuticals are not the only medicines. It's everything you consume is a medicine, and it's important for people to think critically about what they're consuming broadly.
Absolutely. There's a phrase that I'm going to attribute to Jeff Bland that I actually really like, and that is that you steep your genes daily in your dietary broth.
Oh, that's great.
Isn't it?
Yeah.
And if we looked at a fast food eater versus somebody who just, had their greens and some fruit and maybe some nice protein, whether it be vegetable or animal-based protein, and we looked at the gene profiles, what's been upregulated and what's been downregulated, and those two people, they would be completely different, and suddenly you would understand why that fast food eater is going to die 10 years earlier than the other guy is.
Right.
But we don't do that.
Right. Yeah. Well, it's fascinating to me that all of the education that goes on, commercials that come on TV of health campaigns and everything, it's such a simple message that for some reason isn't coming through, that like, hey, your food is also medicine, and, trying to encourage that holistic thinking about health and wellness. And even in the concept of dietary supplements, a lot of people, especially in the United States, fall into the same sort of thinking of like, oh, I'm sick, I need to take a bunch of supplements, treating it just like they would a pharmaceutical, and not thinking of like, well, what's my daily lifestyle? What's my daily regimen? And how do I incorporate things into my daily routines to support my body as it is? And a lot of people aren't even doing, going back to these, like taking breaks from things, like trying to figure out what your baseline is so that you even know what you're working from.
Right.
To know what you might need to incorporate.
Yeah, most people don't know that.
Yeah, no, it's very challenging. And I mean, I'm as guilty as anyone else, so I want to make that clear. Like, I'm not perfect. I eat plenty of fast food and all sorts of, but it's about developing that awareness and trying to make your decisions be more mindful, and just having your whole life be more mindful and trying to be in touch of what you're feeling, what you're experiencing, what's influencing you broadly, and that extends beyond just food, but everything, news and media, all sorts of things. What's influencing you? What's building your reality? Because it's all of that. All of that itself is a symphony, an orchestra.
Exactly.
That can be very discordant if you're not taking that time.
Yeah, you bring up two really good issues here: industry and lifestyle. The industry, the food industry, does not want to know about those studies and has actively tried to block the release of such studies in terms of showing that a healthier diet has a very profound influence on upregulation of positive genes and downregulation of negative genes, the negative effects, versus fast food. So we have a huge lobbying effort to prevent that sort of information getting out and actually doing those studies. Let's take the Harvard researchers that hid the sugar research and pushed the high fat research, right, to protect an industry. And you got lifestyle, and having lived in Europe and having lived in Latin America, I have to say, Americans, we're freaking crazy.
Yeah,
I mean, the way we work, and I'm at the top of the heap in terms of being a bad, lifestyle guy, and just the amount of work that I do in any given day, and then I don't stop. I usually work my weekends off as well. Life is really super busy, but that is not the way to be a healthy person. You've got to have integration time. You've got to have downtime. Our brains can't it's like, you're going to run your engine on high all day long. You wouldn't do that to your car.
Yeah.
Right. But we expect it from our bodies and from our minds. And so there's a lifestyle mismatch with what is healthy, or we think we're going to get rewarded for busting our asses. And in fact, what we're really doing is degrading our health.
Yeah, no, completely. And sabotaging the very thing that you want, because you generally want to be high performing, you want to perform well and all these things. And by stretching yourself too thin, you're actually making your performance worse and making it more likely that you're going to experience negative outcomes later and whatever.
Yeah.
So
back to cannabis.
Yeah. So I love it though, because what I like about cannabis is it's a doorway for a lot of people to get into these very critical topics. And that's why this podcast, I don't want to limit it to only talking about cannabis growing and extraction, all these things. While I do want to talk about those things, I want to get into the deeper concepts that cannabis is sort of a springboard for. But that being said, coming back really directly to cannabis, what do you think are some of the biggest misconceptions that people have, either about cannabis or the endocannabinoid system, that you tend to encounter on a regular basis?
There's a couple, and I would say that they're both actually what I like to call black market hangovers. One is the idea that if we're going to use THC medicinally, that the requirement is that we get high from it.
Really good point, yes.
And I'd have to say nothing is further from the truth. THC is pharmacologically active at doses that don't generate a high or euphoria. And there may be a couple instances in severe pain or cancer where you really are dealing with the central nervous system effects of THC. Right. But otherwise, there's many cases where that's just completely not necessary. And then the other one that just is driving me crazy, and it's definitely a black market hangover, is decarboxylation. This idea that we have to blow off CO2, that we have to...
Got to activate.
We got to activate, right. And, this has been shown. In fact, I've just compiled a list, working on a white paper right now, and I've just compiled a list of all the targets of acidic cannabinoids. And let me tell you, it's big, especially for CBDA. It's huge. I have a friend that was at UCSF for years, a family nurse practitioner who handled kids with intractable seizures. And what she did was made sure that the cannabis oil, the hemp oil that they were getting, was non-decarboxylated.
Yeah.
So she was using strictly CBDA. With these kids and getting great results.
Yeah.
And so, and also TRPV1, which by the way, CBDA binds at high affinity, is a target for seizures. So it turns out that CBD is still working, but CBDA works really well too. So we do not have to decarboxylate everything. And there's probably advantages to not decarboxylating in terms of bioavailability as well.
Yeah, and man, that first point you brought up has been on my mind a lot lately, that you should not equate therapeutic response with the high. Yeah, just the higher you are doesn't mean you're getting more therapeutic response. And I think that's something that just takes people a while to catch on to, especially if they're really new to cannabis, because that's the main entry point, is that they know that cannabis gets you high and it helps people, and so I'm going to experience a high if I want to use cannabis therapeutically. That's really, really important for people to understand. There are so many things happening in your body that you're unaware of, that you just can't perceive directly, and that's why it's really, really important to try to take that time to inventory yourself. I talked about it before. I'll talk about it probably every episode, but journaling when you're trying to treat yourself with any therapeutic agent, taking really careful notes about dosaging, when you're taking things, what you're noticing day to day, even just really basic things. Monitor your pain levels if you're trying to treat pain on 1 to 10, and monitor that,
Encouraging self-awareness.
Yeah, just trying to really critically evaluate your state of being, because a lot of these activities, other than the sort of euphoric effect of THC, most of these effects you're not going to feel, not right away. Some in pain relief, if it's immediate joint aches or whatever, you might notice that. But a lot of other stuff, it's something that builds over time as well with repeated dosaging, especially with cannabinoids or any lipophilic therapeutic agent. You're going to have that building up over time in your body. And so, I think I'm really happy to hear you talk about that. I think that's really important for people to recognize. And I'm sure there are some THC producers out there right now that are unhappy to hear
or
that.
Cursing at the radio or whatever their device is.
I've already warned people that this podcast, it's going to get me hate from every angle. And that's how I assume I'm doing something right. I'm prepared for that. So that's fine.
I just have to credit Ethan Russo. I like the way Ethan says it about THC and the high, the euphoria. He says that if you're using THC, cannabis, chemovar, THC, therapeutically, and you're high, you're having an adverse event.
Right. I mean, in any trials that are done with THC-rich cannabis, that is one of the adverse events that is monitored. And the only time I could see that being different is certain psychological conditions where, you know, the high may be part of the therapeutic response. But other than that, no. I mean, unless you're using it recreationally.
Yeah.
In which case, you know,
fine.
That's fine. Yeah, that's your prerogative to do. But don't confound the recreation aspect of cannabis with the therapeutic aspect, because they aren't always married together, although sometimes they are.
Yeah.
So what do you hope to see as cannabis research continues, and it's picking up at a really fast rate if you're monitoring the papers that are being published? I mean, basically since last year, there's just been a huge increase in momentum.
Half a dozen to a dozen every week.
Yeah, it's really wild. It's hard to keep up with.
Yeah, it's a full-time job.
It really is. It's something that, you know, I try to connect people with research papers as best I can and stay up on top of things as best I can. But, you know, I'm in a perpetual state of being behind, and I'm realizing that's just the way it's going to be because, you know, all this research is coming out. It's like I'm going to have to rely on really well-written review papers at this point to stay caught up. But what would you like to see from the future of cannabis research and endocannabinoid research?
A couple things. One thing that is super fascinating to me is looking at the new research on frontal striatal connectivity in the brain with CBD, with cannabidiol, which, so what I basically just said is synaptic patterning is strengthened or even potentially becomes more complex. And I think that's super exciting. I mean, we're talking now about not only protecting the brain, but we're also talking about making it more agile in a sense.
Right.
And so that's super fascinating to me. But the other piece, and I think more basic, Greg Gerdeman is the scientist who discovered the retrograde signaling of cannabinoids.
Yeah.
And Greg basically just says it beautifully. He says something to the effect that there is a mountain of research showing neuroprotective effects of cannabinoids. And yet we have a scientific community that until very recently has been based on the negative effects of cannabinoids. And so, sure, any medicine, anything that's going to be medicine, whether it's a dietary supplement or a pharmaceutical drug, has potentially it's a double-edged sword, has potentially a negative side.
Yeah.
But the neuroprotective effects of cannabinoids are striking. So I'd like to see more acknowledgement of that from the scientific community.
Yeah. Yeah, I agree. And broadly, man, I just want to see more healthcare professionals get educated on some of this stuff.
That's a really good point.
Because, man,
it's needed.
it's, you know, it's very hard to find a doctor or even a nurse that understands much of this in any sophisticated way. And a lot of those that do operate in the cannabis space still don't have that in-depth of a understanding. I've been pretty disappointed by you know different folks that I've talked to that are actively doing work with patients with cannabis that are still talking about indica and sativa as if it's a significant model to use for finding different effects that things will have, or even talking about strains and not recognizing that you can have different chemotypes among things that are labeled the same strain name. Cultivar names can be helpful in getting you pointed in the right direction towards certain things, but if you're just telling patients to use a certain strain of something to go to their dispensary and find some X strain to treat X condition, X, Y condition, like
you don't know what they're going to come back with.
No, and that's just not really an appropriate way forward. So I don't know. I hope things get caught up quickly because our healthcare system needs it. I think to really push this critical thinking about cannabis and, getting back to the springboard effect, I think it will help healthcare professionals broadly start to open their minds a little bit about medicinal plants just across the board. And I hope to see that humility piece get cultivated.
Yeah, you bring up the humility piece is huge, and I unfortunately don't see that changing, but I hope I'm wrong.
Yeah.
I lost what you just said before that. Cannabis, what was it?
I was saying, hoping they have a better understanding, and that springboards into broader acceptance of medicinal plants.
Thanks. Yeah, that was a piece I was going to comment on. Yeah, so in regard to medicinal plants, I think that we need to realize that cannabis is just one medicinal plant, and everybody's excited about that medicinal plant right now. But that's my hope as well, is that this idea that a medicinal plant that's a complex array of phytochemistry and phytonutrients has the potential to make a very meaningful therapeutic difference in the quality of people's lives. That, to me, is medicinal plants, and that's for 30 years. That's what I've been focusing on, is medicinal plants. And I've made enemies in scientific institutions merely because of what I'm into, and not because of my science, but because of the fact that I have an interest in medicinal plants. There is this bias in science against medicinal plants, as if it's not real. And we need to stop and realize that about half of all of our drugs have been based on medicinal plants, and about 25% of all of our pharmacopoeia is still directly isolated from medicinal plants. If you look at cancer drugs, something like 70% of them in the last 10 years have come from natural products in some sort of way. So when physicians and healthcare providers look at me as, with a disapproving look that I'm into medicinal plants, they don't understand their own pharmacopoeia that they're writing from daily.
I totally see that too. Well, and so you've been extremely generous with your time. I'm going to go ahead and start to wrap this up. One of the last things that I wanted to ask you is, leaving cannabis aside for a second, what's at least one thing that you're super excited about that has nothing to do with cannabis?
I'm super excited about lion's mane right now. I've had an opportunity to look at some of that research, and recently wrote a review paper on it. And the hericinones and the erinacines, these diterpene derivatives that aren't polysaccharides, that aren't beta-glucans. Everybody's excited about mushrooms, and everybody thinks it's all about the beta-glucans. It's not. They seem to have a pretty profound effect on things like Alzheimer's disease and mild cognitive impairment. And so I'm super interested in seeing that therapeutically harnessed to make a huge difference in the quality of aging people's lives. Yeah. Lion's mane mushroom. I've got some dried out lion's mane in the cupboard now. Something I'm very fascinated as well. Well, thank you so much, Kevin, for coming on and talking to me. We'll have to sit down again sometime and pick up discussion. I want to keep track of your work and what you have going on, and I'm sure we'll have plenty to talk about again soon. I'm sure. Jason, thank you. I love what you're doing here, and getting the word out. It's great. Thanks.
Thank you very much. All right. If you want to learn more about Curious About Cannabis, you can go to cacpodcast.com or check out the Curious About Cannabis book, available now on Amazon and other online retailers. Thanks, and have a great day. Take it easy. Hey everybody, this is Jason Wilson with Curious About Cannabis podcast. Thanks so much for tuning in once again. Today I am thrilled to be able to sit down with a friend of mine and very, very experienced clinician and expert in Chinese medicine, Dr. Jason Miller.
Hey Jason, how are you today?
Yeah, thanks so much for being willing to carve out some time to sit down with me and see what conversations we get into about herbal medicine and cannabis and everything in between.
Excited. Thanks for calling me and checking in and asking me to come join your podcast.
Yeah, totally. So just to briefly describe to people some of the work you do, and we can get into your kind of backstory later, but can you just very briefly describe some of the work you do and what your expertise is?
Yeah, sure. I think I'll just preface it with a little bit of background. When I graduated from medical school in traditional Chinese medicine, I went directly into work with an expert in botanical medicine in cancer. And so I just started going into this deep study of cancer and chronic disease, and then the application of botanical and nutritional medicine, including lifestyle and diet and everything else. But and I went into the whole process of evaluating patients and taking the kind of traditional medicine approach that comes from an older medical system, one that's existed for thousands of years, bringing that knowledge and wisdom through a wisdom tradition, and then digging deeply into the research of the modern science model and seeing, what were the best possible solutions for a person struggling with a cancer diagnosis. Mm-hmm. And so that's where, so for 15 years, I've basically been, continuing to practice Chinese medicine. I do acupuncture. I do, all kinds of body work in the clinic. I evaluate neurological conditions and help people with pain. But I do that in a small amount of my time. Most of my time is spent either researching, cancer, different therapies, what's going on with herbs and interactions, and then just working with patients who are basically, coming to me saying, hey, here's my situation. I don't know what to do. I've got this opinion, and it's, and they're, they're like, I need help. And so then that's where I come in and say, okay, well, let's take a look at you. Let's take a look at the ecosystem you live in, and let's take a look at what are all the possible avenues we could choose to help you get through this in a way that provides, longevity and quality of life for you.
Well, and that segues into the first thing I wanted to talk about is, when you think about the role that medicinal plants have in healthcare and medicine and just overall lifestyle wellness, what's your sort of approach and philosophy there? How do you see all of that fitting in?
Well, I think maybe it's good to kind of look at a spectrum that I see, which there's a spectrum of medicine, which really starts with, anything from meditation and prayer, and goes all the way down to things like radiation and surgery. And in the middle of this sort of pyramid, there's all these different modalities, everything from acupuncture and chiropractic work and massage into, food as medicine and nutritional agents, and then into botanical medicine as whole agents and formulas, and then, botanical extracts, and then isolated botanical compounds, and then into, drugs and things. And, to me, it's all part of medicine. And my focus is on the plants because I really think of plants as medicine, and I think of all of life as medicine at some point. Everything is medicine at some point, but plants have a really unique place because they're very familiar to us. It's our microbiome, our microbiota are just so, perfect for working with plant compounds as food, right? I mean, we take in complex mixtures of molecules that we would refer to as, air, fluid, food, plants, medicine. And so there's a place where food sort of bridges into like a medicinal food, and then it moves in from there into like a plant medicine. And from there it goes into potent plant medicines that have a narrow therapeutic index. So I think of plants as really being on this spectrum. And for me, they're the foundational medicine for us. It's our choices every day. That's the medicine that we need to make sure is supporting us and not hurting us. And the more we move from that spectrum, from just sort of that food end to that, more directed and physiologically changing medicine, then the more, we want to be specific about our choices and understand the medicine and how to apply it to a particular person, right?
Well, I think that's really important to illustrate, because the way that I think most people interact with food and plants and medicines and everything is, we tend to grow up thinking of things of like, well, food keeps you alive. And,
right.
Dietary supplements help fulfill some nutritional deficit or whatever. And then you've got pharmaceuticals, which are the real medicine. And, and, what you're illustrating is actually the whole spectrum is influencing your pharmacology, your physiology, and it starts to put much more emphasis and responsibility on a person to manage how they're engaging with all things in their life, and not just thinking of like, I take medicine when I'm sick. but more preventative.
Well, as we expected, we kind of jumped right into a pretty critical topic, I think, in the realm of health and disease, right? And that is just daily choices. And if we think of everything as medicine, or, you know, the dose defines the poison, right? I mean, right. You know, even like you think about music, like, you know, some music is very uplifting and very just focusing and enhances cognition, and other music can be very dissonant and, you know, and that's all okay. It's all choices. But when it gets into what we put into our bodies, I think if we could just, like, I think you said the word was responsibility, you know, take responsibility for our everyday choices and how they affect our health or disease. And that idea that you said that, you know, as a kid, you started out with, there was food that kept you alive, and like there's like supplements that could give you some nutrients, and then there was medicine, which is pharmacy, right? I mean, within that model, there's no responsibility on you to make any choices, you know, about your health. And so we're basically just supporting a pharmaceutical mechanism of disease, of treating disease. You know, if you make the wrong choices, you're going to get to the point where you need pharmaceutical medicine. And that's just kind of the way it goes, right? And the reality that we see is that there isn't a clear distinction. It's basically, if you make good choices along the way, and you have, of course, you got to have good genes, you got to have a decent constitution. There's factors that can come in with, but if you just consider everybody sort of on a level playing field, the choices that we make are going to direct us towards, you know, needing those pharmaceutical medicines or never needing them, you know.
Well, and the whole concept of diet, I mean, it's changing now, but all throughout my upbringing, I'm sure yours too, the focus is on, I don't want to be fat. And so you diet to control your weight, not your health.
Right.
Which are very different things.
Absolutely. And I mean, and like all these traditional medical systems looked at different, you know, archetypes, or, you know, sort of in the Ayurvedic system, they would call it doshas, right? The idea that some people are more kaphic. They have a tendency to build more tissue, you know, like, in that some of that might be a little bit of layer of adipose tissue, right?
I'm one of those people.
Yeah. You know, and again, it's not a matter of like, the optimum health of the human being looks like this, and you've got a picture of Arnold Schwarzenegger, you know, flexing his muscles or something like that. That's right. You know, that's not the real story. The real story is that everybody has a place that works for their constitution, for their genetics, for their microbiota, and how that all works together to provide for them, you know, health.
Right.
And I think you're right. This idea that, you know, being fat is a bad thing is very undermining itself.
Well, it sends you on the wrong track.
Exactly.
Yeah.
You know, instead of, it's just like, hey, you know, making healthy choices. How do you make healthy choices? Just teaching people to make healthy choices. And I think there's a lot of, you know, sickness that goes into, and mental sickness. I think mental health is such a huge piece of what we're suffering from. You know, we look at the physical world and our bodies and how we're, you know, sickness and cancer and everything else, but it's really the mental health, right, that's driving it all.
Well, and it's all connected, right? I mean, the gut brain axis
absolutely.
And everything. You know, we now understand that what's going on in the gut is influencing the central nervous system. You know, all of these different pieces, and vice versa. There's this bi-directional communication going on, and there have been studies looking at things like schizophrenia, bipolar, autism, all sorts of different things that show very strong correlations between gut health and how those conditions present themselves.
Yeah.
And, you know, while you and I might be exposed to that research and, you know, kind of view that as, at this point, kind of common knowledge, I don't think it is to a lot of people, you know, just how intimately those things are connected.
Those old patterns or old habits die hard, right? Or die slowly, you know?
Yeah.
I mean, the whole field of neurogastroenterology has been just so fascinating, because not only is it this bidirectional communication, but we've found that in the vagus nerve system, right, which is this deep endocrine control network, right, which is just basically setting us up for, you know, balance and homeostasis, that 95% of the signaling between the mesenteric, you know, neurological system, the network there, is going from the gut up to the brain. That's pretty fascinating, you know, because I've always felt like that my brain or my mind, it's here. It's not up here. Like it's being processed here and being filtered and being put through all kinds of channels that I've created through my life experience. But there's a place in my gut that I just either know something is right or know something is wrong, you know? And I'm always trying to find out how can I tune back into that. And it's just something that I think a lot of us know. And we're talking about these old patterns and people haven't studied the research yet, but I think most people can agree that a lot of big decisions in their life
are gut decisions.
If they got it, if they feel it here, they really feel it. They really know it, you know? And I think that, you know, just bringing back to your original question about, you know, how do I view medicinal plants or how do I think about botanical medicine? And I think it's a major ecological influencer. I think you and I both talk about ecology and ecosystems a lot. And I think that might be hard for some listeners to just get, like, what are we talking about? And I really just mean inside the body, what is the material that we're bringing through the digestive tract, which for all intents and purposes is not in our bodies. It's actually a separate container that we carry around with us so that we can hold the material that we are, you know, then sticking our roots into like a tree with the ground to pull the nutrients out. And we like to carry some around with us so that we can be mobile, right? And not be a sessile plant, right? Because a plant
it's important for survival, so that we can go periods of time without eating.
Exactly. You know? And you know, plants are sessile autotrophs, right? They just sit in one place and they're nestled into the soil. And that's a beautiful strategy, and it works really well for them. They send out their seeds and spread their pollen and all that stuff. It's beautiful to watch that happen. But you know, where we are is taking the energy that they've produced as plants, eating it, carrying it in our bodies, and being able to move around and do things. And I think that's really an important piece to understand for people, that we are doing the same basic thing that all the other life forms are doing here. We're just basically a tube in one end and a tube on the other end, and in the middle we have the stuff that we're digesting. And that's what I mean by ecosystem, is that it's not like here's a tube and I'm putting some stuff in it. It's like, no, here is a very complex, seemingly infinitely complex network of cells, you know, immunological cells, things that are happening in my blood, you know, all kinds of my blood, all kinds of growth factors and neurotransmitters, and, you know, just a huge amount of structure that makes up me. And that's interfacing with this microbiota, this what we call the microbiome, which is really the DNA of those microbiota, right? But what does that look like inside? Is it's filling, you know, that whole internal canal of my digestive tract with life, and that life is then interacting with the ecosystem, which I'm calling, which is the material I'm putting in the sense of the neurogastroenterology, right? And it's that balance that really has a huge impact on how we think, how we feel, our mood, you know, getting upset easily, you know, being calm.
Your tolerance for stimuli.
All that stuff. And so, you know, when I go back to kind of my study of plants, and, you know, I was going to be a medical doctor. That's where I was headed. I was a, you know, biology student, and I went into pre-med, and I did 3.5 years at the University of Minnesota. I had 2 offices on campus. I was a TA. I was so into it. But at some point I really started to see that the single target, single molecule pharmaceutical approach was an approach that was based on treating disease, not on healing people and healing our planet, you know, healing the way we live. And that's where I shifted. I went into permaculture, and permaculture on this sort of macro scale of how do I live in a way that doesn't leave a bunch of byproducts that the rest of the people have to deal with? How can I live in a way that's clean, you know? And so I started studying sustainability and permaculture. And then when I got to Chinese medicine, it was all about, wait a minute, this is permaculture in the human body. I'm developing the sustainable ecosystem. And the thing that stands out to me is that what makes us healthy is diversity, right? It's not any one bug. It's not any one combination. It's what a diverse microbiota looks like in one person that support like woo-woo stuff, right?
Well, it's all about the language. Like what people have to understand is you're using a language to communicate something that's very hard to communicate in the first place. Like it's, you know, you're using language like hot and cold and cooling and heating and all these sort of things, but really what you're describing is something much more complicated and deep that it just wouldn't really make a lot of sense to try to tease that apart into like a really complicated statement. You know, we have to be able to talk about things kind of quickly, and it makes sense to people, even if they don't understand the details of how that works, which one reason why I like talking to you is because you do understand a lot about the, you know, the pharmacokinetics, these different things that are going on, like what you mean when you say something's cooling or heating. It's like you can get into that if you need to talk about, you know, what's actually happening in the body on a really complex level. But for most people, that kind of language is not helpful to them. They need something that they can digest themselves, and so that language is kind of intuitive, like people can relate to kind of quickly. And it's important to have that balance, to not just be able to speak the language, but understand the science and details of what you're talking about too. But yeah, I totally see that as it's a language game more than anything that people kind of have to understand.
Yeah, and like I said, it's, you know, the word heat in Chinese medicine has a very specific, you know, it has a specific meaning within that system.
Right.
Know? It's not just like you think of heat, you turn on your burner and your stove, you know, or your thermostat, you know. It's a little bit deeper than that. And it encompasses a lot of modern, you know, physiological concepts, right? Mm-hmm. Things like inflammation, you know, exactly. with you know, redness and things of that nature. But, you know, before going too deep into it, just my goal mostly is to try to help people see from that macro ecological place how you can look at a person and see how their ecosystem looks, viewing them. Right. Like I said, their nails, their hair, their eyes, their skin, their lips, their, you know, the way they talk. Yep. All those things are information for me about that person. And then if I understand how the materials in the natural world relate to, let's just say hot and cold.
Right.
I could pick something like silver, right? Which is a very cooling mineral in the sense that it puts a damper on inflammatory processes. It shuts down that infectious, you know, kind of heat condition that we see in people where there's pus and there's redness and there's yellow colors and things. It does a really good job of that. But taking that into the plants, they all have all these different what we call natures and flavors. And so those have been determined and settled on and, you know, and thought about and deliberated on for literally millennia, you know, by doctors who cared about their patients, cared about their villages, and, you know, cared about the natural materials and how to apply them in an ecological way to the person, right? And to that constitution. And so back to that idea of the microbiome and how do we alter the way that people think and feel and make good choices? Well, plants have an incredible power and a role to play there that I think is really unique. And I think it's overlooked, you know. One of the places I see, you know, a lot of people heading in sort of this health revolution is into functional medicine, right?
Right.
And functional medicine is, you know, we have David Jones here, you know, right in Ashland. He's one of the founders of functional medicine. I've sat with him a number of times. Michael Stone is one of their educators. And so I've talked to these folks in functional medicine, but they tend to apply the nutritional concepts and the, you know, like looking at the different microbiota that are present, for example, looking at, you know, what's in the body as far as molds or toxins, Lyme disease, you know, fungal infections, whatever, and then applying, you know, very specific molecules still, you know, and then looking at relationships in lab work and things. But the missing piece is the plants.
Right.
And a lot of these folks do food, but I do plants and I do medicine. I like plants as medicine as a foundational medicine for the world. And that's where I think we should be. I think we should be using plants as medicine from everything from how we grow it and farm it and live with it and know it to sharing it and extracting it and making it a medicine to how we take it and use it in our lives to create balance, you know, in our ecosystem.
Yeah. Well, and bringing this all around to cannabis, where do you see, you know?
Oh yeah, cannabis, right?
Right. Well, and the thing I love about cannabis I've mentioned before is such a good springboard into getting into these deeper topics that need to be talked about and that I think is really important for people to understand as cannabis is getting so popular and people are jumping into it as, you know, folks are recognizing the therapeutic potential of cannabis and are really rushing towards that. And, you know, I was speaking with somebody just the other day where we were discussing that one of our concerns is that while people focus on cannabis, they lose sight that cannabis is one component in this medicinal plant toolbox that you and I have talked about, the toolbox model. And so can you speak a little bit to where cannabis fits in to this overall scheme? And then maybe we can get into a conversation about how cannabis affects the body, the endocannabinoid system, and all of that.
That sounds great. I think, you know, cannabis is, of course, shrouded in all this, you know, prohibition and a lot of political, you know, struggle and
a lot of different energies
surrounding it. It's gotten into a darker place, even like just sort of the information we have about it. You know, I like to start always when I'm looking at what is the benefit of a plant or what are the weaknesses of it or the potential, you know, dangers of it. I want to see what is the record of that plant look like across traditional cultures. And that's my number one form of evidence. Again, it's not like, people talk about, oh, it's woo-woo, it's folk medicine. Whether you call it that or not, those people were carrying oral traditions and written traditions about medicines to help their people that they cared a lot about. They didn't have time to sit around and go to the movies. No, they were living, and life was a struggle for a lot of people, and medicine was really important. So there's a lot of heart in it. There's a lot of intention in it, and there's a lot of empiricism. So I always go there first. And what I've done with cannabis, in my kind of digging into what is cannabis as a medicine, I've had to cut through that same kind of shroud and sort of darkness of like, what really is it, when looking back at what are the records from Chinese medicine and the use of cannabis. And I've looked deeply into it and tried to find what are the threads. Because most of the time in school, in medical school, with Chinese medicine and the doctors from China stuff, it's just kind of, we don't talk about it. You of course, the hemp seed, everybody knows that's
nutritional.
And it's generally, there's a couple of great formulas that use ma zi ren, and ma is the word in Chinese for hemp or cannabis, right? For marijuana. And so ma zi is a seed, and so you got ma zi is the seed of the cannabis plant, right? And so that seed has been used really successfully for moistening up the bowels and helping with constipation. And you combine it with other herbs that have other, peristaltic actions, or, pull some fluids into the large intestine, and then like, mirabilitum, things like that. And then those will all work to kind of create this bowel. But beyond that, you start digging into what was actually cannabis used for as a medicine. And was it cannabis in the sense of, THC rich, or was it cannabis in the sense of hemp? Was it ragweed? It's really hard to determine that from the records that we do have, but there is some consistency across the writings I've examined in using cannabis for conditions that involve, something in Chinese medicine called wind, which I've talked about with you before, which is like the idea that there's an instability in the body. So there's a homeostatic, stabilizing kind of effect from cannabis, where, it's instability in the mood. There's things like tics and twitches and tremors and dyskinesias and things of that nature that cannabis was really good for, that it's been recorded that people would use it for that. And again, it's hard to understand, were they using CBD at that time?
Right.
Or was it a strain that was blended? And we don't really know. The records are hard to follow. But clearly it was used for problems of the mind, people having anxiety, schizophrenia, people that had, mental instabilities. It would really help to bring back some balance to them. And, one of the things that I saw that people used was they made a cannabis wine, and it was made, so they would take it was like more like a cannabis wine extraction, I guess, but they would take their rice or sorghum wine that they would make, right? They'd ferment those grain, the grain, and create, an ethanol rich solution. And of course we know that, ethanol is a great way to extract cannabinoids, right? And so they would take the seeds with the coating on the seed, what do you call that? It's not the calyx, but it's actually, there's a coating around the seed as it sits on the plant, and that coating is covered in little hairs, right, little trichomes. So it's got a lot of cannabinoid on it. But so they would put the seeds with those little trichomes into their solution, and they'd extract it, and they'd give it as this cannabis wine, and they'd give it to patients for different conditions. So it's clearly been used that way, and it has it's very safe medicine. But I think that part of the problem is just that we have a hard time understanding with this plant the differences in its phenotypes, in the different types or cultivars of cannabis, which is one of the things that makes the plant really unique. You can get a very different physiological effect from one strain, right, than another. And that's, and there, to some degree, some plants have that, but very few, and to that degree. And I think it's one of the things that makes cannabis unique and fascinating to me, is just that, God, there's so much. And we talk about, what does cannabis do? I'm like, well,
man, yeah.
Which part of cannabis? Which cannabis are you talking about?
Yeah, how are you using it? Yeah, and it's one reason why cannabis has caught the eye of a lot of pharmaceutical companies is because it's a very effective biochemical factory, as far as its resin content and the types and diversity of compounds that it produces. And then when you start thinking about how that can be manipulated either through breeding or genetic modification or whatever, there is just some massive potential to use cannabis as a tool to create all sorts of different chemical compounds in a much more efficient way in much higher concentrations than most plants are able to do. So it'll be interesting to see long term what cannabis becomes, because now that hemp is legal in the United States, and now that THC-rich cannabis is gaining acceptance in the United States, at least on a state level, we're going to see very interesting things, I think, as far as where people take the plant and how they use it. And, we're still contributing to this millennia long record of medicinal plant use. And so we're entering an interesting time now where we're kind of coming out of the dark ages with cannabis and now generating a lot of information, a lot of data that, people that come through 100 years from now will be able to probably glean a lot of interesting wisdom that we may not be able to perceive directly, since we're kind of in it right now, trying to tease some of this stuff out. Different cannabinoid ratios using different parts of the plant. Even on the beyond the medicine side, how cannabis can be used as building materials and contributing to fuels and all sorts of stuff that and being used as a bioremediation tool. It'll be interesting to see what we learn.
Yeah, and I think, you were talking about the toolbox, and what is how does it fit into the toolbox? And I think I find it difficult to place it because it's been shrouded and because it's in the darkness, because we don't really know which part of it does which, and we've been kind of thinking of it more in molecular terms, right? We're thinking about THC, we're thinking about CBD, we're thinking about terpenes, sometimes thinking about flavonoids.
Right.
But in general, we're kind of thinking about it in these molecular terms, whereas with most of the plants that I use, sure, I like to make sure that the plant is a potent example of that plant species, that it has the medicine I'm looking for in it. So I want it standardized to some percentage of an active.
Yeah.
Not because that molecule is the thing that's doing the work. We found in a lot of studies that the molecule actually alone doesn't do the work. It needs to be the molecule in the matrix of plant compounds that represent that medicine. And so I think it's sort of changing that perspective as a plant on how we identify that medicine, how do we use it. And I think a lot of people are, I've heard this sort of approach from a lot of folks who are deep in the cannabis as medicine field. It's that, what we really need to do is just take apart the plant, pull it into all its pieces, and then select which pieces we want to put them together and apply it to the thing that we want to treat. And that's sort of, that's kind of like Franken medicine. Like, it's not
reverse engineering,
and reverse engineering can be really helpful. But I think we have to be careful here, because it's hard to understand what you lose, from like a purely molecular analysis.
Yeah.
What do we actually lose about, and maybe I'll just say this, that the spirit of the plant, and that's, again, getting into some woo-woo territory, but you know me, I'm going to bridge spirit to the deepest reductionist science.
It's kind of your like role in life, is bridging those worlds.
Bridge
together.
those worlds. I love it. That's and it's really important to me, and I think it's profound for our culture to recognize that those two things exist simultaneously.
Yeah.
But so that there's a spirit of the plant and there's a spirit of it, and I think with cannabis, it's got such a long, rich history. And you think about India, and you think about the Ganges, and you think about this sort of like the steeping of that plant in this spiritual path to enlightenment and what it represents. And and sort of there's a whole part of it too with the sadhus and the sort of like removing yourself from society and culture and like going other, and there's a lot of stigma and there's all this, but it's just really wrapped up in so much of who we are, and we've evolved with it for so long. I think that the plant itself is a very sensitive creature. It's a very sensitive, I don't know what I call it, like a life form, and I think that it picks up energy, and I'm talking again on these little bit more quantum or esoteric level about how the way that it's grown, the way that it's cared for, the way that it's harvested, the way that it's being ingested. Right. I think those things are exceptionally important with cannabis because of its deep connection to our psyche, our spiritual path. You know what I'm saying? I mean,
well, yeah, to put it in more practical terms, the way a plant's handled changes its chemistry dramatically. The way that a plant like cannabis is administered in the body changes how your experience with it, is going to be, whether you're eating it or inhaling it or how you're preparing it. It's all influencing these different things. And because a lot of the compounds that cannabis makes are in the growth cycle, just thinking about terpenes are so influenced by environment, and cannabinoids to a lesser extent, but still somewhat. And so how you process that plant is going to affect that chemical profile, how you store that plant, how you then later process that plant into a product that you're going to consume, whether it be foods or teas or whatever you're going to do, has that influence. And then getting into an equally complex area, trying to understand the endocannabinoid system tone of a person and what influences that, their lifestyle, their diet, all of these things. The way that you live is going to influence how you respond to cannabis, because it's changing all these things. And so, on my side, like when I hear you, using that language, the sensitivity to the energies and everything, to me, that's just because of my hyper like analytical chemistry, side of things, and thinking about my experience with all of that. To me, that makes sense, and I can understand that from my vocabulary and my perspective of how I see it. To me, that gels together. And
like you can see it in that sort of reductionist view.
Yeah,
right?
you can see
when
it.
You look down, when you break down to the parts, you can see the parts have changed, right?
Right.
Based on the choices that were made, and of course we're back to the ecosystem. Back to that permaculture idea. And so, how do we take care of the plant? And I think, just like I'm in Jackson County, Oregon here, right? There's 9,000 acres of hemp in this county,
in this one county
this year, know, and when you see these farms, they almost every single one of them is using sheets and sheets of single-use thin black plastic.
Yeah, huge monoculture farms.
I'm like, again, back to the energetics of the plant, and the plant itself, it's a representation of how we are acting. We're trying to make this medicine to heal, but then really a lot of the drive is for the money, and that unfortunate relationship between money and medicine has gotten us into all kinds of trouble.
Well, and it comes down to like, what do you really value and how are you making those decisions? And to touch on something you said a minute ago that ties into this, of like, we don't know all of the little things about the chemistry of the plant that are influencing therapeutic outcomes. And there's research in Israel that is demonstrating this in a very direct way. There's a researcher, I think his name's David Miri, if I'm pronouncing that right, that has a lab in Israel with like several dozen researchers there that are exploring all of these minor constituents of cannabis. And, they're not just characterizing a dozen or a couple of dozen cannabinoids. They're looking at, 50 to 100 cannabinoids in products and seeing how little manipulations to those affect therapeutic outcomes. They're looking at, hundreds of terpenes at a time and trying to understand those differences. And, the wisdom that they're gleaning is that, this is a very complex picture here, a very complex puzzle that we're trying to piece together. And that there are a lot of components of cannabis that, right now in Oregon, if you go to a cannabis lab, you're not going to be able to get quantitative data on, the 100+ cannabinoids that are in the plant, or the, the hundreds and hundreds of terpenes, the several dozens of characterized flavonoids, the stilbenoids, the sterols, all these different things that are interacting together. And there's research now that shows that flavonoids interact directly with the endocannabinoid system in a very direct way, which then has implications for all plants and how they're interacting with the system, which then ties into, and this is just one example, but how that ties into, well, what you're eating is going to affect your experience with cannabis, and because it's influencing your endocannabinoid system tone. The puzzle is super complex, and we have to be really, really careful about how we're making decisions about what to value. And in these larger agricultural models, it's like, well, we're valuing CBD content, THC content, maybe a few other cannabinoids, resin yield, bud structure, picking a few choice variables, valuing those, and then creating high intensity production models to churn, that out, and sometimes not recognizing that the consequence is sacrificing the unknown and possibly unknowable variables that are getting lost in that, some of the more sort of craft cultivation, that's kind of what's been called now, the craft cannabis, world, where people were putting a lot of time and energy into much smaller numbers of plants, having a much more intimate relationship with those plants, understanding how they're changing and cultivating, handling them very delicately as they're being processed. Those products are very different than what gets churned out in high intensity sort of big ag, models. And I think there are places for all of this to coexist. I don't like seeing huge monoculture farms, period. And so, I've studied permaculture, so it's good that we have that common background. And my background in ecology, because before I got into all of this, I was working with the BLM, working on restoration projects, trying to understand native plant populations, the roles that they're serving in ecosystems, and kind of how we need to think about how to restore ecosystems that are in the process of falling apart, essentially, usually because of human activity. That thinking really needs to be integrated into cannabis, because cannabis has this such rich opportunity right now to do things differently than as has been established in big agricultural models for corn and wheat, all these. Yeah. And, unfortunately, the momentum is coming so fast and there's so much money to be made that it's like you're swimming against the current. But it's important, I think, to keep pressing that message and highlighting. And I think my point of saying all of this is, even with a reductionist model, the research, I think, is going to prove that perspective worthwhile and very valuable, that there are these hundreds, thousands of compounds being influenced and getting lost, and the therapeutic outcomes are not going to be the same.
Well, this all ties back to, how does cannabis fit into the medicinal plant toolbox, right?
Yeah.
And it's a complex answer, and I think there's a lot of folks out there. Well, and I'll say for me, I like to use cannabis a little bit separate. Most of the plants here, I'd like to think of them in terms of a formula.
Yeah.
How would I formulate that plant with other plants to make a medicine that's good for a person, for a condition?
Right.
And with cannabis, in the realm of topicals, I've been able to do that, much more easily, integrating cannabis in with topicals as far as mostly pain relief or inflammation control. But when it comes to, giving people an herbal formula to treat their constitution, I generally have cannabis as a separate tool. It's over here, and I can add it to what I'm doing, or I can, bring it in as a synergist if I wanted to help somebody sleep. Maybe they'll bring in some CBD to help them sleep, or if they're having anxiety, I could bring in some CBD, but also have a formula of herbs, that would be there to help them sleep as well. A nervine tonic, with things like Scutellaria, lateral flora, the American skullcap, and passion flower and things of that nature, kava, piper, things that are just going to be nice and calming, and then just bring the cannabis in separate. And again, I feel like I'm kind of playing in the dark with cannabis because of what you're talking about, because it's such a diverse plant with such a diverse array of constituents that then impart a diverse array of physiological effects that it's just hard to know it. And so, I feel like these sort of, I think I use the word boutique, you use the word craft, the kind of like the smaller care for medicine